morning, everyone. Welcome to session 4, the physiology and metabolism session. So we're going to start this morning with the first presentation, and then we'll follow that with a special presentation by the Minister of Science and Higher Education. So it is my great pleasure to introduce our first speaker this morning, Professor Eugene Ponomarev, who will be telling us about metabolic reprogramming of macrophages towards the regenerative phenotype.
Minutes. After 15 minutes, minister will come. When minister will come, he should finish it.
Oh, so we'll stop.
We will tell to him.
Okay, can you hear me? Okay, good. Just first clicker. Actually, first of all, I would like to thank organizers to give me opportunity to present my work. And actually, I'm not metabolic research biologist, I'm immunologist, so I will talk more about macrophages and how to treat inflammatory diseases. And actually, the topic I've been interested actually for many years, for I would say 15 years, maybe even longer, is about macrophage polarization. Actually, it's similar to Th1 and Th2 cells. Macrophages might have actually two types of activation. Classic type of activation, when they have actually very pro-inflammatory phenotype. And typically it's induced by bacterial products or Th1 cytokines such as Infer and gamma. And also there's alternative type of activation. What is important when you look at the gene expression pattern in classical macrophages versus alternative, you would see that alternative macrophages, they have more like anti-inflammatory phenotype. They're important for tissue repair and homeostasis. And that's important because you can try to use it, for example, to switch from 1 to 2 to deal with certain chronic inflammatory disease. But what is also became quite interesting to us recently that if you look at the metabolism of classically activated macrophages, they mostly use glycolysis, which means they don't require mitochondria. They just lyse glucose and make actually 2 molecules of ATP. Whereas alternatively activated macrophages, they actively use oxidative phosphorylation, which require mitochondria, oxygen, and fatty acid oxidation. And that's quite interesting because it can be used actually to try to switch metabolism of macrophages and control switch from M1 to M2. And one of the switch is important, I just try to illustrate it here. It's just a wound healing, like a skin wound. And what happened actually in the beginning, actually first stages. Yeah, first stages of the wound formation, you have actually blood coagulation and inflammation. Then you have basically first stages of post-injury tissue formation. Then you have tissue regeneration, remodeling. And during this process, you have gradual switch from M1 to M2. And this switch is extremely important because during certain type of pathologies, for example, diabetic wounds, which often lead to amputations, you don't really have a switch from M1 to M2. Also, there are some chronic autoimmune diseases, for example, like multiple sclerosis. You don't really have the switch. And you have actually inflammation lasting for a very long time and very extensive actually tissue damage. Actually, we started this work at Harvard Medical School quite a while when we've been interested in basically finding certain factors which can basically change a balance towards M1 to M2. And we found that certain microRNAs like miR-124 was very important for that. And later in Hong Kong, we continued our research and we found that there is a specific actually drug which came from Indian plant. It's called forskalin, the plant called cholinol, and actually this plant acts through CMP pathway, upregulate miR-124. And when you use a forskalin, I try to treat experimental autoimmune encephalitis, which is a model of multiple sclerosis. You can see that several injections can completely shut down the disease. which means reprogramming from M1 to M2 is really important if you want to treat very, how to say, untreatable autoimmune diseases. So now actually we've been interested in some other factors which can promote such a transition from M1 to M2, and that we found actually quite by chance that we found that different batches of the serum promoted M2. Probably many people who work with M2, especially with the human cells, they realize that it's very difficult to do it in vitro. Sometimes polarization works towards M2 very well, sometimes it doesn't work very well. So we found it really depends on the quality of the serum, or even from the, depends on the company or batches of the serum. And we did actually quite a simple experiment. We just use a fetal bovine serum, adult bovine serum, adult human serum, adult mouse serum. And we use actually here mouse macrophages. And you can see here that if you just use serum-free media and use IL-4 only, the most important marker for M2 arginase is not upregulated at all. Then FBS has effect, and adult bovine serum has much better effect, and the most dramatic effect has a mouse serum, which again was surprising. These are mouse macrophages. And we look at the arginase expression on protein level, and also found that indeed you need a serum for regulation of this marker. And same true for arginase enzymatic activity. So then we try to basically figure out what is this factor. So we did actually a very simple experiment. We did a fractionation. We took actually high molecular weight fraction, which is higher than 100 kilodalton, and low fraction. And we found that for both FBS and ABS, high molecular fraction, which is more than 100 kilodaltons, actually has this effect for arginase expression. Also what we did, we tried to use actually FBS low fraction as a supplement for growth factors, because most of the growth factors are less than 100 kilodalton, and it's a very good scheme. You use supplement of FBS low as a source of the growth factor, and ABS high as an active source or active component that's induced M2. And basically what is shown here. Here we made actually bulk RNA-seq when you use macrophages activated with IL-4 alone, ABS high alone, or both IL-4 and ABS high. And what was quite interesting that most of the genes which are related to M2 dependent on both ABS high and IL-4, although some of the genes also, M2 genes dependent on ABS high alone, but very few genes been only IL-4 dependent, which basically we figured out from this experiment that indeed, actually M2 activation is more complex. You have something which acts from the serum and also from IL-4, so basically these are two pathways which are involved. Here what we did, we just decided to see whether blood coagulation is required for this effect for M2 activation in serum. Because remember how you make a serum. You basically take blood, you put it in 37 degrees for several hours, then centrifugate, and during this time blood coagulation happens. To check it whether blood coagulation is required or not, you can use a plasma. And you use sodium citrate as a basically agent to prevent coagulation. And we found that if you use adult mouse serum, you have very nice effect of arginase upregulation, but if you use a plasma, effect actually almost negligible. And that was clear result that we do need blood coagulation for M2 activation and just basically for basically expression most of the M2 associated genes. It took us actually for a while to figure out which factor in a serum, again it's high molecular weight, it's more than 100 kilodaltons, until we found that this factor mostly related to lipoproteins, HDLs and LDLs. And it was quite interesting that for bovine serum, both LDL and HDLs had effect, and for mouse serum, mostly have effect for HDLs, which is again not surprising because mice, they have very little amount of LDLs in contrast to human or bovine. Here we did experiment when I tried to just look at isolate HDL from the blood, and we treated with different enzymes, because HDLs, they contain proteins on the surface, they contain lipids, and many other agents. And we tried to treat our HDLs with the proteases, glycosidases, which removed basically all the sugars, and the effect was still there. So they realized that the only component which is left in the HDLs are lipids, and we created liposomes from these HDLs, and liposomes again gave us 50% effect, which means mostly lipids mediated this phenomenon. So here we've been interested what happened during blood coagulation, because in blood coagulation we have many components. We have granulocytes, we have lymphocytes, we have platelets. So we isolated all of them, different subsets of the cells, and activated them with a thrombin. So the thrombin is the main activator which become activated during blood coagulation cascade, and has an effect on many cell types. And the result was actually quite interesting, because we thought mostly platelets mediate this effect, and we found that mostly granulocytes or granulocytes-derived microparticles mediated effect basically of arginase upregulation. Here just a model. We found that during the blood coagulation, the granulocytes become activated, they release microparticles, then exchange with HDLs, for example, from the plasma, and all the lipids like triglycerides and fatty acids get loaded on them and get actually observed effect. Here we just try to check whether you need a serum to basically upregulate OX4s. I already mentioned that M2 macrophages mostly use oxidative phosphorylation, and here quite complex diagram, but this is basically shows the higher, you have a graph, the higher level, you mean this, especially this area, the higher area, the better oxidative phosphorylation it is. It's basically based on oxygen consumption. And you can see that the best effect you have when you have IL-4 and a serum, whereas the plasma does not give you anything compared to control, even plasma with IL-4, which means it just shows that to stimulate OX4s and M2, you do need these HDLs. And finally, we decided to check what is on HDLs. We know that the lipid loaded on HDLs promote this effect of M2 polarization. So basically, we performed targeted global lipidomics and tried to compare plasma versus serum, and basically plasma versus granulocyte-derived microparticles, and found the most of the difference when we tried to compare one group after another, that indeed serum and granulocyte-derived microparticles mostly enriched with a triglyceride, diglycerides, and fatty acids, which again is an indispensable component for oxidative phosphorylation. So finally, we just look at the receptors and the pathways downstream of these HDLs. Actually, it's known that HDLs are bound to scavenger receptors, and indeed we found that scavenger receptors, actually SRB1 and SRB3, are important for that, and we can block it using blocking antibodies. And when we look at the kinetics downstream of the scavenger receptors, we found that most AKT pathway is involved. Especially here, you can see quite well that IL-4 alone give a very fast spike of AKT phosphorylation, like 15 minutes of the peak, whereas ABS give you actually very long-term effect, like 24 hours. And both of them give actually very nice M2 polarization, which we typically checked 24 hours or 48 hours after we added IL-4. So here's just a picture that I can find from the manual, that during the blood coagulation, we have actually neutrophils, we have platelets, and we have macrophages which have actually a homeostatic phenotype, making actually many factors to promote tissue remodeling and repair. So we modify this picture. Now we know that thrombin during blood coagulation activate both platelets and neutrophils. Neutrophils actually release lipid droplets, they're loaded on HDLs, and make this M2 phenotype. So finally, just a conclusion that If you try to polarize macrophages towards M2, if you have just IL-4 alone, they don't really get really M2. They only upregulate 6 out of 32 M2 related markers. But when you have IL-4 and HDL with fatty acid complexes, which are released by activated granulocytes during blood coagulation, you do have actually a real M2 phenotype. So thank you for your attention. We do have my laboratory, mostly Tatiana Veremeyka did this work, and of course we have collaborators from NU, Hong Kong, and Oxford University. Thank you.
All right, thank you, Eugene, for that fantastic presentation. We're going to hold off on the discussion for now because we're going to have a very special presentation, so I want Das to come up here for the introduction.
Thank you, Dave. Thank you, Eugene. As I was telling yesterday in my introduction that science is a priority in Kazakhstan, and yesterday our Minister of Science and Higher Education was overlapping with a major event, so he could not come. But today, so we would like to invite to the podium our Minister of Science and Higher Education, Mirza Sayasat Nurbek. Please, greetings from Minister of Science.
Good morning everyone. And it is a pity I missed the opening. We had a big session yesterday. You might have known in Astana we have the Convention of World Religions. So from religion to life sciences. So yesterday was religion, the essence of life, the meaning of life. Now we're talking about essence of life. And I'm really grateful to President of Nazarbayev University, Waqar Ahmad, for supporting almost 20 key conferences in different areas. And I missed most of them, unfortunately. There was this great conference we just had in mathematics, right, which is kind of Oscars for mathematics, and we had about 700 participants, top mathematicians, top researchers, top scholars. So please do put us in loop. It is really important for us to show our support. I'm really thankful to Professor Sarbasov. And this conference today coincides with 2 milestone events, 2 milestone anniversaries. We're celebrating 10th anniversary of Astana National Lab. We are celebrating 15th anniversary of Center for Life Sciences. We're very young, but what happens in research at the moment is AI is giving this new drive and opening new frontiers for specifically biomedical research and medical pharmaceutical research. I just, about 3 weeks ago, I was in China for this Shanghai Cooperation Organization Summit, some call it historical, and I had a chance to meet the founder of DeepSeek. Very interesting fella. He has 2 principles. He said, I only hire graduates of top Chinese technical universities, Jiaotong Polytechnical Universities, and 2, I do not hire people with more than 2 years of experience. For radical, disruptive innovation, it's his personal belief, experience is a limitation factor. He says when you talk about radical, really disruptive innovation and disruptive ideas, experience actually doesn't help or even hinders that disruptive innovation. So it's an interesting idea. But he still did come with DeepSeek, micro, large, whatever call it, micro LLMs, the new approach for structuring algorithms. And they revolutionized this happening revolution. But coming back to AI, and we believe we focus on AI. Kazakhstan puts, and our president puts AI as national strategy, which is, happening in many countries. I just came back from Seoul, and I met the newly appointed Minister of Science and ICT, and he's their top AI guy. So he's former head of AI at LG, former head of AI at SK Telecom. And South Korea has this big new ambition to become the 3rd global AI superpower after US and China. So Kazakhstan also has this big focus on AI at the moment. We're creating, we already created the new AI ministry. And a lot of countries now creating these institutions that would focus on spreading or installing AI to as many areas as possible. So biomedical research, you go to San Francisco, south San Francisco, you're just entering the city from the airport, there's this huge biomedical cluster, and all these great big pharma companies now clustering in Silicon Valley. You'd see, AstraZeneca's, and AstraZeneca is in Cambridge. There are major campuses in Cambridge, again, tapping into resources of university. So there are at least 3 areas where AI can add value, or even some radical transformation. It's drug research, pharmaceutical research, by enhancing or speeding up drug discovery. And this is what Nazarbayev is doing at the moment. We're working on a really, really interesting and promising new drug. Molecular optimization, where AI can cluster or analyze big data sets and model promising molecular structures that would take months and years without, synthesizing formulas. So modeling is becoming a big new thing. Clinical trials, right? That's what we're doing right now. So we're pushing Nazarbayev research, cutting the red tape. I'm working really close with Ministry of Healthcare to help expedite everything. But AI can actually help, and there's this big new thing with wearables, where you can in real time monitor health conditions of a person, a human trial phase. You can have all these data in real time, analyzing. You talk about screening, monitoring, so enhanced accuracy. CNNs, convolutional neural networks, that's the new word, opposed to cable news network. So X-rays, MRIs, all these scans. AI is so concise now, is so precise now. You could find this new things in scanned images human eye might miss. It could prioritize, and we have great startups in Kazakhstan working in this field, having some really great success, being pitched in Silicon Valley, having a big venture capital investments and go into next stages. So AI is a big new thing, and then hopefully we can help life sciences. Kazakhstan is one of the very first nations in the world to make AI compulsory component of national curricula. So starting from February this year, every single higher education college student has to take AI courses. Out of 650,000 bachelor students we have in Kazakhstan, we've already trained about 430,000 students. And we're expanding. We want to make sure every college student is given AI skills for free. We didn't stop there. We'll start big selection, President Vakar, and we want new students and faculty members be part of that big selection in November, where we would train 100,000 students in AI entrepreneurship. And we have hired Paul Kim. He's arriving next week. 1st of October, we're going to have AI Council under the President. We'll have Kai-Fu Lee. We have all these great AI experts coming to Astana, talking about the future of AI and how AI can add value in different areas. So 100,000 students will be given seed funding. So basically, we're creating a large funnel out of our higher education system. A lot of countries now follow that model. So this September, you had compulsory AI training in China. They start at age 6, elementary school, 8 hours of compulsory AI training per year. United Arab Emirates, they start in kindergartens, 12 hours of compulsory AI training. United States, you have AI for K-12 initiative of President Trump. But now United States doesn't have Department of Education. So I was in April in DC, I said, I want to meet the secretary. I said, sorry fella, no more Department of Education. I said, well, good luck, best of luck. So it's going to be very decentralized, kind of a bit chaotic process as we predict. And many countries, they just taking this AI seriously. We have what we call constrained optimism towards AI. It's a bit hyped at the moment. It's kind of this big new word, but the impact is very promising. The added value is very promising. And we have extremes. We have a group of experts say revolution is coming. It'll be radical transformation of everything. We have more restrained experts who says, yes, big new tool, huge, might be something really, really big, but it has to be applicable. So that great promise of AI hasn't been delivered yet. It's still domain of large tech companies, of big national strategies. So I do urge to speak more about AI. It's our national priority. We're training as many AI talent as possible with hope to catch that AI wave. The deficit of AI talent is just, it's huge, and it's going to grow. It only grows. The startups, they're very promising. We have very good success stories at the moment with several startups, Kazakh startups, going to the global markets, focusing on AI and then power of AI. And we do believe life sciences, a lot of people and venture capital, they were focusing on that digital revolution. And now that tide is coming down. So there's more focus on deep tech, because digital can give you only so much. It can add value. It won't create basic goods, basic products, basic platforms. So deep tech is a new word. So we'll be focusing more on deep tech. And just for your information, President Ahmad Wakar, we have a new function now. The Ministry of Research is Innovation Policy. It's given from Ministry of Digital Development. So we'll have more resources. And we really see Nazarbayev University as this hub for innovation, fundamental and applied research. We're working on science park idea. So we'll have next phase of development will be creating of this large science park. It will be great if our participants could see the expansion plans and then feel the expertise we're trying to accumulate here. So a lot of countries understand without research, without fundamental science, without investments in human capital, there's no way you could stay within this big new wave that's coming. So countries like South Korea, countries like Kazakhstan, countries in Middle East, Saudi Arabia, they all have these big ambitious plans. Not only we want to be part of that rich countries club, we want to be what is called middle powers. And it's a very old concept tracing back to Cold War, when we had global powers. You had America, you had Soviet Union, but there were countries like Canada, France, Australia. We said we also want to matter. We want to impact on certain areas. Now there's a new term coined by South Korea, GPS, Global Pivotal State. Not global state yet, not middle power, something in between. And a political scientist by my training, so it's, I'm going to look into these new terms coming out. But Kazakhstan has this ambition, and this ambition is backed by investment in talent. We nurture talent. We bring 40 solid research-intensive university. Nazarbayev University has this excellent platform, array of partners. You would see a lot of great research partnerships, academic partnerships, and President Wakar, please tell more. But now we have universities like Cardiff, KAIST, Korean Advanced Institute of Science and Technology. So competition is growing. We have Moscow Engineering and Physics. We have University of Minnesota, 2 universities from Arizona, University of Arizona, Arizona State University, Colorado School of Mines. I'll be flying tomorrow to United States, accompanying our president. He'll be speaking at United Nations General Assembly. So Kazakhstan is country to watch. There's really interesting dynamics happening in the region. And then my last thing would be demographics. Why life sciences are so important for this region. This particular region, what used to be called Greater Eurasia, former Soviet Union, Central Asia, India, Iran, Pakistan, it was a vertical axis of Great Silk Road. So a lot of people think Silk Road was east-west, west-east, but there was also a vertical axis to Western Siberia, Northern India, a lot of trade, a lot of cultural exchange. Then came the Great Game, British. The whole region got split into two. We went under influence of Russian Empire and then what is now Pakistan and India under influence of British Empire. And what happened, there are more babies born in this particular region than anywhere in the world. We have over 2 billion people living in this particular region. Over half of them are young people below age of 25. We have one of the youngest after Africa. We have one of the youngest generations in the world after Africa. Europe has been aging for decades. Southeast Asia is now aging. So Japan, South Korea, China, they all have these problems. There are 500,000 babies born in Kazakhstan as we speak, on average. 1 million in Uzbekistan, 5 million in Pakistan, 60,000 babies daily in India. So India gives Kazakhstan's population in 8 months. South Korea's 52 million population in 1.5 years. We're talking about 36 million babies annually. 250,000 babies born in Korea. And India gives that childbirth population in 6.5 days. Kazakhstan's annual childbirth population in 2 weeks. So we're talking about a lot of people that need to eat, that need to get some education, some treatment, medical treatment. So the personalized treatment, new drugs, more efficient, cheaper, fertilizers, it's all becoming a matter of big demand. So life sciences will have this new revolution, I would say, it's back on the menu. The population of Earth has grown from 7 billion to 8 billion in 12 years. And it grew from 6 billion to 7 billion in 150 years. And growing and counting, we have about 8,232 plus million people living on this planet, and it grows really fast. So, good luck. I wish all the participants great conference, exchange of ideas, exchange of experiences. We're here to be partners of any great initiatives. If President Wakar does, if you find a good partner here and that they need extra funding, just tell me, okay? We're really ready to invest because we believe life sciences will be the next big thing, including energy research, robotics. There's a big robotics revolution coming, and AI will be speeding up, will be pushing this research, expanding, giving new frontiers, and we are creating and nurturing this massive AI talent in the country. So thank you very much, and have a good conference. Thank you.
There is an idea. Picture time? We're going to take a picture with the
Thank you very much, Minister, for that wonderful presentation. It's incredibly impressive and inspiring to see what's happening here in Kazakhstan in the life sciences. and AI. Wow. All right, so let's move on with the session. Our next speaker is Anton Vatovtov, who will be talking about beyond genomics, how proteomics is transforming precision medicine. Thanks.
Dear glorious colleagues, it's a pleasure, indeed an honor, to present right after the minister, and a pleasure to present my company today. So my company is called Zalma, as you obviously know, and we will soon celebrate our 20 years anniversary. So also what I like to highlight is that we were the first company who supplied the first genetic analyzer to Kazakhstan, and we registered many instruments for healthcare to enable the local government and local doctors to use the advanced technologies in healthcare. The topic of my speech today is very specific because just recently Thermo Fisher purchased the Olink company. Olink company was founded in 2004 in Uppsala in Sweden, and after 20 years of its existence, Thermo Fisher purchased this company. This company is well known for its proteomics solutions, so that's kind of the only solution that this company is providing, it's proteomics. Now that Thermo Fisher purchased it, and the Zalma is available and able to supply those solutions to local customers in Kazakhstan to support the proteomics solution that will be established soon here. So the company is indeed very well known. There are many different articles, and there were multiple samples that were analyzed using the Olink solutions. So the solution, unfortunately, the video here is not working. The solution that company is providing is very, very, very simple. So it uses the pair of antibodies to each antigen. Those pairs of antibodies are very specifically adjusted, and it also uses the conjugated oligos. So 2 oligos for each antibody. They form the bridge, and if you had many, the high abundance of antigen, so you will get the high abundance of the amplicons after the PCR. So after the double-strand bridge is formed, the amplification occurs, and you can measure the amounts of the amplicons using the qPCR method or NGS. So indeed, the 2 methods are possible for this technology to analyze. You need high throughput NGS to analyze multiple proteins in one panel, so there are multiple panels available, and you can modify your panel in case if it's needed, so any proteins that you need. And also, you can modify your panels for qPCR. I would like to highlight that the very specific instrument is needed. This instrument is using the microfluidics technology, so ordinary qPCR instruments, unfortunately, will not suit this workflow. So what's next? The one of 3 DNA, one of 3 antibody pairs will form the panel. So when the Olink collects the panel, so they tested many different antibodies, and those antibodies are being tested using the very diverse portfolio of different tests. The tests in portfolio is rigorous, and now in the portfolio there are more than 5,000 different antigens available for testing. The technology is very well correlated with the mass spectrometry. There were multiple articles that highlighted it, but in contrast with the mass spectrometry, this technology is more suitable for low abundant proteins. Usually those proteins are of interest for researchers because the proteins that are very highly expressed are not so interesting in serum, in plasma, because you want to adjust and analyze the very minor differences between different proteins that you research, and usually those low expressed proteins are the biomarkers of interest. So red dots on the upper plot are the proteins that were found using the mass spectrometry, and you see the proteins that were found of the mass spectrometry are very highly expressed. So in case of low expressed proteins, so the Olink technology is much more suitable. Unfortunately, the slide does not work as well, so that's the workflow. The workflow is incredibly simple, just 2 days, including the incubation. So obviously to allow the antibodies to hybridize, to form the double-strand DNA bridge. Then it goes amplification, and after amplification, you will be able to get your library or using the qPCR method to analyze the amount of the protein that was initially there. This technology is very well known, so that's why this technology was chosen for the most biggest proteomics study in the world. 600,000 samples will be analyzed in the research, in the study that was just recently announced, and its study is currently going on. So there are multiple articles that I will not stop here. There are many diseases that markers were analyzed using the O-link panels. So for example, this interesting study. This interesting study was focused on aging, and it was study 50,000 samples. They found that was a longitudinal study. There were already multiple longitudinal studies because this technology is available in the market for already 20 years. So they studied the proteins in the serum initially, and then some causes, diseases, and outcomes after many years since after that. This interesting fact that proteomics can show here is the correlation with aging. So in case if you assess the polygenic risk scores for multiple diseases at birth, at the early age, they will be very, very directly correlated with proteomic age. So there is also the parameter that is called proteomic age. Scientists found the correlation between the real age and the proteomic signatures. So proteomic age is indeed the proteomic signature, and at low age, that's well correlated. With the progression of the age, with the aging, it starts to discorrelate. And at the late age, the proteomic is not correlated. The proteomic age is not correlated with the polygenic age, because in the beginning, if you even had the very good genes, so-called good genes, but you had bad habits like smoking, like the improper nutrition, like the other habits that can impact your health, the proteomic age will be definitely more impacting and showing your real age. So there were many, many studies that shows the good correlation of proteomic signatures with real ages. The first plot here is using the UK Biobank study, that is using the Chinese Biobank study, and that is from Finland. And they correlated the real age. The real age is here, and the proteomic age. So the proteomic age, if it will be reversely calculated and compared with the real age, is really, really very, very well correlated with the real age of the human. So using the proteomic signatures, it is really very easy to assess the real age of the person. Also, proteomic signatures, here it's shown in the red, the proteomic signature of 204 proteins. That is a really small amount of 20 different proteins, and they were discovered many different proteomic signatures that shows that this proteomic signature can directly be associated with, for example, at the upper side of the plot, with dementia, with some kidney diseases, with stroke, and many, many others. So if you will assess the proteomic age, if you assess the proteomic signature, which is to be shown associated with some kind of diseases, so you can predict the very, very high risk of the causing such diseases. Also, it was shown that oncological diseases are well correlated with some protein signatures, and just 83 proteins are needed to distinguish if this person is healthy or has the cancer, and distinguish between different types of the cancer. So proteomic signatures can be a really, really good biomarker of cancer and different cancers to differentiate between different types of the cancers. So the Thermo Fisher not only supplies the link for proteomics, but also Western blot, mass spectrometry, Luminex assays, ELISA assays, and many, many others. And we are happy that a link became a part of our portfolio as well. At the last slide, you will see the QR code at which you can get prices, more information about the link, and thank you so much for your attention.
All right, thank you, Anton. Any questions for Anton? If not, I think we'll move right along since we're a tiny bit behind. Thank you for that. All right, our next speaker this morning is Professor Rashan Kurmasheva, who will talk to us about exploiting DNA repair vulnerabilities in pediatric solid tumors. dual targeting of PARP1 and alkylation damage response. Thank you. Good morning.
Okay, good morning, everyone. I would like to first thank you, the organizers of this meeting, for doing such a great job putting together this wonderful conference. And particularly, I would like to, Dos is not here, but I would like to thank Dos, whom I know for many, many years now, and who has been a great colleague, great friend, and great person over the years. So today, I'm going to talk about one of the projects in my lab, and it is about DNA repair response in pediatric tumors, particularly targeting PARP1 and alkylation DNA damage. So briefly about what is pediatric cancer and how different it is from adult cancer, for those who don't know. On the left side, you can see the diagram depicting the the different subtypes of pediatric cancer, with the most common being, as you can see, leukemias and brain tumors, and followed by different other types of pediatric cancers like bone cancers, kidney, etc. And on the right side, you can see the 5-year survival rate for all these different types of pediatric cancer. So as you can see at the top, it's the overall 5-year survival for all types of pediatric cancer is about 85% currently, which is a big increase in the last 40 years, probably from about 10% up to 85% now. But you also see the different rates of different percentages on this picture. For example, some pediatric cancers like acute myeloid leukemias or brain cancers and some other still at a very low cure rate. So there's still a lot of work needed and research to be done. And I also wanted to mention that, there's such a coincidence that September is actually an International Childhood Cancer Awareness Month. So I'm glad to also bring attention to this focus in research. So a few points, just again to kind of highlight what is cancer in children. In terms of, in the context of cancer, it would be not right to consider children as little adults. And for that, there are several reasons. I'm not going to go into all, but just one of the most obvious ones is the genome landscape, which is very different between pediatric cancers and adult cancers. Pediatric cancers are usually arise from chromosomal translocations, while adult cancers from point mutations. So one of the other important factors about pediatric cancer to know is, that pediatric cancer is considered to be a rare cancer. In the United States, 43 children are diagnosed with one of the pediatric cancers every day, and 5 of these children die every day. And finally, one very important point is the survival. Children who undergone therapy still expected to have many, many years of life. And if the therapies that they were applied to them are toxic, then that leads to so-called late effects, which could be very devastating diseases, secondary cancers, heart failures, different other organ failures. Again, in the United States, about 70% of children experience these so-called late effects, and 32% of those experience really debilitating, really really difficult diseases later in life. And this diagram in the center shows number of survivors in different decades of life. So each bar is a decade of life, and you can see that after the second decade, approximately, that's when the therapy is given to children, the survival declines significantly. So the important point to understand about pediatric cancer is that the therapies should be not only shrinking the tumor, but also be not toxic for children. Today I will focus on one of the pediatric cancer types, malignant rhabdoid tumors. There are two major subtypes, intracranial and extracranial, depending on the site of where they arise. Extracranials can be kidney or soft tissue. Prognosis for these children is very poor. About 10% of children only survive over 5 years. And usually this cancer happens to infants, like newborn up to 2 years old, which also makes it more difficult in terms of therapy. One of the important genomic features of this cancer is SMARCB1 biallelic inactivation. About 97% of cases have this gene completely deleted. In other 3-4%, it's SMARCA4. This gene is part of the Swiss-Nef chromatin remodeling complex. And I'm not going to go through all of these, but generally these rhabdoid tumors are genomically stable without amplifications or deletions and diploid. There's a lot of attention in research has been given to understanding the structure of this complex and the role of this gene, which participates in epigenetic modifications and affects abnormal proliferation. However, the role of DNA damage response remained underexplored in these tumors. So what we did, we were interested in understanding the effect of PARP inhibition combined with DNA damage. And usually, so the rationale for this combination is in so-called synthetic lethality. On the left top, you can see that schematic of synthetic lethality. Many of you probably know what that is, but in the context of PARP1, it means that when there are single-strand breaks created by certain damage and PARP1 is not available in cells, and there's additionally homologous recombination deficiency, such as BRCA deficiency, this can lead to cell death. So we kind of using this idea in designing our therapy, where we use talazoparib as PARP inhibitor, which traps PARP on DNA, which is by itself toxic to cells. And we combine talazoparib with temozolomide. This is an alkylating DNA damaging agent, which through the lesions that are created by this drug, it also leads to homologous recombination deficiency at a certain point. So combined DNA damage and repair inhibition creates lethal combination, specifically targeting cancer cells. That's the idea. And this is a busy slide. I'm not going to also go into deep detail, but I wanted to point out to understanding that a good response to temozolomide, a DNA damaging agent, requires low level of methylguanine methyltransferase. This is one of the repair proteins required to repair one of the most toxic and mutagenic lesions created by temozolomide. And I'm showing it on the left figure. So temozolomide creates 3 major types of lesions. 2 of them are repaired by PARP through base excision repair. And the third one, 6-methylguanine, which is the most cytotoxic one, is repaired by MGMT. There's another pass that involves mismatch repair, which I'm not going to talk about, so just shown here. I also wanted to note that we recently showed, and this is published, the direct interaction between N-terminal PARP1 and MGMT proteins. We also showed that PARP1 actually is able to pyrolate MGMT, and pyrolyted MGMT, the function of this pyrolation is increase in the repair capacity of MGMT. So technically, this interaction is probably one of the mechanisms of resistance in these cells to DNA damage. So for going back to malignant rhabdoid tumors, so we started with understanding the cellular fitness in response to this therapy of PARP inhibition combined with temozolomide DNA damage. So we had this cell line, isogenic cell line with DOX-inducible SMARCB1, and we showed that we're able to keep the induction for at least up to 9 days. So IMR blue staining here shows potentiation of PARP inhibitor to DNA damage. So you can see the shift between the curves. However, there's no really role for SMARCB1 in this context, so SMARCB1 doesn't really increase potentiation. And interestingly, PARP inhibitor alone, it's shown separately here because of the different concentration range. So PARP inhibitor alone was much more effective in the presence of SMARCB1. Similarly, we investigated the colony formation ability of cells, and so you see the single agents, temozolomide, talazoparib, and at the bottom is combination. So very low combinations of each drug were able to induce complete death, and even more pronounced, this was in the presence of SMARCB1. Next, the cell cycle effect also was impaired, so PARP inhibition and DNA damage therapy arrested cells in G2M phase of the cell cycle, while each agent separately did not really have such an effect. And SMARCB1 also had some additional potentiation effect. If you look at the normalization, you could see that. And of course, we were interested to see whether there's apoptosis induction. If you look at these graphs at the bottom, the blue line is the combination. So the combination induced stronger cell death, and SMARCB1 actually induced it even further. And DNA damage was also induced. We looked at these major DNA damaging markers, P53, BP1, and gamma H2AX at early and later time points. So we saw significant induction of foci formation of both by the combination of drugs, not by the single agents. I'm not showing single agents data here just because of the limit in space, and so that I don't confuse you more. So then we kind of understood that in the cell context, we see good effect of this drug combination on rhabdoid tumor cells. So the next question was whether we see similar effect in vivo models. So we have a number of rhabdoid xenograft models developed in our group over years, and with corresponding demographic data. Previously, this is a published work already. We have already tested talazoparib and temozolomide combination in Ewing sarcoma. Ewing sarcoma is also a pediatric cancer of bone. This work is part of the pediatric preclinical testing program study. And as you can see, on the left is a group of responders, and on the right is a group of non-responders, so-called. There's two combinations were tested. It's the same drugs, but just different doses. And despite such a great response, you can see the complete regression of tumors in both combinations on the left, and of course, lower effect on the right. But despite such a great regression of tumors, we observed high toxicity. And this is, you can see it on the bottom, tolerability profile. So at approximately the same concentration we used here, we saw significant weight loss in mice. So this was a limiting factor. Even so, this combination actually went into clinical trials in children, and similarly, they observed toxicity. So the question was, we have a good, effective in terms of tumor regression combination, but it is toxic. So we collaborated with this company that developed pegylated talazoparib. This is a nano-formulated form of PARP inhibitor. The idea is that the nanoformulation allows delivering of the drug to the site of the tumor, avoiding the exposure of the drug to the normal tissues. And through this, we were expecting to increase the therapeutic window and reduce toxicity. This is just the chemical, physical characteristics of this conjugate. It's a slow-release conjugate consisting of a drug connected through the linker, and it's released at certain pH when it gets into the system. On the bottom, you can see the pharmacokinetic profile simulation. So the zigzag line shows the daily dosing of talazoparib. That's how the concentration would change in plasma. But this conjugate PK corresponds to those curves, red and black. So it's a smooth, slow release over 336 hours. Initially, we tested tolerability, of course, of this pegylated talazoparib combined with temozolomide. On the left, we picked a little bit, higher dose of temozolomide with different concentrations, doses of pegylated talazoparib. And at the first step, we did a screen of 28 different pediatric tumor models using so-called single mouse testing approach that was developed also in our group. On the right, you can see the Kaplan-Meier plot demonstrating the mouse survival. So the shift in between the curves shows that there was an extension in survival in the treated group compared to control. Additionally, we also introduced a specific schedule here. So we gave pegylated talazoparib to mice on day 1 and allowed distribution for 2-3 days, and then we started giving temozolomide from day 3 onward. This is just to show, to say a little bit about single mouse testing approach. The idea is to use 1 mouse per group versus 10 mice per group, which is this kind of gold standard. This is how the single mouse trial would look. So instead of using 420 mice, we would use 42 mice. And it can be, this approach can be applied to really any type of in vivo study. So it allows, of course, gives more cost efficiency. It's quicker, and could be a great first screen type of in vivo experiment. And it is based on this published work, also our group, it's already about 10 years ago. This is a deep broad, let's say, statistical analysis of data generated in our program from in vivo studies over 10 previous years. And what we showed here is, on the left is distribution of deviation for more than 2,000 observations. The tallest bar in the center at 0 shows the number of accurate predictions. And to the left and to the right are under predictions, so over predictions only by 1 category. On the right, it's another data for correlation between observed and versus calculated values, just to confirm that there is a great correlation. So the bottom line is that we observed 78% of predictive accuracy for 66 out of 67 validated drugs. So this is very currently well accepted and used approach in vivo studies. So for the rhabdoid tumors, we went into the efficacy testing step, and we observed these really great responses in these three models. The red curve you can see is the combination one. So in the WT16, for example, the tumor completely regressed and never really grew back for over 12 weeks at least. And there's significant survival also at the bottom in the Kaplan-Meier plots. In this group, we also observed partial response in 2 tumors, and one tumor didn't really respond. It was G401. But even with partial response, there was significant survival in mice. The question we had next, with 5 out of 6 tumors really showing good response to this therapy, are there any markers of response that we can capture? So we went back and we wanted to see whether there's correlation between the response and the SMARCB1 protein expression. So SMARCB1 was only expressed in one of the tumors, so no correlation. We then looked at PARP1 and MGMT, which I mentioned about earlier. And it was very interesting to see that MGMT protein expression had inverse correlation with the response. So you can see, I don't know if you remember, but G401, if you see on this table, G401 was non-responder, and we have the highest level of protein expression, and WT16 was the best responder, and MGMT almost absent. Even so, the number of tumors for statistical analysis is not high here. There is a good correlation, and it's very interesting because we repeated this experiment several times. And then we just initially did this in vitro study. So we did a triple combination of talazoparib, temozolomide, and now we added MGMT inhibitor, 6-benzylguanine, and about 4-fold sensitization has taken place here. Okay, sorry, time, right? Okay. I'm almost finished, actually. So key findings, I probably don't need to go over everything, but going forward, exciting conclusion would be that we have potential biomarker of response. And going forward, we are currently performing multi-omics analysis to identify more response predictors. And I wanted to acknowledge, I think this slide is as important as any others, right? So pediatric preclinical in vivo testing program, which is actually being funded by NCI since 2005, nonstop. Collaborators from St. Jude, Prolinx, Utasca, and my lab. There's some people from my lab. Thank you so much. Sorry.
No problem. Thank you for that fantastic presentation. So for the sake of time, we're going to move on and I think save the questions for the end of the session or during the coffee break. So we'll move on to our next speaker now, Professor Svetlana Menkovskaya, who will talk about experimental models in vivo, the basis of translational medicine. Thank you.
Добрый день, уважаемые коллеги. Но прежде всего я хочу поблагодарить Организационный комитет за приглашение и надеюсь, что данное мероприятие будет отличной площадкой для обмена опытов и завязывания новых контактов. Но мое выступление будет посвящено использованию лабораторных животных в биомедицинских исследованиях. Так, не это. Минуточку. Значит, немного истории. Одни из первых воспоминаний, наиболее ранние воспоминания об использовании животных встречаются в трудах древних греков еще 4-3 века до нашей эры. Но здесь необходимо вспомнить очень известного человека того времени, Аристотеля, который один из первых стал проводить вскрытие животных для исследования органов. Ну и дальнейшее, в принципе, развитие всей науки непосредственно связано с использованием животных. Ну, такие ключевые даты, которые хотелось бы отметить, это 1880 год, когда Луи Пастер, изучая микробиологию заболеваний, провел заражение овцы сибирской язвой. Ну, далее здесь также необходимо вспомнить великого русского физиолога Павлова Ивана Петровича, который проводил исследования условных рефлексов, используя собак. Ну и в конце XIX века появляется первая лабораторная крыса и вводится понятие «лабораторное животное». В 1929 году появляется первая лаборатория по линейным животным. В настоящее время...
Извиняюсь, мы еще не раздали нашим иностранным гостям этот переводчик. Может, быстро раздадим?
Ага. Ну, в принципе... Ну, тут такие общие сведения, тут...
Is there anyone else who needs the translator? So I guess we can go on.
Ну, в настоящее время животные довольно широко используются. Это начиная от простейших и кончая человекообразной обезьяной. В 2005 году, по данным Европейской ассоциации, оказалось, что где-то 12 миллионов животных использовалось в научных исследованиях. Причем здесь речь идет только о позвоночных. Доля мышей и крыс в этих исследованиях составляет почти 85%. Значит, лабораторные животные очень широко используются прежде всего в медицине и фармакологии, дальше в фундаментальных исследованиях, в химической оборонной промышленности в космосе и около 1% являются учебными пособиями для медиков и биологов. При формировании экспериментальной модели сегодня доступен довольно большой диапазон факторов. Это возможно использование физических, химических факторов, хирургического вмешательства, диеты. Но в ряде случаев при формировании модели здесь необходимо соблюдение определенных условий, то есть необходимы уже определенные виды животных, это касается 3 последних моделей, и определенные условия их содержания. Но я хотела бы представить буквально 2 проекта, которые проходили на базе Института физиологии совместно с нашими зарубежными коллегами. Первый проект, это был проект с американской компанией Regenerative, и он был посвящен проведению терапевтического лечения ишемии головного мозга на модельном эксперименте с использованием крыс. Значит, сама модель, в данном случае мы использовали крыс, сама модель широко описана в литературе и связана, моделируется путем окклюзии срединной артерии. Значит, при окраске коронарных срезов вот, неповрежденная зона остается розовым, а там, где возникает очаг некроза, появляется вот такой белый окрас. Значит, что касается препаратов, то рекомбинантный фактор роста фибробластов и рекомбинантный фактор роста дифференцировки был передан на базу Института физиологии американской компании. Были сформированы опытные группы. Значит, в опытных группах препараты вводились через час после формирования модели. Доза была одинакова, однако режимы терапии отличались. Для FGF была внутривенная инфузия со скоростью введения 0,5 микролитров в час в течение 3 часов. Общий объем введенного препарата составил 1,5 миллилитра. Для GDF мы вводили 100 микролитров в виде внутривенной инъекции с 1 по 5 день эксперимента. Ну и контрольная группа получала растворители. Значит, на 21 и 42 день был изъят головной мозг и готовились коронарные срезы, которые окрашивались по мислю. При морфологическом исследовании оценивалась морфология нейронов и рассчитывали глиальные и нейрональные индексы. Значит, при морфологическом исследовании наиболее серьезные повреждения нейронов, естественно, были выявлены в контрольной группе. Там встречались клетки с пектоническими ядрами, там же были выраженные отеки. Что касается нейронального идеального индекса, то к 42 суткам эти показатели улучшались во всех группах, однако наиболее серьезные повреждения, естественно, были выявлены в контроле. Группы, которые получали биотерапию, имели более низкие показатели повреждения нейронов и более высокий показатель глиального индекса. Но наиболее лучшие результаты были показаны для FGF-18 полученные. Значит, дополнительно мы посмотрели смертность животных. Естественно, в группах, которые получали биотерапию, смертность была невысокой. И опять же, здесь, так же, как и в предыдущем случае, наилучшие результаты были получены при FGF, что позволило нам сделать заключение, что это может быть один из перспективных препаратов для лечения ишемии головного мозга. Но следующий проект – это уже разработка белорусских ученых совместно с Институтом биоорганической химии. И он был направлен для разработки и создания новых модуляторов ангиогенеза для лечения ишемии нижних конечностей. Значит, опять же, в исследовании мы брали крыс обоего пола, и нами было опробировано 3 модели экспериментальные. Все эти модели создавались хирургическим путем. В основе лежала легированная перерезка бедренной артерии, но если в первой модели мы удаляли 4 миллиметра артерии, во второй модели 12 миллиметров, в третьей, кроме удаления участка 12 миллиметров бедренной артерии, мы еще накладывали лигатуру на бедренную вену. Во всех случаях оценку этих моделей мы проводили с помощью аппарата, где оценивали отпечаток оперированной конечности и интенсивность отпечатка, площадь и интенсивность отпечатка оперированной конечности. Все 3 модели показали уменьшение как площади, так и интенсивности отпечатка оперированной конечности. Однако при исследовании морфологическом удаленных мышц бедра и голени оказалось, что первая модель имеет очень незначительные морфологические нарушения, и к 21 суткам практически мышечная ткань не отличается от нормы. Третья же модель характеризовалась обширными некрозами, и животные тяжело ее переносили. Поэтому наш выбор, мы остановили свой выбор на второй модели. которая, начиная от 7 к 21 суткам, наблюдалось постепенное нарастание ишемических повреждений, животные переносили хорошо. Однако, с учетом того, что у крыс, это особенность их кровоснабжения, довольно сильно развиты так называемые компенсаторные механизмы, мы модернизировали эту модель, и модернизация заключалась в том, что, во-первых, мы взяли более взрослых животных, а во-вторых, после удаления бедренной артерии мы ограничили подвижность животных на 28 суток. Это позволило нам получить модель, которая в течение 70 суток четко было видно, что у нас уменьшалась площадь и интенсивность отпечатка оперированной конечности. Ну и вот получен патент на эту модель. Ну и далее, с использованием этой модели, мы провели тестирование 2 препаратов, созданных на плазмиде. Первый из них это содержал фактор роста, эндоваскулярный VEGF, короче, и второй, это была комбинированная конструкция, которая кроме VEGF имела еще участок гена ангиодинина. В обоих случаях терапия начиналась на 28-й день после операции, доза была 100 микрограмм на килограмм, вводилась однократно локально. Ну, при оценке площади интенсивности видно, что в обоих случаях мы получили, в принципе, неплохие результаты, однако комбинированная конструкция, она показала результаты намного лучше. Уже к 42 суткам мы получили четкие достоверные различия по отпечатку конечности, по самому с контролем, а к 70 суткам, в принципе, ни площадь отпечатка, ни интенсивность отпечатка не отличалась от показателей, характерных для нормальной конечности. Ну и это позволило нам сделать вывод, что вот данная комбинированная конструкция может быть перспективным препаратом генотерапевтическим для лечения ишемии. Ну и буквально пару слов я бы хотела сказать о нашем виварии, мощность которого составляет 2500 голов. В основном это у нас конвенциональные животные, это крысы, мыши, мозги и свинки, ну и имеется 5 линейных животных. Виварий неплохо оснащен, имеется операционная комната, имеется помещение для проведения лабораторных, морфологических исследований, ну и, конечно, достаточно большая линейка для оценки функционального состояния животных. На сегодняшний момент на базе института опробировано более 21 модели. Вот. И я очень надеюсь на сотрудничество с казахскими учеными, потому что в рамках межправительственного соглашения существует 2 конкурса. Но со стороны Республики Беларусь эти конкурсы курирует Национальная академия наук, со стороны Республики Казахстан, я думаю, что тоже Национальная академия наук. В этом году был впервые запущен конкурс ШОС, в рамках ШОС, это конкурс для мегапроектов. И там как раз есть 2 направления, где мы могли бы найти какое-то взаимодействие. Ну и второй конкурс, он ежегодный. Здесь также, скорее всего, с вашей стороны курирует Национальная академия наук Казахстана. Вот. Все, спасибо за внимание.
Okay, thank you very much. Again, we're going to save questions to the end and move along to our next speaker, who is Professor Murat Saparbayev, who will talk about repair of 5-hydroxymethyl-cytosine residues in DNA and its possible biological role. Thank you.
Thank you, Dave. First, I would like to thank organizer of this conference, especially Professor Nazarbayev, and also organizing committee, Aynur Akiljanova, Almagul Kushugulova, especially Dinara Begimbetova and Yulia Safarova, who helped me to come here and present my work. So here I would talk about more about fundamental science, not a translational one, and we are still working. So I'm working in hospital, in Cancer Research Center in suburb of Paris, Gustave Roussy. So this hospital first was organized by Dr. Gustave Roussy. He first realized that cancer is a disease which is apart, because before him in 19th century, for example, cancer of the lung cancer was treated by physician who treated the lung disease, you see, or kidney cancer was treated by physician who treats the kidney disease. So he first realized, among others, of course, that cancer should be treated apart from other diseases. That's why he constructed in hospital Paul Bruce cancer clinic. Then this center was named in his name, and I'm working there. So actually, Gustave Roussy, it's huge hospital, and it's becoming now kind of analog of MD Anderson in Texas. So we want to create big scientific hub where, so we invite many researchers worldwide where, so and I would say that I profit from the cancer. You see, cancer is a huge problem for modern society, so there are a lot of money. So this money stimulates also research in fundamental science, you see. So let's go. So first I would talk about interpretation of information which is encoded in DNA, you see. So you can treat it in linear fashion, you see, or in other fashion, you see. You can read it during replication and transcription translation, but it also can read in epigenetic way, you see. You have this two types of organism, you see, it's a butterfly and caterpillar, you see. So we have the same DNA, but we exist in two completely different forms. And we have one genome, but two proteomes. Why? So it's for this question, it's epigenetic who studies this question, why it happens. So, you know, it's also, you can observe in embryogenesis, organ development tissue. So we all originated from single cells, which called zygote, and this zygote forms blastula gastrula, and then body is formed, and since we have only 2% of DNA which codes for proteins, and we have about 98% of protein which doesn't code anything, you see. But this 98% of DNA serves for other purposes, especially for regulation of genes, especially genes which are involved in development, you see. So there are promoters, but you have also enhancers, you see. So promoter itself would not function if there is no enhancer. So there are enhancer which can turn off the promoter and then develop, for example, face, brain, other body organs. So this is regulation of gene expression, as you know. So how the gene expression regulated, I will tell you today just one, let's say, facet, one side of this regulation is based on methylation of cytosine. You see, cytosine can be methylated at 5' carbon. No, yeah, it's methylation 5' position, and this methylation typically does not interfere with the replication transcription, is not miscoding. Cytosine keeps it. So this methylation serves to suppress expression of transposable elements and also regulate expression. Usually methylation associated with silencing of expression. So what is interesting, if you watch the methylation of global genome in animals, vertebrates, or let's say mammals, in mouse, you would see that there is dynamic of DNA methylation, you see. So it's the first time the gametes eggs and spermatozoids, they have, especially spermatozoids, have high level of methylation, yeah? And when they form zygote after fertilization, this zygote goes to complete demethylation of genome, you see? So you can kind of erase all memory which you have in differentiated cells, and you reprogram. All genome is reprogrammed, all memory is erased, and then this memory is restored during the embryonic development. So when you have somatic body, you restore the original level of methylation. About, let's say, 80% of CpG denucleotides are methylated. But to form the germline cells, you again need wave of demethylation. So how demethylation functions? So demethylation has also kind of a drawback because methylated cytosine are prone to deamination, and deamination of cytosine creates thymine, which is much difficult to repair than uracil. So there are glycosylase about which I would talk today is TDG, thymine glycosylase, which repairs with deaminated 5-methylcytosine, T opposite to G, but this DNA glycosylase also participate in the transcription regulation and protection of CpG island from de novo methylation. Okay, what is interesting, it was started to be more clear starting from 2011. Researchers discovered that this demethylation, wave of demethylation, which occurs during embryogenesis, it can also occur in differentiated cells, but on lower scale, is a process when this methyl residue at 5' position of cytosine can be oxidized by TET proteins, yeah? TET proteins oxidize with oxygen and in iron cofactors. We oxidize it to form of 5-formylcytosine and 5-carboxycytosine. But before doing 5-formyl, 5-carboxycytosine, they go from intermediate state, they form also 5-hydroxymethylcytosine. So 5-formyl and 5-carboxycytosine are removed by TDG. That's how the genome in fertilized cells during embryonic development are demethylated. So I used to study this thymine DNA glycosylase and MBD4 protein, and it turns out that this protein is also can do bad things like aberrant repair, because since this protein removed thymine, thymine is normal base, you see classical, it's not damaged base. So sometimes we missed, and instead of removing the damaged residue, we can remove normal residue like this thymine opposite to damaged adenine, which we discovered in 2014, and we hackerized it, and actually mutation actually can occur and fixate it without replication because of this aberrant repair. And there is evidence that in our neurons and muscle cells, yeah, we still have mutation. Even these cells are not divided, but we can fix this mutation due to this aberrant process. And recently we found that this DNA glycosylases, which are participate in DNA demethylation, they also have very strange properties. If you incubate them quite long with DNA, they start to excise thymine and cytosine in normal, non-damaged DNA. It's kind of futile repair. The kinetics of this repair is very slow, extremely slow, so maybe it's not it doesn't present danger to our genome, you see, but still it's very interesting phenomenon that DNA glycosylase, DNA repair protein sometimes introduce strand breaks endogenously. Very slow, but constant level. We also find with thymine DNA glycosylase becomes stable. You see, it loses its activity if you incubate at 37 degrees. Any enzymes can lose its activity if you incubate it at physiological temperature, yeah? Because of protein denaturation and misfolding. But with DNA glycosylases, what is interesting, they can be stabilized in presence of DNA. So they form complex of DNA, kind of, and in this complex of DNA, we can actually keep the activity until 48 hours at 37 degrees. This is one. Let's back to the epigenetic regulation of gene expression. You know why enhancers are so important in the genome of high eukaryotes. As you remember, high eukaryotes, it's most of DNA non-coding. More than 90% of DNA non-coding. There are a lot of mutation and in regeneration non-coding part of genome. And they contain these elements, regulatory elements, which we call enhancers, you see. You have promoter, yeah, but promoter itself would not work. So in order to promoter to work, to start transcription, you need bring to promoter enhancer, which is long away. It could be 1 million base pair apart from the promoter. So how this enhancer could interact with promoter, what forces are played a role in this interaction is still question, and one of the possible way to interact, to make interaction between enhancer and promoter is to introduce single strand break. So actually, recently was a nice publication in Science and Nature by laboratory of Andrei Nussensweig. He found that actually in neuronal cells, enhancer which are required because neurons express a lot of genes with specific to neuron, neuroimmediators, etc. So the expression of this constant expression of these genes are under neuronal program. So this program are regulated by neural enhancer. So to make work with neural enhancer, our neurons suffers 1,000 DNA strand breaks every day, even without any damage, no damage. Why we suffer a single strand break? Actually, because there is constant methylation and demethylation at neuronal enhancer. So 5-methylcysteine are oxidized, become 5-carboxy-4-methylcysteine, which is removed by thymidine acetylase that create breaks. With breaks it's repaired, and it's repeated over and over again. It's kind of futile repair, you see, futile repair. And this is necessary for keeping the neuronal activity. So it's why brain, neuron tissue, are very sensitive to DNA damage, like Roshan said, timolizumab, ionizing radiation, because they already suffer from the breaks. If you put more breaks, so they become more sensitive. So I would talk about hydroxymethylcysteine because its role is not very clear because it's intermediate product of active DNA demethylation by itself is not repaired. It's very passive element, but what is interesting, hydroxymethylcysteine is accumulated at high extent in the differentiated tissue, you see? Again, with this role of genetic embryonal development, you have about 200, more than 200 cell types, which is formed human body during the development. And what is interesting, more cells is differentiated, more you have hydroxymethylcysteine. So it has some role. We don't know why, and how this hydroxymethyl performed this role, we don't know. So what we found recently, we actually showed that, this is just to show you how demethylation occurs, yeah? Because you need to remove 5-methylcysteine in order to convert the chromatin from hetero to L-chromatin, to close to open, to make genes express and work, you need to remove the 5-methylcysteine, and it goes in dynamic way. So, as I told you, with TET proteins, they oxidize 5-methylcysteine to hydroxymethylcysteine, which is intermediate, you see? But it accumulates for some reason. And then for the next step, we oxidize it to 5-formyl carbo, which are removed, and you demethylate completely DNA, you see? So what we found recently, it's a work of Kazakh PhD student, financed by Guertin Fellowship, Diana Manapkadze. She demonstrated actually thymidine acetylase can also remove hydroxymethylcysteine, but in very, very slow manner. So it requires about 18 hours to remove about 30% of hydroxymethylcysteine. So hydroxymethylcysteine can be removed like formyl and carbon carbocytosine, you see. It can be moved, but in very slow manner, you see. What is interesting with slow repair, slow repair is actually associated with the fact that hydroxymethylcysteine, were typically enriching enhancer and gene bodies of transcriptionally active tissue-specific genes. And what is important, hydroxymethylcysteine accumulates in post-mitotic cells. Slower cells divide and proliferate, more it has hydroxymethylcysteine. The highest amount of hydroxymethylcysteine you can find in brain neurons. Neurons do not divide, you see. We function whole our life, for 20 or 30 years, so we accumulate a lot of hydroxymethylcysteine, and this hydroxymethylcysteine. Whereas cancer cells, they proliferate very fast. They lost most of hydroxymethylcysteine. There is very low level of hydroxymethylcysteine in cancer cells. So apparently, there is this bad phenomenon that you may heard about with cancer persister cells. You see cancer persister cells, if you treat patients with cancer or mouse model, you see that if there is response to the treatment, cancer cells die and tumor shrinks. But what is interesting, that most of the tumor, when you treat them, they die, most of cells die, but about, let's say, 50% of cancer cells in any tumor, even cancer cell lines, they survive. They survive not because of mutation, they reprogram, transcription program. They capable to reprogram all cancer cells, capable to reprogram, they start to proliferate, they start to divide, they do not die, they do not go out. Apoptosis, 50% of them, you see. You call them persister cells, you see. There are always persister cells. This create a problem of cancer resistance of cancer which is resistant to treatment. You need very high doses of anti-cancer treatment to eradicate these cells. But anyway, if your cancer are metastatic, you have very low chance because of these persistent cells. These persistent cells, what is important, it's not mutation, because if you take these persistent cells, which are resistant to drug but do not proliferate, if you put them back to media without drug, they start to proliferate. So the way to survive, cancer cells can reprogram their genome to survive. So I think our hypothesis to study this repair of hydroxymethylcysteine in role of appearance of these persistent cells, because as soon as cancer cells stop to proliferate, they accumulate logically 5-hydroxymethylcysteine. If they accumulate hydroxymethylcysteine, that means they turn on the tissue-specific genes. So the cancer cells start to acquire the memory which was in the past. It becomes normal cells. Of course, temporarily normal cells, because it accumulates hydroxymethylcysteine. So also about aging, as you understand, hydroxymethylcysteine is very important to keep cell differentiated. So it's cell commitment, cell fate, because to keep the epigenetic memory stable, but neuron stays a neuron, muscle cells still the muscle cells, even after cell division, they keep this memory. So this is important because the way to store the epigenetic memory is methylation of cysteine and hydroxylation of this 5-methylcysteine. What is interesting, upon accumulation of hydroxymethylcysteine in cells, you have reduced greatly the transcriptional noise. You express only necessary genes. You don't express other genes. Maybe it's also there is much less splicing variants, you see? So cells become, of course, more effective, more functional, but at the same time, with the age, with cells lose its plasticity. So it's become more fragile. It become more and more professional, you see, more professional, but it loses its fantasy and adaptation, capability to adaptation. So we are kind of programmed to the aging. So, okay, this is my conclusion. Thank you for your attention. So.
very much. All right, well, again, we'll have questions at the coffee break after. Thank you. All right, the last speaker is me. I am David Guertin, and let's see. I am going to talk about targeting adipocyte nutrient sensing to treat obesity and metabolic disease. So something we haven't really talked about at all in the meeting. So I'll have some background slides in there to hopefully teach you guys something. So I want to thank the organizers, first of all. It's amazing to be here. This is so inspirational to see what's happening. Special thanks to Das Sarbasov as well. A little note that Das and I overlapped as postdocs in the Sabatini lab many years ago, and Das actually trained me how to do cell culture. He was a fantastic leader then, and he's clearly a fantastic leader now. So I want to say that thanks to Das. All right, I'm already going to skip this first slide for the sake of time, get right to what we do. So the big question that drives us in the lab is, how do nutrients shape health span? And so we look at this in several different ways. For many years, as I just indicated, I've worked on the nutrient sensing mTOR pathway. Over the years, we've started to study more how nutrient sensing pathways like mTOR talk to metabolism and how metabolites can feed back and regulate signaling pathways. And for about 10, 15 years now, we've really been focusing on this in the context of adipose tissue, and in particular, thermogenic adipose tissue, which is an energy-expending fat tissue. And I'll give some background into this since we haven't talked about this at all. Most of our work has been in this area, but I'm going to try to focus in the second half of my talk on mTOR. I can't come and visit DOS and not talk about mTOR, so I'm going to present mostly at the end an unfinished, unpublished project that we're pretty excited about, and we'll see what you guys think. All right, so fat cells come in 3 different shades or flavors. White, which are the most abundant adipocytes in your body. These are the largely energy-storing adipocytes. But there are also these 2 very interesting types of, we call them catabolic adipocytes, brown adipocytes and beige adipocytes. And so brown adipocytes are distinct. They store energy like white adipocytes, but for the purpose of burning that energy to generate heat in a process called thermogenesis. And that's mediated by a protein called uncoupling protein 1, and I'll tell you how that works in just a second. These cells also, they burn the lipid that they're storing, but they also consume nutrients from circulation as well. And there's this very exciting and interesting other type of, it's a brown-like adipocyte called beige adipocytes, and these exist within some white fat depots. You can see them here. And they also have similar properties to brown adipocytes. When they're activated, they consume energy to generate heat. They also use UCP1, but more recently we're learning that there are other futile cycle pathways distinct from the UCP1 pathway that can also burn energy to generate heat. All right, and so these adipocytes exist in distinct locations in the body. Brown adipocytes in humans tend to be located in the axillary, infrascapular, or interscapular regions. Mice have more of these brown fat depots, and you can see they're in a generally similar location, where beige adipocytes tend to exist in the subcutaneous white fat depots. And the best natural way to activate these thermogenic cells is to put mice or people in the cold. Their evolutionary function is to help maintain body temperature. They use glucose as a fuel, so you can easily image where brown fat exists in a person by putting him in the cold and doing FDG PET-CT scanning. In fact, that's how brown fat was discovered to exist broadly in humans. Okay, and so why is there such an excitement about brown fat? Well, it seems that having brown fat or the ability to make beige fat is healthy. And so it was really only 15, 16 years ago that it became appreciated that adult humans have brown fat. And in the ensuing 15 years or so, there's been many reports now that correlate the abundance or the activity of brown fat with protection from many different metabolic diseases. So there's been this intense interest in trying to identify therapeutic strategies by which we might be able to activate these thermogenic adipocytes. And some of the best work, I think, in this area is coming from Paul Cohen at Rockefeller, where he's analyzed PET scans retrospectively of hundreds of thousands of patients and shows a clear correlation between having brown fat and lower blood glucose, lower triglycerides, and many other health parameters. So unfortunately, we don't know exactly the mechanism yet by which brown fat protects. I'll talk a little bit about what some of the theories are, but clearly having brown fat is associated with metabolic health. All right, so why is it protected? So one of the leading theories is called the catabolic adipocyte theory, in which brown adipocytes need to consume fuel from circulation, and they expend that fuel, those nutrients, as heat. And so one idea is that when you turn on brown fat, it can combat obesity by essentially consuming up and burning off excess nutrients that can become toxic. And so the classic conventional view is that glucose, fatty acids, amino acids are all consumed by brown fat. They enter into the mitochondria, convert it to acetyl-CoA, which then powers the TCA cycle. This generates reducing equivalents that are fed into the electron transport chain. And unlike the electron transport chain in all other cell types, brown adipocytes and beige adipocytes have this UCP1 protein, which essentially bypasses complex 5 and short circuits the electron transport chain. So protons generated in the membrane potential are shunted back into the mitochondria uncoupled from ATP synthesis, and the net byproduct of that is heat is released. And that's why this is called uncoupling protein 1, and this is called uncoupling respiration. Okay. And so we've been interested in testing this model, and we like to do questions like what quantities of nutrients are taken up, how are they used, what is released back into circulation. So I'll just summarize some of our work in this area before moving on to the mTOR project I promised. But when we started this work, it was generally thought that fatty acids in the blood were the major fuel source for thermogenesis. And so we've teamed up with Chol-Soon Jang, who's a fantastic scientist at UC Irvine, to develop techniques like arteriovenous metabolomics and stable isotope tracing in vivo to try to understand how nutrients are used. And I'll just summarize a lot of work. We discovered that actually glucose by far is the dominant carbon source for thermogenesis, far more than lipids, which are actually third to amino acids. And we found that glucose isn't just used as a fuel for the TCA cycle, but it actually fluxes into many interesting pathways that help support how thermogenesis occurs and helps maintain the viability of the cells. And so, for example, glucose fluxes into the pentose phosphate pathway, glycerol 3-phosphate pathway. We've also discovered that there are a couple of very interesting futile cycles that exist in brown fat. So we discovered that a lot of glucose fluxes into this glutamine futile cycle where the cells make and break down glutamine, and that consumes ATP. And one that we've been most interested in, which is really a paradoxical cycle that occurs in very few cell types, but here in brown fat, most of the glucose actually fluxes into the mitochondria, into the TCA cycle, but then right back out to make lipids. And those lipids are immediately sent right back into the mitochondria. So you have this futile fatty acid synthesis, fatty acid oxidation cycle, and we've been trying to understand why. One reason is that we know that this running metabolites this way helps reduce stress on the mitochondria, and that was something we published recently. But if you notice, there's a lot of ATP hydrolysis steps in here as well. So another advantage for brown fat or thermogenic adipocytes to run all these futile cycles is they generate a lot more heat by doing this. So this is a very exciting area. There's lots of people working on trying to solve the metabolism of these fascinating cells. All right, but there are some potential challenges with targeting brown fat in human. And so there's limited mass in adults. There's only about 50 to 150 grams, rough estimate, which isn't much. It's highly variable between individuals. Here's just one study where only 11 of 19 people actually tested positive for brown fat, and this is one of the better studies. Its activity declines sharply with age, so brown fat, the abundance or activity declines very sharply shortly after birth and early childhood, whereas these beige adipocytes tend to persist much longer in life, or at least the ability to form these beige adipocytes. And so a big question is in the field, because of this problem with targeting brown fat, is can we turn white fat into brown fat or stimulate the formation of these beige adipocytes? And so I put this mTOR pathway model in here because I was going to tell a fun story about DOS, but I'll save that for dinner tonight. We worked on mTOR2 together, which was one of the best times in my career, but yeah, I'll tell you some fun things later. But for many years, we've wondered that hidden maybe within the mTOR pathway might be a switch where we could inhibit this in white adipocytes and turn them into these catabolic brown or beige adipocytes. And the reason is, as we've heard, the mTOR pathway is famously known as an anabolic pathway. It drives anabolic processes like protein synthesis, lipid synthesis, nucleotide synthesis, while simultaneously suppressing catabolic processes. So the hypothesis is, can we inhibit something here, turn off anabolism, turn on catabolism, make beige adipocytes? And so today, I focus mostly on mTORC2 in my career, but this project is going to be all about mTORC1, which is a bit exciting and maybe a bit provocative, we'll see. And so the first question we wondered, well, what if we inhibit mTORC1, the main downstream target in the mTORC1 pathway? If we inhibit that in adipocytes, will that turn on the formation of these catabolic adipocytes, and will that be protective to metabolic health? We did this many years ago. Many groups did this at the same time. We can achieve this by using an adipose tissue-specific Cre driver to delete a critical scaffolding protein in the mTOR complex called Raptor. So if you delete Raptor, mTOR is a dimer, but if you delete Raptor, the whole complex falls apart, and it's essentially a tissue-specific mTORC1 loss of function. And so we did this with the hypothesis that it would improve metabolism. We were completely wrong. Deleting mTORC1 in adipose tissue is highly detrimental to the health of mice. And so some of the major phenotypes were that we inhibited lipid storage. These mice develop progressive lipodystrophy. They have severe hepatic steatosis, and we actually inhibit the formation of these beige adipocytes. And this is just some of the data here. You can see that this is brown fat, subcutaneous fat, visceral fat, all the depots decrease in mass. This is only after 8 weeks, and that differential will continue to decrease with age. And the livers grow quite considerably in size, and you can see what happens essentially is the adipose can't store the lipid. It all ends up in the liver. These mice get fatty liver, and that's very bad for health. And if you further put these mice on a high-fat diet, it's even more severe. So adipose expansion is initially a protective mechanism against overnutrition. In the Raptor knockout mice, these are mice, again, no Raptor, no mTORC1 in their adipose tissue. They gain slightly less weight on the high-fat diet. This is only out to 12 weeks. But they eat a lot more food than controls. And the reason is they don't absorb a lot of that food. A lot of the lipids end up in their feces. And what happens is they have no capacity to expand their adipose tissue. You can see here there's very little adipose tissue compared to the control mice. It's quantified here in a graph, but what happens again, all of that lipid ends up in the liver, and they get massively fatty livers that will eventually progress to hepatocellular carcinoma. So again, emphasizing the important point, you cannot inhibit mTORC1 in adipose tissue. So it kind of sat here for a number of years until a scientist in my lab, senior scientist, wanted to dig a little deeper into the mTORC1 pathway and look upstream at one of the regulators of mTORC1, these RAG GTPases. And so I'll explain this in a second, but incidentally, the person who discovered the RAG GTPases in the Sabatini lab is also here, Yasmin Sankag, and she spoke yesterday. And so what Yasmin discovered was that mTORC1 is activated by recruitment to the lysosome. And that requires these proteins called the RAG GTPases, which exist of RAG-AB or RAG-CD. And the way this system works is when nutrients are present, amino acids in particular, RAG-A or B adapts a GTP-bound state, where RAG-C or D, the GDP-bound state. And this recruits mTORC1 to the lysosome, where it can be activated by another GTPase called Reb in response to growth factors. And this ensures that mTORC1 is only activated when both growth factors and nutrients are present. And so if there's no nutrients, the GTP state on RAG switches, mTORC1 falls off, and you lose activity of the mTORC1 pathway. And this triggers activation of autophagy and lysosome biogenesis and other catabolic pathways. And so Qingbo in my lab, one of my senior scientists, wanted to know, well, if we delete RAG-A, the RAG GTPase, can we stimulate the catabolic pathway in adipocytes? And I said, well, you're going to get the same phenotype, because essentially inhibiting the RAGs is going to lead to inhibition of mTORC1, and there should be at least considerably overlap with the Raptor or the mTORC1 deficient phenotype. But he went ahead and did this anyway, and it turns out that I was wrong. And the loss of RAG, again, which we predict would mirror mTORC1 loss, is completely different. So let me show you some of that data here. And this is a huge surprise for us. So the only adipose tissue that decreases in mass when you knock out RAG in the adipose tissue, we knock out RAG-AB, but this essentially ablates the whole RAG-GTPase complex. Only the subcutaneous fat is smaller. But what was most exciting to us and caused us to pursue this model further is that the liver showed relatively no difference in size. And you can see here, these livers look completely normal. We've profiled these livers. They're completely normal livers in the absence of adipose RAG-GTPase. So this is completely different than what we saw with Raptor. Again, expecting them to be the same. So here's the subcutaneous adipose tissue from Raptor and RAG knockout here. You can see there's a decrease in size in both, but what's clearly different in the RAG knockout is there's a high vascularity in this tissue. They have a redder or pinker color. And when you look by histology, you see that the white adipose tissue here forms a very characteristic appearance of these beige adipocytes I talked about, these catabolic or thermogenic adipocytes. And if you look at the main marker of thermogenesis, UCP1, you can clearly see in the RAG knockouts, there's this very striking upregulation of UCP1, again, different than what we see in the Raptor knockout. Oh, yeah. So if you take these mice, the RAG mice, now put these mice on a high-fat diet, completely different again than the Raptor knockouts. So these mice gain less weight on the high-fat diet, but they eat the same amount of food, suggesting they have increased energy expenditure. We haven't done the measurement yet, but that's the implication. And you can see that here. So they have normal lean tissue mass, and the only difference is total body weight drops a little bit, and that's due to the decrease in adipose tissue mass. So this is a perfect phenotype for people looking to protect against high-fat diet. They also have improved glucose tolerance as well. So a metabolically beneficial phenotype if we inhibit the RAG-GTPase. And the liver, of course, is we're most interested in what's happening in the liver. It's strikingly different. So in the control after a high-fat diet, you can see there's a little bit of lipid starting to form in here. Nothing in the RAG-AB knockout. And again, here's the Raptor knockout under the same conditions. A massive steatosis and an increase in liver size. So two different phenotypes. All right, so the other thing we can do with these mice is we can take them and put them in the cold to stimulate the formation of these beige adipocytes. So RAG already has them at room temperature, but if we take these knockouts, put them in the cold, you see the control mice increase UCP1, but the RAG-A knockouts, RAG-AB knockouts have an incredible ability to upregulate this uncoupling protein 1. And if you just look at the histology of the cold condition, you can see the controls forming these beige adipocytes, but there's this incredible formation of beige adipocytes in the RAG-AB knockouts, and this looks a lot like a brown adipose tissue. You know, there's a few adipocytes in there that aren't responding, but adipose is highly heterogeneous, so we don't actually know what the difference is between all these adipocytes, but there's a clear upregulation of this beiging phenotype. Again, here's a comparison with Raptor, completely the opposite. In the absence of Raptor, there's a reduced ability or no ability to increase the expression of UCP1, and histologically the tissue looks much more disorganized and full of these large, odd-shaped adipocytes. There's a lot of immune cells in here as well, so very different phenotype. So we've done some bulk RNA-seq on the adipose tissue. Again, this just emphasizes the opposite effects of deleting RAG or Raptor. RAG knockout is in blue, Raptor in orange, and you can see the RAG knockouts, this is in the cold, upregulate all these pathways that are consistent with increased beige adipocyte formation, so fatty acid oxidation, mitochondrial biogenesis, respiratory chain genes, nucleotide metabolism, and in the Raptor knockouts, we basically get massive inflammation, so again, completely different. We've done trans-FAC analysis, which essentially looks at all of these genes for transcription factor binding sites that might be overrepresented. And you can see that what pops up high on the list are these ERR targets. These are the estrogen-related receptor target genes. So ERR is an orphan nuclear receptor, and it has a well-known role in driving the thermogenic program. So we think there may be a connection here, and I can come back to that at the end if there's time. So consistent with the thermogenic adipose program being engaged, you can see that RAG knockout increases mitochondria number, and this is just based on mitochondrial DNA to nuclear DNA ratio. We can also look at markers of the respiratory chain, and you can clearly see in the RAG knockouts we have nice upregulation of all of the markers, at least of the key components of the respiratory chain. And I didn't show the Raptor data here, but it's completely opposite. All right, so here's the question. We have the same pathway. Losing RAG, losing mTORC1 is supposed to do the same thing, but we're getting opposite effects. So why is this? And so we're working hard on this, and I'll show you what we think is going on now. We have some preliminary data. And the first piece of evidence came when we took these mice, they're at room temperature here, and we do a nutrient challenge. So we fast the mice and refeed them. And so the common, most used reporters for mTOR pathway activation are phospho-S6K and phospho-4-BP1. And you can see in the RAG knockouts, if you fast and refeed, you do see a reduction in the classic mTORC1 signaling. So RAG is having a role here, as expected, in the nutrient response to stimulating mTORC1. Raptor, very similar. But the clue came when we put these mice in the cold. So in the cold, mice eat a lot, and so all their nutrient pathways are always stimulated. But what was interesting here, if you look at the RAG knockouts, 4BP still shows inhibition, so the classic dependency on RAG for mTORC1 signaling, but there's this apparent recovery or lower or higher level of S6 kinase phosphorylation that seems to be uncoupled in the cold from RAG dependency, and we don't see that in the Raptor knockout. And so Qingbo, working on this, thought that, S6K may be uncoupled from RAG-AB during cold exposure, so he came up with this idea that, well, maybe is it possible that RAG-GTPase is actually suppressing a branch of the mTORC1 pathway that's pro-catabolic? And so the way we can test that is we can delete RAG-GTPase, potentiate the formation of these beige adipocytes, and then treat with rapamycin, which is an mTOR inhibitor. And so if RAG loss is forming these beige adipocytes through an mTORC1 dependent pathway, it should be inhibited by rapamycin. And that's exactly what we saw. Now these are just in the cold, control versus RAG knockout, treated with rapamycin. You can see here the mTORC1 substrate S6K still phosphorylated in the cold, so no dependency on RAG. But if you treat the knockouts with rapamycin, you can see that the UCP1 protein is strongly inhibited, S6K is inhibited. So it does seem like UCP1 expression is indeed dependent on rapamycin. So the RAG deletion stimulated UCP1 expression is still mTORC1 dependent. And here's the histology to go with this. You can see the rapamycin-treated cells tissues have a lot more of these big white adipocytes, and there's a lot of immune cells, again, packed in there, where vehicle-controlled looked like an amazing browning effect in the controls. Now, rapamycin is given systemically here, so it could affect any cell type in the body. So we've also shown that this can occur in vitro in a cell-autonomous manner. So we've generated some RAG double knockout cells, and I'll just go quickly through this. These are in culture. If you stimulate the beta-adrenergic signaling pathways, which is what drives this browning phenotype, in the absence of RAG, you get a beautiful potentiation of UCP1. This is mRNA expression here, but we use this as a marker in this experiment as beige adipocyte formation. And so you can see RAG loss really potentiates that. Raptor blocks that. We can block that effect with rapamycin, and we can also block that effect with ERR inhibitors. And so we're still developing this cell culture system, but hopefully we can use this to get deeper into the mechanism. But what it's telling us is that in a cell-autonomous manner, RAG signaling seems to suppress this pro-catabolic mTORC1 pathway. And I'll end with the model, and then you guys can criticize it after. But what we think the data is telling us is that there is the classic RAG-dependent anabolic mTORC1 signaling pathway, but that there's this other pathway that's RAG-independent, and it seems to be upregulated in these cold conditions, which is when beta-adrenergic signaling is stimulating the formation of these beige adipocytes. And it seems that if you take away RAG, you allow this pathway to upregulate. So RAGs, in this case, might be functioning as a suppressor of this sort of pro-catabolic hidden mTORC1 pathway that hasn't been described before. So biologically, it kind of makes sense to me, because when nutrients are abundant, you want to store energy and lipids. RAG mediates that process. But when nutrients are low, or when you want to turn on this catabolic pathway, you want to shut down this pathway, but there are still pathways like mitochondrial biogenesis that might require mTORC1 for that process. And so we're actively trying to solve the mechanism now. There's lots of huge questions here. New biology in mTORC1 signaling. What are the signals that stimulate this? We think it's beta-adrenergic signaling, possibly through cyclic AMP PKA pathway. Where is this happening? All this mTOR-dependent signaling is happening in the absence of RAGs, which localize mTORC1 to the lysosome. So where is this signaling? We need to figure that out. And what are the targets? So we've got some candidates to look at, S6K, ERRs. We actually found in ERR there is an mTOR and an S6K phosphorylation site, so that's an interesting candidate. And previously it's been shown that S6K can phosphorylate PGC1 alpha to drive programs similar to this in the liver. And so we think there's translational relevance here as well, because we have a new pathway to target where we can turn on catabolism and maybe leave the other important mTORC1 pathways alone. And at UMass, we do a lot of research in RNA therapeutics. And with siRNA-based therapeutics now becoming a reality, I think there's ways to go after almost anything in any tissue in the future. So we're pretty excited about this. And I will stop there. And I will just say a quick thanks to Qingbo Chen, who's done all the work on these adipose knockouts. And I know we're behind, so thank you for your attention. And I will be outside after, take some questions if anybody has them. Thank you. All right. Oh yeah, I'm the moderator. Okay, so now we have a coffee break. We're a little bit behind, so are we going to do 15-minute coffee break? So be back here at 11 o'clock, roughly?
11:10.
11:10? 11:10, okay.
We'll try to
Thanks.
speed up the program. Thank you. Coffee break is in the atrium near the poster session and exhibition. And we will be waiting everyone back.
поинтер есть? Вот видите, оно вообще маленький, пропадает. Да, так что я здесь оставлю только одну. Раз. Ну, думаю, сейчас не надо, да, менять батарейку? Кажется, все нормально. Да. Если что, вот те два, которые сразу поменяем. В один здесь, да? Ха. Они воду не пьют?
трибуна, мы можем здесь. Вот этот микрофон. Ну, он так... Жмете, он загорелся и начал работать. Первый раз, когда вы кликаете, он чуть-чуть больше. Не, замечательно. Теперь смотрите. Кликер работает на LED, поэтому ориентироваться нам нельзя. Поэтому вот этот суфлер не ориентируйтесь, ну, как бы он будет небольшой дилей. А он вообще зачем? Он вообще там время еще высвечивается, там будет написано 15 минут, но там, знаете, здесь никто вообще никакого времени не делает. Вышел память, будет ее секция, она будет просто самостоятельно. Конечно. Да, но там будет время, потому что мы сначала будет мероприятие киновскую форму, и потом еще 5 минут некоторые какие-то горы по полчаса.
I was expecting Italian. No,
Yeah.
I didn't know Julia speaks Italian.
Yeah, I also speak Italian a little bit. It's okay. То есть вот это вот опаздывает, правильно? Да, вот этот экран опаздывает. Как раз он готов.
use it is what I'd rather have, actually.
So you had the two options, see? Then you can use this one and it works.
Okay.
And in this case, like, it works also.
Okay. Oh.
Yeah, it works.
So can we turn that off? That's off, is it?
A bit tight.
Hi, Julia. Very nice to meet you. We're going to introduce it. Thank you.
Hello, everyone. So if anyone is left somewhere in the halls, please call them. And it's my pleasure to start this session of regenerative medicine and bioengineering. We are glad to see you on behalf of organizing committee. We are really glad to welcome every guest here. And today our session is opened by Professor Giulietta Rao. She is Director of Research at the Institute of Structure and Matter from Italy. And her title of presentation is Emerging Biomaterials in Medicine, Antimicrobial, Bioactive, and Implantable Solutions. Thank you, Juliette.
Thank you very much, Julia, for presenting me. So it's my great, good morning, everyone. It's my great pleasure to be here today. It's my first time in Kazakhstan, and I hope I will come again soon. So my presentation will be about biomaterials in medicine, and especially focusing on antimicrobial and bioactive properties. I'm from Rome, from the Italian National Research Council. Italian National Research Council is a big organization which is divided into departments, and we have several departments, for instance, environmental technologies and earth sciences, bioagri-food sciences, chemical and physical sciences, biomedical sciences, engineering and transport, human, social sciences, and cultural heritage. And my institute belongs to physics department and is called the Institute of the Structure of Matter. We have two research areas in Rome. One is Monte Libretti, and another is Tor Vergata. Tor Vergata is near a very big second university in Rome, Tor Vergata University, and we have several institutes here in this big area. So I would like to talk about biomaterials and their applications in clinics. And what is a definition? Let's start with a definition. It is engineered materials designed to interact with biological systems, supporting different functions like therapeutic, diagnostic, or regenerative functions. And there are several types of biomaterials, and main types are polymers, hybrid materials like composites or nanomaterials, ceramics like hydroxyapatite or bioactive glasses, and metals like stable titanium alloys or biodegradable alloys. Just a few words about the history of biomaterials. In 2000 years before Christ, humans used some linen pieces for wound healing or metallic wires. Then 200 before Christ, iron artificial teeth were found. Then in 16th and 19th century, metals started to be used for hard tissue applications. Then in 1931, there was first application of polymer as biomaterial. In 1950, Some metal alloys were applied, like titanium or cobalt-chromium alloys. In 1970, bioactive or resorbable materials started to be applied, like ceramics, polymers, and so on. And from 2010 and even before, we talk about biomimetic materials, hydrogel, 3D bioprinting, so personalized medicine, engineered tissue and organs, and so on. So what are the current challenges of material scientists today? There are still many problems to be used, and even if still the focus is on biocompatibility and bioactivity as a priority, we need also regenerative potential. We need to minimize inflammation and rejection of material, which happens. A very important issue is antimicrobial potential. Then there is also personalization, personalized medicine, like 3D printing, nanotechnology. And of course, a very important point, especially nowadays, is sustainability and cost-effective production. So when trying to design new materials, we always talk and try to have a discussion with clinicians. And here you can see, just to show you a very important point, like antimicrobial properties of materials. Here there is an example of prosthetic joint infections in orthopedic surgery. This is remains one of the main problems for orthopedic surgery, and there are some numbers you can see here, like infection rates, 4%, about 4% in elective surgeries, up to 33% in open fractures, and especially this is a problem for elderly people, because it happens often that there is infection problems in elderly people, and taking into account antibiotics resistance problematic, we definitely need to create new materials which can solve this problem. And also the treatment costs. You can see here some numbers from United States, for example, what it costs annually to solve this problem. So in our group, we work on ceramic materials and on metals and metal alloys. Ceramics can be divided into two classes, like bioinert ceramics, which is used for many years already in clinics, for instance, in dentistry, and also in orthopedics. And then there are bioactive materials. In case of bioinert materials, we only mechanically attach our material to bone or other tissues. In case of bioactive materials, it is different because we attach it to bone not mechanically, but by chemical interaction. And they also can be bioresorbable or non-resorbable materials. And here you can see the main classes of bioceramics we develop. They are based on hydroxyapatite, which is resorbable, which is non-resorbable because for some applications we need non-resorbable materials. Amorphous calcium phosphate, which is more resorbable with respect to 3-calcium phosphate. 3-calcium phosphate, which is more resorbable with respect to hydroxyapatite, and so on. And so then we talk about bioactivity. We need bioactive materials, as I told you at the beginning. And what is bioactivity of material? There are some processes which should happen if we develop bioactive materials. It's like dissolution from ceramic, or dissolution of ceramic material, precipitation of some compounds on ceramic material, ion exchange on the interface between ceramic and tissue, interdiffusion. A very important point is solution-mediated effects on cellular activity, cell attachment and proliferation, cell differentiation, and extracellular matrix formation, and so on. And so here, and another class we develop of materials is titanium and its alloys. Titanium is very widely used in medicine in many applications for the body, for human body, and also for animals. But still, it has some problems because this material is inert. Or it may happen that biofilm formation, biofilm forms on the surface of titanium implants and so on. There can be allergic reactions because it is a metal and it is first generation of biomaterial. So we need to improve the properties of titanium. And what are the key points of metallic bone implants like titanium or also biodegradable implants? It is increasing demand for orthopedic implants due to trauma or other bone-related conditions, and also population aging. The more we have elderly people, the more implants we need. Requests for implants are long-term stability, good osseointegration, and infection-free surface. And limitations can be secondary surgery, because if we have infection, we sometimes need secondary surgery due to infections or due to the fact that we simply need to remove some metallic part when the bone is fixed. Or bacterial infection, biofilm formation, as I already mentioned, and implant failure due to insufficient bone formation. Then, what is the alternative to stable metal alloys? The alternative can be biodegradable metals. This class of materials is appearing nowadays as research, but in some countries it is already applied in clinics. The definition is metal that degrades in the body over time, promoting bone healing. An example is magnesium alloys, zinc alloys, and iron alloys. Advantages that, for instance, in orthopedics, that there is no need for removal surgery and reduced long-term complications, and challenges is the degradation rate, because sometimes metal degrades too fast, and we need to maintain its mechanical properties for a longer time, mechanical strength. And here you can see some iron stents applied for cardiovascular surgery, or pins which are applied to fix the plates for orthopedic surgery, and so on. But one of the main problems of biodegradable alloys is the point that it degrades too fast. And we need coatings materials in order to decrease the rate of degradation of biodegradable alloys. Coatings can also regulate and improve biocompatibility and can reduce inflammatory reaction. And the main function of coatings is to ensure a balance between degradation rate and bone healing. So main types of coatings can be ceramic coatings, and in our group we develop ceramic coatings. They are based on hydroxyapatite, substituted calcium phosphates, or also bioactive glasses. Polymer coatings that can offer flexibility and control over degradation. It can be polycaprolactone or other polymers. And composite coatings which combine the properties of ceramic and polymer coatings. And also in our lab, we focus on biomimetic properties of coatings and antibacterial potential. When talking about biomimetics, I would like to go back in time and to mention here Leonardo da Vinci, which was a great engineer, scientist, and also painter, and so on. A really great, one of the most important people in Italy, one of the most important scientists, and so on. And he was, because we often say that the term biomimetics appeared like, let's say, 20 years ago. But it is not like that, because already Leonardo da Vinci used to say that we need to be close to nature and try to copy or to reproduce some natural structures. Here you can see one of them, and so on. And indeed, if we talk about biological hydroxyapatite, it seems to be a container for microelements, because here you can see hydroxyapatite. It has various substitutions in its structure, and biological apatite has a lot of ions in this structure. So it's full of ions, and each one has its particular function in the human body. And then these ions substitution into the structure, they influence the bioactivity which we need to promote for biomaterial. They influence the surface chemistry, solubility, morphology, and so on. So we start from here. from this biological appetite. And indeed, in our group, we developed many various coatings for titanium based on substituted hydroxyapatite with all these ions and 3-calcium phosphate in case we need resorbable coatings, for instance, for biodegradable metals. And we also introduced some antibacterial ions in order to promote antimicrobial properties for our materials, for our coatings. So we start with synthesis in our institute, in our group. We start with synthesis of the materials. Then we produce also ceramic materials. Then we deposit our new materials using various physicochemical techniques like pulse laser deposition, magnetron sputtering, and so on. We characterize our materials with a set of physicochemical techniques which we have in order to get some surface characteristics important like topography, chemistry, wettability, and so on. And we collaborate with other groups for in vitro cell test and microbiology test. So, and these are a set of materials. These are biomedical cements we developed for, as you can see, some of them have antibacterial, but not only, but also other properties, like cements, which have in its composition zinc is antibacterial and has also osteo and angiogenic properties. Copper is toxic for bacteria and beneficial for healthy cells. We also developed cement containing iron 3-calcium phosphate, which is antibacterial and beneficial for magnetic hyperthermia therapy perspectives. And also strontium is a very interesting material, and I will show you on the next slides. So here, as an example, we developed manganese-containing 3-calcium phosphate ceramics because manganese is an essential element in the human body. And after we characterize our material and find a suitable concentration of manganese to introduce into 3-calcium phosphate, we perform, in collaboration with other groups, cell tests and bacteria tests. In that case, it was depending on the concentration of manganese, and here you can see some results when manganese was effective against gram-positive bacteria, and the effect was more intense for gram-positive rather than for gram-negative bacteria if we look on these numbers. Then this is also very interesting material when we introduced strontium into hydroxyapatite or 3-calcium phosphate structure. So why strontium? Because strontium is actually already present in the bone, especially in young bone. It has 3 or 4 times higher concentration in young bones than in old bone. And also strontium is already actually applied for in drugs. Strontium ranelate is known as drug for osteoporosis and so on. So we introduced strontium, and this here you can see on this slide the coatings, the morphologies and topography of strontium hydroxyapatite coatings deposited on titanium. Then we also performed, as we usually do, MTT test and also evaluation of osteogenic differentiation on the surface of this coating. But what is even more interesting and unexpected, that strontium, which is known to have osteoinductive properties, we discovered it to be also antibacterial. This is very, very interesting fact. We performed this investigation together with Orthopedic Institute of Rizzoli. They have a microbiology lab, and what was unexpected, that on the surface of strontium 3-calcium phosphate coatings, you can visibly see that there are less bacteria adhere to the surface, both after 4 hours, and the effect was even more pronounced after 24 hours. So this was unexpected, and we performed also several tests, like bacteria adhesion on the surface of the coating and biofilm formation on the surface of the coating. And so the conclusion was that this is a very interesting material and can be applied in clinics. Then the next group of experiments, then as a part of research which we perform in our institute regards biodegradable alloys. And we coated these alloys with hydroxyapatite coatings and with glass ceramic coatings. And this was important, and we perform now also tests, bacteria and antimicrobial tests in this period. Then we also collaborate with the Chinese group for zinc biodegradable alloys. As I told you, these alloys in some countries are already applied in clinics like Singapore, and I know in China. And the pins for orthopedic applications are made of biodegradable alloys. And in case of zinc, and I know the Chinese group also develops cardiovascular stents based on zinc, we worked for orthopedic applications, and our goal was to develop coatings for zinc biodegradable alloys. And so we decided to introduce into 3-calcium phosphate not one ions, but 2 ions. In this case was copper strontium ions, and we got some antibacterial properties. And also another couple was manganese strontium coated zinc biodegradable alloys. But what I would like to stress here is that actually we performed both theoretical investigation of the different couples, because we need also to optimize the concentration of these ions in order to be not toxic for cells. They should be toxic for bacteria but beneficial for cells. So we investigated these 3 couples and then performed also antibacterial tests. These are experimental data. And the best couple from the point of view of antibacterial properties was copper strontium. Indeed, this couple of ions, when we introduce them into 3-calcium phosphate, they possess antimicrobial activity resulting in more than 90% inhibition of bacteria growth. This is a very good result. We published it, and we decided also to prepare granules made of this material, and we collaborate with Kazakhstan, with Yulia Safarova and Shalpan Askarova in order to investigate also the biological properties of this couple, because we think that this material can be applied, we should move it forward in order to be applied in clinics. It's a good material. Indeed, also the biological test produced very good results. Various tests were performed here in Kazakhstan, like alizarin staining, like proliferation assay, and also were very important were angiogenic assay, and these are results of this assay. So this is a very good material. And also now we are talking with a group from Kazakhstan in order to perform also in vivo tests. Then we don't stop here. We introduce other ions. This is a collaboration with a group from Moscow University. We introduce also other ions, and as usual we change the concentration and see what is the best concentration in order to boost antibacterial properties. And also this material, we started biological tests, and we will continue to prove also with this material. So what I tell you until now was chemical approach. So we introduce some ions, and we expect the release of these ions from the surface of our material. But the numbers I showed you, I don't know if you notice, in some cases there was 100% of inhibition of growth of bacteria. But in this regards the bulk of material, but when we deposit as coatings, we have less material. So inhibition of bacterial growth is less. But we need 100%. We don't need the bacteria at all on our surface. So what we plan to do, we already started, but I don't have time to show you our results. We perform to do also micro and nano patterning on the surface. And in this case, we will have also physical approach. And synergetic approach of these two, chemical and physical, we expect to have a surface of our material free of bacteria. And again, going back to biomimetic properties and to the nature, here regarding nano-patterning and or micro-patterning on the surface, we would like to take the example of some nature structures. Here you can see the cicala wing, and here you can see some examples of micro and nano patterns on the surface of materials. Then, just a few words, I would like to mention that we collaborate also with other groups for another very important application of materials is for cranioplasty, and because nowadays, unfortunately, these materials which are applied in clinics is titanium. This kind of cranioplasty, these pieces are made of titanium or polymethyl methacrylate, which are materials of first generation, and they are absolutely not suitable, especially for young patients. If after a car accident or some trauma, young patients have problems, and titanium plates or polymethyl methacrylate pieces are used for their head, for their skull, is not suitable because young patients they are growing, and so they need then to have a secondary surgery and to remove this piece again. So what we work now, we developed together with a group from Naples, developed a material based on polymers, and we added also copper tricalcium phosphate in order both for osteoinductive and for antibacterial properties of this material. So here you can see some results of this research. And the directions where we're going to move is, of course, improving antimicrobial potential of our material, integrating some drug delivery functions, but not antibiotics because of the problematics of antibiotics resistance, and scaling up production of materials for clinical applications. We'd like to be close to clinical applications of materials. And also for this, we organize for clinical applications, and I will explain you now how. We organize this conference in Rome every 2 years. This is an international conference, a small, just a few hundred of people. So we organize every 2 years in October, and the next one will be next year. It's called Biomaterials and Novel Technologies for Healthcare. And the main goal of this conference is to boost our materials to clinical applications. Indeed, we organize sessions together with clinicians, like orthopedic session or like cardiovascular and so on. There are many sessions of dental materials. We invite clinicians and we discuss about the problems which they have nowadays in order to be to speak the same language, at least what we try to do, to speak the same language. It is not so easy to understand each other, and to develop something which is really useful, not just materials to publish papers and so on, which is often happens. And now I would like to thank you all for your kind attention, and these are beautiful images of a very beautiful country, which Italy is.
Thank you, Professor Rao, and we have 5 minutes for questions. Or we can save it up for the lunchtime. Any questions from the audience? Okay, then I have a question. So how long usually does it take to process from in vitro to clinical practice? I mean, in Italy, what is the experience that you have like in your institution?
No, it's a long journey. Because in vitro that we need to perform in vivo, at least with our materials, because not for every kind of materials it is necessary to perform tests with animals. But for our, especially for orthopedics, it is necessary to do. And this is a very long journey, especially in Italy, because there are many regulations, and it is difficult to organize in vivo tests in Italy. Really, is difficult. But
okay, this is a really thing.
Yeah, but now
really
we have
topic
actually
in
a
Kazakhstan
project
as well.
with Campus Biomedico in Rome, which is half private university, and they are very fast, very good organized, and we have project which has just started, and we hope to be faster. So,
Thank you,
but
Professor.
I can't tell you how much time it needs, but years, let's say.
Years, yeah, it's usually 7 to 10 years, like that's what we encountered here in Kazakhstan, and I think we can discuss it during the lunch. So thank you a lot, Professor Rao, and I just want to mention that we have a long history of collaboration with Institute of Structure and Matter, and it's a great pleasure to have you here as a keynote speaker. Thank you again. And we are moving to our next speaker is Professor Primsin from School of Medicine, Nazarbayev University, Kazakhstan. Thank you, Professor Primsin. And it will be about age reprogramming, cell rejuvenation by partial reprogramming.
It's always good to start off with a slide like this. One should never take anything for granted. In the middle here is my name, and I'm going to be talking about age reprogramming. And before doing so, I thought, by way of background, I would give a little review of work of individuals on whose shoulders regenerative medicine actually was built. And this is in the next slide. And these two individuals won the Nobel Prize in 2012. And to my right and your left is John Guertin, and he was one of my old professors when I was a student in Cambridge. And we have Shinya Yamanaka next to him. And I'll just give you a brief review of what work they did to win the Nobel Prize. This is John's work, and it concerns a technique called somatic cell nuclear transfer. And at the top there is Xenopus laevis, a clawed toad, and that female donated the eggs from which the maternal chromosomes were removed, and a somatic cell nucleus was placed into that egg from an albino donor. And you can see what happens when that egg then develops. You can see all the individuals below it there, which are the same genotype as the donor. Now, what John concluded from that was two things. The first is that differentiation is not due to gene loss. The reprogramming of that differentiated nucleus back to an embryonic state could result in the recapitulation of development. And the second thing, and they were the two things actually. The second bit is that recapitulation of development from that differentiated cell type, and it's not due to gene loss. It's a change in the epigenetic state of that cell. Now, this work was done in frogs, of course, and the work that came from that, which actually revolutionized regenerative medicine, with this work on Dolly. And I was very fortunate to join this group just after the time they had actually cloned Dolly to work on the molecular mechanism. And why was this a revolution, you ask yourself? Well, the reason was that what it allowed is allowed the production of stem cells, and these stem cells have the ability to self-replicate, to self-renew, and also differentiate into any cell type that you wish. And this is how the experiment's done. If you start off on the left there, you take out the maternal chromosomes, you introduce the donor cell underneath a proteinaceous structure called the zona pellucida, and then you undertake electrofusion, and the very act of electrofusion results in activation of the reconstructed embryo, and then you get a ball of cells. It undergoes geometric doublings, 2, 4, 8, 16, so forth. And you get this ball of cells. And I've depicted a ball of cells there. It's inside a structure called the blastocyst. But that ball of cells, you can do one of two things to it. One is to put it into culture, which allows you to make large quantities of it that can differentiate into many cell types, and you can see the usefulness of that. If you took it from a donor, you could get any cell type you like, and then take it and give it back to the donor. Or the other thing is to implant it into a pseudo-pregnant mother, and you get a clone. And of course, that's an ethical problem when it comes to human beings. And that's something that people dwelled upon for a long time. And the next piece of work by Shinya Yamanaka actually overcame that problem. Because what Yamanaka did was to ask the question that if you have an embryonic cell, there must be factors within that cell type which make it what it is. And if we could search for those factors, perhaps we could then take them and force their expression in a differentiated cell to make it an embryonic cell. And that's precisely what he did. And he started off with 20 candidates and boiled it down to 4, which are given at the top for OCT4, SOX2, KLF4, and c-MYC, OSKM. And if you put these factors, unfortunately I was told by the gentleman there that the pointer doesn't work, so I'm going to have to just, in narrative, tell you what's going on in this slide, is that if you put those factors, say, in a skin fibroblast, say from somebody who has a genetic disorder, and then you could express them, and that's what's found in the middle there, and you can get these stem cells that you find on this side here. And they're called induced pluripotent stem cells. Now, these stem cells have that capacity for constant self-division indefinitely, and also can differentiate into whatever cell type you like by directed differentiation, which you see here. And if you correct the genetic defect here, you can give the cells back to the donor. Now, one of the problems is, as I've mentioned to you now twice in 2 slides, is that these can indefinitely divide. Now, when you differentiate these cells into these different cell types, there's always a small proportion that remains like this, and that's always been a problem. One of several problems, but a major problem is this one. And that problem, because there is another cell type that divides indefinitely, and they are cancer cells, so that's the problem you have to face. And so this method, while it rallied a lot of troops and many people work on it, in fact, it's been something that's been very difficult to apply clinically. So how do we get over this problem? So I put my little thinking cap on and I said, why don't we try something called age reprogramming? But what in heaven's name is it? Well, let's go back to the Dolly the sheep experiment. If we go back to the Dolly the sheep experiment, Dolly was cloned from a mammary gland epithelial cell from a 6-year-old animal, which is middle-aged for a sheep. The nucleus, the cell was taken from here. It was placed under the zona pellucida of the egg that was taken from a black-faced sheep. You notice that sheep's got a black face. This one's got a white one. And then you electrofuse. That very act leads to the development of the reconstructed embryo. There's the blastocyst. There's that ball of cells in the middle. Then you put it into a surrogate mother, and you get Dolly. White-faced, white-faced. So it's from the donor nucleus. So that's a 6-year-old cell, and Dolly is a newborn lamb. So there is a formal process for being able to rejuvenate cells, but you have to go through that embryonic state. Now, the same thing happens when you do iPS cells. On the top left there, your left, you can see the somatic cell from a young individual, and that cell type has certain characteristics, age-related markers, which are peculiar to the youthful state. You can also take a cell line from somebody who's 100 years old, and you can actually reprogram them. These factors that you see have the canonical Yamanaka factors and 2 more, nanogun LIN28, in order to actually promote the reprogramming effect. And here you find there's that ball of cells, those iPS cells. Now, curiously enough, if you take those cells and differentiate them again back into fibroblasts, they're suddenly youthful. But you have to go through this embryonic state. Now, ask yourself the question, would it be at all possible? And usually the way the way it's normally done in England is that you do this sort of thing on the back of an envelope. So if you have, there's somatic cell nuclear transfer, how Dolly and the frogs were created, and they're the Yamanaka factors, including the 2 non-canonical non-ag and LIN28. And there's the de-differentiation into the embryonic stem cells. And if you redifferentiate, you become young. I've shown you Dolly's young, and also that centenarian. Now, what if you just draw a line from here to here? Can you cut that out? Can you cut that out? What do you think? Yeah, why not? Yeah, sure. That's what I felt. And the funny thing is, and I have to be frank with you, the funny thing is, the older I get, the more I get interested in rejuvenation. I can't see the connection, but it's all true. I just get more and more interested in it. So how do we test this idea? How do we test this idea? So the way we tested it is you take an old cell, put the factors in, and then on the trajectory from the old cell to the iPS cell, is there a state at which the cell retains its differentiated phenotype but becomes younger, so it becomes aged reprogrammed? This interruption is partial reprogramming, partial, not full reprogramming. Does that state exist? So, and the way we tested this was actually use a biophysical technique where what we did was GFP, we fluorescently labeled a protein that is involved in packaging DNA. And it's a nuclear protein, and what you can do is you can irreversibly irradiate that protein in a certain area of the nucleus so it doesn't fluoresce anymore. And then what you do using time-lapse fluorescence microscopy is look at the recovery of the fluorescence. And interestingly enough, and again, red is young, blue is old, but blue there's got the OSKM factors. Then we have a look at the mobility of the protein over time. And what you find is that the red line, you can see that the protein recovers very quickly in young cells. That means it's moving around, it's lithe, it's quick on its feet. And as you get older, you get a bit slower. We all know that feeling. And it gets a bit slower, it takes longer. But if you get to 9 days, you can see the overlap. So that means the way the chromatin is, it's got a youthful phenotype, yet that cell is still a fibroblast. Using this procedure for senescent cells, it takes 49 days. So it's to get to an iPS cell. So it remains as a fibroblast, but it shows that age reprogramming is separable from, is the first indication that is separable from developmental reprogramming. Subsequent to that, and I'm only going to give you the bottom part of this slide, they used a transgenic mouse system, DOX-inducible, which was constructed by a group in Barcelona under Manuel Serrano, Abed et al. It's a well-known paper where they used this system and introduced into it a lamin A knock-in, which gives rise to prematurely aging mice. And if you cyclically express 3 days on, 4 days off, 3 days on, you actually make the mice more youthful. In fact, that's the case with naturally aged mice, not only the lamin knock-in mouse. And you can increase the lifespan by about 15%. So if you express this, so the notion that age and developmental reprogramming can be separable actually acts at the level of the whole animal. And the question I'm sure you're asking, is there a dollar value associated with making old people young? I will come back to that. So the next experiment was an experiment done, that experiment, I just go back there. The individual is Alejandro Ocampo, who's at the Salk Institute, who is a competitor, but an honorable gentleman who's done some great work in the area. So at the Salk, which is a well-known institute. And and I will come back to his name actually a bit later. And this is work that was done at Harvard, where in mice, if you look at them in cages, sometimes they're often blind because as they get older, they do go blind. And it's usually due to a problem with the retinal ganglion cells. But if you put in the Yamanaka factors and inject them in every so often, you can actually they can gain their sight back again. So the question we ask ourselves, and this is that chap Ocampo from his lab, and he's a competitor, as I mentioned, so this is not my paper. And what he did was list the papers as they, and you can see at the top there is John, my old professor, and then Yamanaka and Wilmot, and you can see as you go down the contributions that have been made. But what I'd like to talk to you a little bit is about the commercial landscape. So the commercial landscape is that increasing health span in elderly comes to 38 trillion. That's the cost that, in order to, and at the moment, very recently, there's been a lot of interest because there is a dollar value associated with making old people young. And the individuals, and there's a new lab which has been set up in Cambridge and in Stanford in the US by Jeff Bezos for $3 billion. That's his initial, as Ocampo says, not me, that's the largest by more than computing, more than AI, more than all the rest. It's the largest investment made. So how are we getting on and what are we doing? And this is one PTF grant that we've recently completed, and with some very, very kind and illustrious colleagues of mine, and also some individuals in Germany. And we've completed that, and our outputs were this. Rather than give you a horde of references, and we've actually been awarded a second PTF grant now, which is there are very large grants. And our focus there is on epigenetic rejuvenation, which is a main driver of age reprogramming. The way the DNA is packed and how it is eroded with time is the main driver. If one does not rejuvenate the epigenome, all other hallmarks of aging are stopped. So we are focused on this, the most, which we think to be most important. How DNA is packed is, of course, important. So thank you very much.
Thank you, Professor Primsingh, and we still have some time for one or two questions. Any questions in the audience? Yes, please. Can you raise your hand again? Yes.
Good afternoon. My question is, I'm Aliya Sikenova, National Center for Biotechnology. Thank you for your presentation. My question is there can be a warning on that partially differentiated cells can be de-differentiated to cancer cells?
Pardon?
I mean that if there can be a warning that partially differentiated cells can be differentiated to cancer cells, because we know that embryonic stem cells, if we use them if we want to use them for clinics, just there is a concern that they can de-differentiate to cancer cells.
It's true that iPS cells can give rise to cancer cells, fully differentiated cells. I showed you an experiment by the gentleman Biocampo et al., which was that experiment only used partial reprogramming. Those animals did not show any increase in cancer. So you only use it partially reprogramming. The notion that actually drove me to that idea was actually to avoid that. The only partially reprogram. It was a long shot that age reprogramming and developmental reprogramming could actually be separable, because on the face of it, they seem to be processes that are seamless. They occur one and the same time. The fact they are separable was extraordinary. I'm still baffled by it now. But it does actually happen, and many people have done it now. So partial reprogramming in those mice, that's formal proof of the notion, does not lead to an increase in cancer.
Okay. Thank you.
Thank you very much for a very interesting talk. A question, so how many years, like in the future, how many years do you think a life could be extended using this technique?
I think the idea is to, rather than a gradual degradation, that you live for a long that lifespan is healthy for as long as possible, and then you suddenly the light goes off. People have sort of estimated that at 120 to 150. Okay. And it'll come down to the physical properties of chromatin, is my view. I've just written a mathematical paper on that, in fact, about how one has to, you can think actually of chromatin in terms of something called a block copolymer, and how that is assembled and how it is maintained in a manner which enables the cell-to-cell inheritance of gene expression patterns is going to be key. That's why we focused on epigenetic rejuvenation, because how the DNA is packed ultimately will be the key. Once that goes, everything goes. But that's the limit people have placed on it. It's a guesstimate, of course, and and without firm evidence can be safely ignored.
Sure. And do telomeres play a role there as well with the limitation? Do telomeres play a role?
Well, the telomeres have an effect, of course, in humans because we have short telomeres. But the actual best clock for measuring age, biological age, as opposed to chronic, because normally we measure our age by the length of time it takes the Earth to go around the sun in years, but that's far removed from biological age, of course. And that the best estimate for biological age is a change in the epigenome and DNA methylation, not telomere length. Yes, there was a question there.
Okay, thank you. Sorry,
we are over time,
we can talk during during lunchtime, okay?
I'm around for a while.
Yeah, because we need to go ahead.
Information, I'll be around a bit longer.
Thank you a lot, Professor.
Thank you very much. So our next speaker is Dr. Johannes de Munter from Maastricht University, Netherlands, and his presentation is entitled "The Challenges of Performing a Phase 2 and a Phase 1 and a Phase 2 study with an autologous stem cell product, neurocells, to combat neurodegeneration." Thank you very much. The floor is yours. I can give you mine.
No,
the pointer.
Where is the pointer?
It was.
First, congratulations to the organization for the university, and thanks for giving me the opportunity to talk to you. And I'm a little nervous because just before me, someone is telling the future of cell technology and regeneration, and I'm using autologous non-manipulated cells. Dus it will be very difficult to compete, but I will try. Before I start, I want to disclose. I'm a CEO of a company. That's my business. But above all, and that's why I stand here, I'm a desperate scientist looking for a solution for neurodegeneration. And you see that I'm not young, so maybe to complete my mission, I need your technology so that I get more lives to do more research. It's a journey of a long time, and a journey which a lot of universities helped me establishing that. But before I start, I have a question to all of you. Did you know that 1/3 of the world population will suffer a neurological disease? And if you look carefully in your family and put only your hand up, can you remember a family member, a friend, which suffer a neurological disease? Alzheimer, Parkinson, spinal cord injury, traumatic brain injury. May I have your hands up? Only one? Okay, that's better. There's hope. What are the cells? We gave it a name, neurocells. But it is your own repair cells. It is what's in the bone marrow. And it is a fast procedure because it is important for cells, if you don't change them, that you speed up. The only problem in humans is that we have a protective barrier around the central nervous system to protect us of infections. And stem cells are so big that they cannot trespass in enough numbers to the brain or to the spine. We have to help a little, and that's what I did. What's in it? It's a mixture. And that is maybe a little strange that I tell you about the mixture, but if you repair something, you need cells who can repair vessels. You need cells who can maybe influence the immune response. Spinal cord injury, that's the issue which I would like to address, is a multifaceted disease. And one cell cannot do the job. So that's why we selected the bone marrow and take a mixture of cells. Important for us was to keep the silent condition which is present in the bone marrow as long as possible to the cells. And that is the diseases which I'm focused on. 1/3 of the world population. And you know the phases, but I will start with the most difficult one, spinal cord injury. This is all the preclinical research we did, and this university helped with accomplishing my preclinical issues. So I'm now addressing to phase 1, phase 2 in real patients. And the challenges which you have to face when you go with a cell product to a patient. I thought it was simple, but it is absolutely not simple. This is the mode of action. I'm not replacing cells. I'm not going to make new neurons. I'm not going to make a new spinal cord. The only thing what I want and try is stop the collateral damage. We know that after an incident, there is an ongoing collateral damage by secondary inflammation. Just to give you an example, if you get a motor accident, you get paralyzed, 10% is caused by the incident. 80%, that's my interpretation, is caused by the inflammatory process. Can you imagine? You make a crash, 10% gone. Then you have to wait. You wait, you wait, you get revalidated, and you lose more cells. You lose more neurons. And that's what we did. That's a part of my PhD, which I did very late. First you have to see the light, and then you start doing your PhD. Anti-inflammation, that's what I wanted to present. Can I heal? No. Can you do marathon walking after a spinal cord? No. But maybe I can save 10% of your long fibers so that you've been able to walk or stand, because you only need that. Most important is, and it's a question to all of you, what do you think is the most quality of life impacting symptom of spinal cord injury? Is that walking? Put up your fingers? No. It is your autonomic nervous system. If I ask a patient, what would you prefer, walking or controlling your bowel? He said, controlling my bowel. I'm used to sit in a wheelchair, but the most devastating thing is I'm sitting in a meeting and I smell myself because I cannot control. I don't have the courage to go to people. That is really impacting. And then part of the spinal cord injury patients, and I don't know if you know that, get a stoma because the whole bowel system is paralyzed and not functioning. This is what we did, multi-center with autologous stem cells. I'm located in the south of the Netherlands. That's a very small country with a lot of people. And we did the study in Denmark. That's around 800 kilometers to the north, and Toledo, that is 2,200 kilometers to the south. Very good centers. And that are the two investigators. And if you see the approval, I submitted the approval, and it took me nearly 3 years to start with the clinical trial, because they think that autologous stem cells are toxic. You have to provide a lot of evidence. But we did a placebo-controlled, randomized, double-blind study with a living cell, with living cells from the patient itself. That was a real challenge. We have small numbers, I agree. But we did a 6-month period of complete blindness. No, the patient doesn't know what the patient got, and the physician doesn't know what was given to the patient. So we worked with 2 teams. That was, by the way, a design which was approved by the European patient organizations. And now I will tell you the results. You see the first two that are changes to baseline. And baseline is when we started the treatment. And the treatment was given 6 to 10 weeks after the incident. And that was in the period that there was corona and that there was a quarantine policy before patients were transmitted to another hospital. Can you imagine? Patient comes in, I take bone marrow, fly it to the Netherlands, process it, give it back within 24 hours. That is the policy. That was what we wanted to do. And that's what we did. You see in the blue, that is in testing. That's the gold standard for spinal cord injury, meaning that they test your upper limbs, your lower limbs, and that's tested. You see a small, the red one is the placebo, the blue one is the treatment. It followed up at 90 days, that was the difference, 180 days, and after a year you see still that it's growing, but the placebo received a late treatment. So it's a design with two answers. Placebo controlled for 6 months, and a late and early administration form. And now you say 5 points, 4 points. What does that mean? And I know that people want me not to use my finger, but I will use this one. What can you do? This is 4, 5 points. You can do, what can you do with it? Physician asked me, what can I do with it? I can use a wheelchair. I can go myself. I'm not depending. That is what it impacts to patients. Don't forget that. For me, using one finger when you have 10, no problem. Then I cannot do this. I can only point. But if a patient who cannot use anything can use a finger, that's a major improvement. Sensibility, that was significant. You see the spread, you see the small numbers, so it was not right to put only bars. I want to give you the individual date. This was bowel management. That's an independent, validated screening instrument, seeing what you can do, how you handle your bowel complaints. Significant after a year. That's one test. The second one is more interesting, is a dysfunction test. The higher the score, the worse your condition. And it was significant improved, not only between the two groups, but also the progression in time. This is really impacting the life of the patients. Then the other one, neurogenic pain. If you ask a spinal cord injury patient, I know, are you familiar with phantom pain? Phantom pain. It's a kind of neurogenic pain. I think in spinal cord injury, it's not the same, but there is an enormous pain sensation which often asks for morphine. We improved that condition. And now it is more difficult. Did I improve the condition or did I not worsen the condition, which was the case in the placebo group? That are the major things I want to share. And then a summary of all, it was safe. We have no adverse event reported. So your own cells injected in your spinal and your cerebrospinal fluid by lumbar puncture was well tolerated. No side effects, no product-related side effects. So it's safe. We had strong, significant improvement for bowel and neuropathic pain, 8, and then strong significance. Remember, it is a very small group. We had even influence on vital functions. I don't know if you know, but in spinal cord injury, there is a tendency of a lowering, of an increased cardiovascular profile by lowering your heart rate and by lowering your blood pressure. And that fluctuates. That's not really controlled. What we see, there was a significant normalization of your heart rate. I didn't show that here. And there was a tendency of your blood pressure to normalize. So your cardiovascular risk, which is also very important in spinal cord injury patients, was positively influenced. And we did a lot of more. Most important is what the expert finds. We ask an expert, what do you think? Do we have to perform a phase 3 to get bigger numbers? He said, Guy, you're crazy. Those patients have no perspective yet. At the moment, there is no therapy which show those outcomes. I would prefer that you go to a real data, that you go to gather real-world data and not placebo-controlled. And that's now what we're discussing with the regulatory agency to get permission to do a phase 3, but then compare that with standard care. What are the lessons learned? Maybe you find this simple, but can you imagine that you do everything, then autologous product, and you fly back with your product? And then Madrid, that was the airport. There was a snowstorm. We couldn't land. So the plane has to go to a different airport. That is autologous. That is difficult. You have to be prepared. Then the customs services. We had a GMP facility checking everything. And then they said, okay, what's that? I open it, I look at it. Your cold chain is broken. Your seal is broken, autologous. We have a lot of quality issues. The incident, I underestimated, which normally is the case for every research with patients. I planned 6 months enrollment, and in general, because in the corona period, people were not allowed to gather, sport, do anything, there was an enormous drop of incidents causing spinal cord injury. So my enrollment period was twice as long as I expected. Then, what is the golden standard? The golden standard was not enough to measure the real progress. I measured the hands, my arm, my legs, but I didn't measure the breathing, my diaphragm functioning, my trunk, which was maybe influenced, but was not measured. And the expert gave me a list of tests, the patient asked something else. And then, and ladies and gentlemen, I think this is a real challenge for everyone. If you go to IPS or autologous or embryonic, it doesn't matter. But regulatory is asking so many quality controls that nothing is left to give back. And autologous, I will have a superb product because I test too much, but nothing to give back because I used it for release. That is a real issue. And then if you want to read something, everything what we did, we published. We published together with people from here, from all the universities collaborating, because I want to be as transparent as possible. It's not I have no spinal cord injury, but I want the therapy come into reach. That's my physician heart. I'm an emergency combat physician, wanted to influence something. And I think we are nearly there, I hope. Otherwise, I need yourself. And this is what I want to acknowledge, all the parties helping me, because I could not do it alone. There are a lot of universities helping me to achieve the parts we did. Oxford, this university, Moscow, all those parties helped me in finding evidence, doing preclinical, doing all the expertise. And now I have a lot of blood, I have a lot of liquor, and I address all to you, because you are the scientist, what is wise to analyze? And that's a question I have, and I hope that you can help me those days in defining what we can do with the body materials. I want to prove inflammation and regeneration. And I have a lot of samples from real patients. Maybe you can help me. Thank you.
Thank you very much. Thank you very much for your very interesting, very exciting presentation. Unfortunately, we are late and there is time only for one short question, but after, during lunchtime, you can ask as many questions as you like. Now, for just one question.
Hello, thank you for your presentation. It was very interesting. Actually, I wanted to ask, does your research or your healthcare services or your product you've mentioned at any point covers the cerebellar ataxia diagnosis?
The?
Cerebellar ataxia, one of the neurodegenerative diseases with the injury of the cerebellum.
Yeah, we did not really... it is difficult to detect the real regeneration in the spine, and especially in human. So we focused only on immune modulation, and there we had a lot of data for, and that's also in the preclinical. I know that there is also evidence that we do regeneration, but to claim that, and you need to be very careful. I can easily say, my cells can do everything, but that's not true. The only thing what I've proven so far is that it influences immune modulation. I know it's more, but it takes more research and work to find evidence for that.
Okay, thank you very much.
Okay, thank you very much again. Thank you very much. I would like to invite you now to the podium Professor Vyacheslav Agay from National Center of Biotechnology of Kazakhstan. His presentation is entitled Hydrogel-Based Delivery System for Regeneration of Articular Cartilage Defects.
Okay, we'll stay here. So, good day, dear participants. Today my presentation so in my presentation, I will present our recent development in articular cartilage regeneration by using hydrogel-based delivery system. So I will show the complete investigation from idea to clinical application. So as you know, large cultural defects remains one of the most challenging problems in orthopedic surgery, because articular cartilage defects, articular cartilage has a vascular structure and limited capacity for self-repair. So there are two types of defects: chondral defects, which penetrate the cartilage, and osteochondral defects, which also penetrate the subchondral bone. So currently, there are several biomedical methods, for example, to treat the cartilage defects, for example, multiple microfracture, matrix-induced autologous chondrocyte implantation, process allograft cartilage, osteochondral autograft, and allograft transplantation. However, this current method for treatment the articular cartilage defects lead to short-term functional regeneration for articular cartilage defects, and cannot provide sustainable restoration of hyalinic cartilage. So due to the limitation of traditional methods, tissue engineering strategies are particular interest, as they are capable of first promoting the regeneration of hyalinic cartilage and the subchondral bone, second, creating a favorable microenvironment for cells, and last one, supporting the localized delivery of bioactive molecules. So in this slide, we can see the main strategies used to design hydrogel-based delivery systems for cartilage repair. So many research groups use this strategy. For polymers, they used, for example, natural or synthetic biopolymers, for example, fibrin, alginate, collagen, gitazan, PEG or PLGA. And to deliver bioactive molecules such as growth factors, DNA, or viral vectors, so they use these polymers that loaded in this hydrogel, for example. They functionalize, for example, by heparin molecules or microsphere nanoparticles to retain, for example, this growth factor in the matrix. So this strategy is needed to effectively regenerate the hyalinic cartilage in the defect. So we also use this tissue engineering strategy in our study, and we develop injectable HCF hydrogel for articular cartilage repair. HCF, it is heparin conjugated fibrin hydrogel. Here we can see the composition of our HCF hydrogel. We use active components. We use signal-derived mesenchymal stem cells and two recombinant growth factor like TGF-beta1 and BMP4. And for jelly agent, we use heparin conjugated fibrinogen and thrombin. And we mix cells in equal proportion with heparin conjugated fibrin hydrogel and transplanted into cartilage defects. Here we can see just example of this, the prototype of our hydrogel. So gelation time about 3 minutes at room temperature. And the property of our hydrogel, HCl hydrogel, first, we found that our hydrogel are biocompatible, biodegradable. Hydrogel can control release of growth factor into defect area and significantly accelerate the repair of secondary defects. So next I will show our in vitro studies, in vivo studies, and clinical trials. So in vitro studies. First of all, so we isolate and characterize signal-derived mesenchymal stem cells from laboratory animals, from rabbits. So we can see the typical morphology of these mesenchymal stem cells, and we found that they can form the colony forming units based on CFU assay, and they can differentiate into 3 kind of specialized cells like adipocytes, chondrocytes, and osteoblasts. Next, we continue our experiment, and we study the effects of individual and combined application of TGF-beta1 and BMP4 on homogeneity of signal-derived mesenchymal stem cells. So we can see these results. We can see the histological sections of these androgenic spheroids after treatment by TGF-beta1, BMP4, and the combination of 2 growth factors. So we can see that the combination of 2 growth factors gave us better results. So it also was proved by biochemical analysis, for example, by DNA content in these spheroids, and by amount of glycosaminoglycans, GAG. Yeah, so we observed that TGBeta1 and BMP4 together can better effect on the chondrogenesis. So next, we synthesize heparin conjugated fibrinogen using carbodiamide chemistry. So we can see we use a low molecular weight heparin and activated activated by these chemicals. And after that, we make conjugation with fibrinogen, lyophilization, and characterization by high-resolution NMR spectroscopy. So we can see the results of this NMR analysis of HCF, and we found that heparin conjugated fibrinogen exhibits strong covalent bonds between heparin and fibrinogen. So next experiment was to evaluate biocompatibility of our HCF hydrogel. So we can see the prototype of hydrogel after gelation with encapsulated mesenchymal stem cells, figure A. And we use several in vitro methods like live data assay and cell viability assay, and we measure DNA content by biochemical assay. And we found that our synthesized heparin conjugated fibrinogen, fibrin hydrogel was intoxic. Because we found the proliferation between during the 7th day, so we can see on 7th day the amount of DNA was much higher than in third and third day. Okay. So in order to examine whether our hydrogel can retain or control the release of growth factors, so we use ELISA test and incubated our hydrogel during 4 weeks. As control, we use just fibrin hydrogel without heparin. And as we can see in this slide, so heparin conjugated fibrin hydrogel can control release of these two growth factors, and we can see the release of this BMP TGF-beta1 was released up to 4 weeks. So after that, we conducted preclinical studies. So we use laboratory rabbits for this study. And we divided laboratory animals into 4 groups. The hydrogel was as control, hydrogel plus MSC, hydrogel TGF, and BMP4, and hydrogel plus MSC and 2 growth factors. So by using a special medical device for chondroplasty, we performed the osteochondral defect and made implantation of our hydrogel with MSC or growth factors. So in figure C, we can see the implantation of our hydrogel into osteochondral defects, and in figure D, we can see gelated hydrogel after 3 minutes at room temperature. So after 3 months, so we examined our articular our joints, joints in experimental rabbits, made microscopic evaluation for osteochondral defect repair, So we can see the differences between 4 groups. So we can see that hydrogel with encapsulated MSC and TGB1 BMP4 result to both better regeneration in comparison to other groups. So after that, we made histological study and stained by different staining, including immunohistochemistry. So we can see 4 groups. In figure A, the histological sections were stained by hematoxylin and lysine. Figure B by safranin O to stain the extracellular matrix. Okay, and in figure C, the immunohistochemical analysis to stain collagen 2. So if you compare, you can see that in figure 4, in group 4, hydrogel plus MSC and TGB and plus BMP4, we found that after implantation this hydrogel, we observed hyalinic-like cartilage. It also was confirmed that by safranin O, because amount of the extracellular matrix staining, extracellular matrix was more better than in other groups. So the same result was also observed after collagen 2 staining. So in preclinical studies, we found that hydrogel with MSC and TGF and plus BMP4 is best candidate for clinical trials to regenerate the articular cartilage defects in the patients. So our clinical trials were studied by National Scientific Center for Traumatology and Orthopedics during 2 years. So we recruited the patients with osteoarthritis grade 2. We enrolled 80 patients and divided in 2 groups, hydrogel group and PRP group. 40 patients in first group and in second group 40 patients. And we made the functional assay test and MOCART assay by using MRI. So average age of patients was about 45 years old, and patients, male gender 34 and female 66%. So we can see the MRI images on the knee joints of 2 patients with osteoarthritis grade 2. So this white arrow shows the articular cartilage defects. Here we can see the arthroscopic observation of articular cartilage defects. So it was made in hospital by arthroscopic equipment. So we can see case 1 and case 2, the cartilage defects in the knee. So before implantation, our HCF hydrogel with MSC and 2 growth factors, so we make some preparation of these cartilage defects for hydrogel implantation. First, we made debridement of the defective area. Next, microperforation of the defective area. So by using this device, medical device, so we purchased this device from company Baxter from USA, Doppler jet system for minimally invasive hydrogel implantation. So in first syringe, we filled HCF, MSC, and growth factors, and second syringe we filled by apratinine and thrombin. And we use 10 centimeter double limit tip for minimally invasive surgery.
Professor Agay, sorry, time is over. Could you go to question?
Okay. So here we can see the process of implantation, and here we can see the implanted hydrogel in the articular defects. Okay, just a moment. Okay, next we make the clinical radiological methods for evaluating the recovery of damaged knee joint. We use WASH, WOMAC, COS, and MOCART analysis. So we can see the comparative analysis of functional indexes of this of WASH, WOMAC, and COS within 6 months after hydrogel implantation. So as we can see, the better results we observe at 6 months after implantation in a group hydrogel in comparison to PRP group. For example, hydrogel implantation significantly reduces the pain, also symptoms, and so on. And here we can see results obtained from MRI analysis in the hydrogel group and PRP group. And here we observe after 6 months a significant regeneration of articology effects in medial tibial condyle and medial femoral condyle after 6 months. And here I will show the one clinical case, this 12 months after treatment. This patients with left side idiopathic knee osteoarthritis grade 2 before surgery. Here we can see the minimally invasive implantation of hydrogel. And in this slide we can see the result after 12 months after treatment, before and after 1 year. So MACAS showed significant regeneration of articular cartilage in the defect, and WOMAC score showed that before surgery point was 47 points, 6 months 24 points, yet after 1 year 80 points. So I made this short conclusion. So our clinical trials show that HCF hydrogel with MSC and growth factor is safe and effective, significantly reducing pain, improving knee function and quality of the life in osteoarthritis patients, and significantly promoting articular cartilage regeneration. Okay, thank you for your attention.
Thank you very much for your presentation. We have time only for one question. Please.
Thank you. Very promising results. And my question, where are you going to continue your clinical trials, second phase or whatever? When that treatment will be available for public? Yeah.
Of course, we plan to make multi-central clinical trials. So currently we talk with some investors to continue our clinical trials in Kazakhstan. So I also talk with some director of hospitals and medical center in Karaganda, Almaty, Shymkent, in Astana. Totally 5 clinical bases hospitals will be involved in the multi-central clinical trials.
Thank you very much. Very good. Thank you very much again.
And our next speaker is Professor Maaike Alerts, University of Antwerp from Belgium. She will be talking about iPS-derived cardiomyocytes as a tool for precision medicine in inherited cardiac arrhythmia. Thank you.
Okay, so I first like to thank the organizers for inviting me, and especially Ainur for inviting me to be here and celebrate the anniversary with you. I will tell you a bit more on iPSC-derived cardiomyocytes as a tool for precision medicine in cardiac arrhythmia, because I'm from the cardiogenomics research group. So first of all, cardiac arrhythmias are inherited autosomal dominantly mostly, and they affect together about 1 in 1,000 individuals. Patients can be diagnosed by finding specific abnormalities on the ECG. They can have also palpitations and syncopes, but often there's no symptoms until sometimes sudden cardiac death can arise. And about 2% of sudden cardiac deaths are explained by cardiac arrhythmias inherited. More than 50 genes are known related to these disorders, and they are mostly encoding cardiac ion channels and also accessory proteins, sometimes structurally helping to get the channels properly in the cell membrane. Now, still in 70% of the cases, the genetic cause is unknown, meaning that there's still a lot of research to be done. And if we want to study the pathomechanisms at a molecular level, we need a good model. We would like to use tissue of patients, but you cannot just take a heart out of an individual and start studying that. So that's where the very neat model of iPSC-derived cardiomyocytes comes in. And I don't have to explain iPSCs anymore because Professor Singh did that very nicely for me, but you can start with patients, specific patients with specific mutations, and then take some of their somatic cells, add these transcription factors, and reprogram them into these iPSCs, so the stem cells that can divide themselves and then be differentiated into any cell type. And since we're interested in the heart, we will then differentiate these cells into iPSC-derived cardiomyocytes. And then we, in fact, have the heart tissue or the heart cells of the specific person in a dish in the lab that we can study. We also need control individuals because we want to compare the diseased and the healthy situation. And a very nice technique we can use is CRISPR, where we can introduce genetic variants or genetic mutations specifically into control cells, or we can correct mutations to provide like isogenic control lines, where the only difference between the different iPSC lines is the specific mutation. The next, we need to study these cells, so we can look at morphology under a microscope, we can electrically study them, electrophysiology, we can do this molecular phenotyping, looking at multi-omics data, and, and then we can also perform drug testing, gene editing on these cells to then inform personalized therapy. Another interesting area to use iPSC cardiomyocytes is this area of variants of uncertain significance. So by sequencing more patients, we find in the genes variants that we're not sure, is this the variant that's causing the disease, or is it just a variant that's benign and not doing much? So we can then again use the iPSC cardiomyocyte model, especially these isogenically created lines, to check whether the variant on its own is doing something or not. So we check the pathogenicity, and that could also influence then the therapy of the patient. So what we are doing in our lab, so we can reprogram somatic cells to iPSCs ourselves, and then these iPSCs are then differentiated to cardiomyocytes, and we've optimized the protocol where in the first 4 days you use chemical factors like Wnt activators and inhibitors to get to mesodermal stem cells and then cardiac progenitor cells, and then we mature them with the specific protocols of fatty acids and T3 hormone, and we get functional cardiomyocytes that start beating around 8 days after we started the differentiation. And normally this should, so this is a little movie to show that when these cells are beating, you really see it under the microscope, and it's really, nice to see what you've produced. So how we use it in our lab, I will show you 3 examples. So we've used them to model a specific Brugada syndrome mutation. We use them for modeling a VUS in a calcium channel, and we're trying to build a Gevi-Geki model, which I will explain a bit later. So the first way we've used them was trying to model a specific founder mutation, a specifically Belgian founder mutation, that we found in 29 different families. And so the mutation introduces an intronic indel, and which causes like a cryptic splice site to be activated, and part of exon 27 is lost, leading to a 32 amino acid deletion in the protein itself and making it unfunctional. The cardiac sodium channel is responsible for the import of sodium ions in the upstroke of the action potential, and as such influences the cardiac rhythm. And we have studied, like the characterization, phenotypically characterized our cohort of the patients and the families, and all the patients carrying the mutation. There was a very wide expressivity. So individuals being really old and having no symptoms at all, and also sudden cardiac death at young age. So we were wondering whether we could model this variable expressivity in iPSC cardiomyocytes and then find the differences and find why some individuals are severely affected and others are not. So what we did was selecting 5 patients with different different phenotypes, so sudden cardiac death at a young age, at higher age, syncopes, and then some asymptomatic individuals. And we made iPSC cells of all these individuals and then differentiate them. So you can see here that we made nice, proper iPSCs, and then we differentiated them into cardiomyocytes. We also added 2 unrelated controls, and we made one isogenic control from one of the symptomatic patients. We corrected the mutation with CRISPR, and so we have a real isogenic cell line that we also used. So for each individual, we used 2 clones, and we performed at least 2 differentiations to make sure what we see is really what the reality is, let's say. What we found first is that actually, in iPSC cardiomyocytes, you don't have just like a wild type and a mutant transcript, but there are 2 mutant transcripts. So there's the deletion that was predicted, and that was seen in heterologous cell models. But then we also found an insertion of the GTGG, creating another transcript, but also causing a frame shift, and so like a faulty protein. And here you can see in all the patients, we see an expression of these 2 different types of mutant channels and the wild type. In our control individuals, only the wild type channel. Then we try to find differences in this expression, maybe explaining also the difference in the expressivity of the phenotype of the patients, but we didn't find differences between patients and controls, symptomatic, asymptomatic patients. And if you rank the patients by severity, you can see that the deletion allele has a bit of a trend in higher expression in the less severely affected patients. We looked at the protein expression as well, so looking at the channel itself in the membrane, you can see a Western blot there, and where we didn't find like 2 bands, so the mutant channel with the 3rd mutation, GTGG insertions, is not expressed on the membrane, but the 2 others are. And if we looked at then the membrane expression itself, it wasn't different between patients or control, symptomatic, asymptomatic patients, or even if you ranked them by severity. So we didn't get much information from this study. Then we tried to patch clamp the cells, so we used an electrical technique to look at the action potentials and look at the cardiac rhythm of the cells, let's say. Action potential characteristics, so we look at the amplitude of the action potential, the duration of the action potential, and upstroke velocity. And again, we didn't see a difference even between the patients and the controls, so the ones with and without a mutation. Also no differences between symptomatic and asymptomatic patients. And you can see here, if you look at the plots, that there is quite a high variability. So you see like a huge spread in the data, which is caused by the different differentiations and the different clones. So if you rank to the more severe phenotype, you found a lower amplitude, but also a longer action potential duration. So that didn't really fit with what we thought. Only in the the patient BRS12 and its CRISPR control, we could find that the action potential duration was statistically significantly lower than in the corrected control individual. So that's something we would have expected. So high variability is something to remember from this part of our functional testing. Then we also checked the sodium current density. So using the same patch clamp technique, you can look at specific sodium current. And as I said, it's the sodium channel that's affected. So we would really expect to see a difference there between patients and controls. So that's what we want to see. If you look at the plot here, so the lower or the more negative the current, the more current there is. You can see that this is the control number 1, and indeed that he has the most current, but then the second control is buried somewhere between all the patients. So we didn't really find a difference between patients and control, even in the sodium current density, which is the main characteristic we would have expected to see a statistically significant difference. But if you look at the standard deviation, and that's a bit what we've seen also with the action potentials, it's huge. So there's really high differences between differentiations and clones. And that's what I try to show here. So from one individual, you can take like 2 different clones, then you differentiate them several times, and the sodium current density ranges from something that is very typical for a patient to something that's also typical for a control individual. So you really see a high variation there, and that's why we couldn't find a statistical significant difference. So control 2 had a current density very similar to the patients, as you can see there. Then if you remember from my first figure, the patients were chosen like 2 from a same family. So we looked at the current density within the families as well. And then sometimes, like one time, the asymptomatic patient had a higher current density, but in another family, the symptomatic patient had a higher sodium current density, so there's no correlation there. Only if we look between the BRS-12 patient and its specific isogenic control, there we found that the current density of the control was higher than that of the patient itself, as you can see in this image. And so that just shows the advantage of probably making real isogenic controls and comparing those. So what we can conclude from this part of the study is that there's a high variability, so maybe this model and looking at the action potential characteristics is not very useful when you only have small differences and small phenotypic effects. But then what we also did on these cells was doing RNA sequencing, so we hope that we can still use these data to look for altered pathways and genetic modifiers to look for potential therapeutic agents, and that's now in the process. The second study I wanted to tell you about is where we used the iPSC cardiomyocytes for modeling of a variant of uncertain significance in the CACNA1C gene, and the CACNA1C gene encodes the calcium, L-type calcium channel on the membrane, and that's involved in the plateau phase of the action potential, the cardiac action potential, and is also involved in excitation contraction coupling, because then calcium enters the cell and you get contraction of the muscle cells. We did it in vitro in iPSC cardiomyocytes. We also used the zebrafish model to check the effect of this FUS. We did a CRISPR ourselves, so we did a CRISPR knock-in. We designed 2 guide RNAs to guide the Cas9 protein to the specific location of the mutation that we found in a patient, or the VUS that we found in the patient. And indeed, we managed to change the C into a T. And the nice thing of working with iPSC is that you can make like a heterozygous clone here, so it has both a C and a T allele, and also homozygous clones with only the T allele. So in a patient, you would have, it was heterozygous, but then we can model even like what would it do if we have 2 of these mutated alleles. So we did 4 differentiation rounds. So we now have the control line and then the homozygous and the heterozygous line with the mutation. We did 4 differentiation rounds, and you can see we got very nice iPSC cardiomyocytes. And we didn't see a difference in the membrane expression of the ion channel, which is something you would expect because it's only a missense mutation. So the ion channel gets expressed, it's just maybe non-functional. Then we used the calcium dye imaging. So I add a dye to the cells that gets inside the cells, and upon the calcium concentration differences, it fluoresces. And that's what you see in this image. So you can really see a flash of fluorescence with every calcium transient passing by. We do this in 96 well plates, so you can get quite high throughput imaging. And then when you plot the fluorescence in function of the time, you can see like a peak, and then again going down, and a peak and going down to the normal background level. You can use these data to then look at the transient duration. You can analyze the peak-to-peak time interval, and you can look at the amplitude. And so if we did that, we didn't find any differences in any of the calcium transient characteristics between the heterozygous, homozygous, and the control line. We didn't see any peak-to-peak variability, which you would expect in an arrhythmia, so we also found that there's no real effect. Even when adding drugs to the cells, so that's also an advantage of working in a cellular model, you can add drugs to the cells. So we added calcium blockers, verapamil, and nifedipine, and we saw that they react to the drugs, but we didn't see a difference in the reaction between the mutation and the wild type. So we conclude that the fuzz is rather benign. We also saw that in the zebrafish model, and we're performing some extra differentiations to prove this. And then the last thing, I'll keep it short, is how we were trying to build a GEVI and a GECI model. So GEVIs and GECIs, these are genetically encoded calcium indicators and genetically encoded voltage indicators. So these are proteins. If you introduce the gene into your cells, they express these proteins and they will fluoresce upon calcium changes or upon voltage changes. And we chose the two novel ones, so a green fluorescent NCAMP7 and a red fluorescent ARCON1 indicator. So when you do imaging, you can look at the green channel to watch the calcium changes, and you can look at the red channel to watch voltage changes. So you only need confocal microscopy to look at the cells. You don't have to manipulate them anymore after that. So the idea is to get the GEVI-GECIs into our cells. We're using CRISPR, so you can put them specifically in a locus that's known to be a safe locus in the genome. Then we have our GEVI-GECI cells. We can CRISPR them to bring in specific mutations if we want. Then we differentiate them into cardiomyocytes, and the idea is to make also microtissues where we add endothelial and fibroblast cells, and then you have these fluorescent cardiac microtissues which you can screen, use high throughput for drug screens, characterization of the VUSs, or even CRISPR screens on this model. We did some experiments doing some AAV transfection with NCAMP7 and ARCON1, and maybe you can see it here. So you can see the cells fluoresce. So we showed that they work in our iPSC cardiomyocytes. And here you see for NCAMP7 the signal that we get. If we add some drugs, you can see the effect of the drugs on the cells. So it's working, and we can do the fluorescence imaging. Now, the CRISPR is currently ongoing, so we already managed to do the CRISPR experiment. We found the introduction of the GEVI and the GECI in the genome. We're now purifying the cells and validating some clones that will then be used afterwards in our next projects. And then thank you very much for your attention, and this is the group I'm working in. Thank you very much.
Thank you a lot, Professor Alert, and that was a really great presentation. And I just want to make a small announcement. Let's keep all the questions to the lunch, because otherwise we're going to skip the lunch itself. Thank you a lot.
So I would like to announce our next speaker, Professor Eugene Ponomarev from School of Sciences and Humanities from Nazarbayev University of Kazakhstan. His presentation is entitled Sevinase Functionalized Oxidative Drug Nanocarriers Enhance the Treatment of Colorectal Tumors in 3D Culture Model.
The screen is activated, so
okay. All right, thanks. Okay, so hello everybody. I would like first to thank the organizers for inviting me to speak here and to present some of our recent research, which was done here at Nazarbayev University. So I'm going to talk about a specific topic on using Sevinase functionalized oxidative drug-loaded nanocarriers to enhance the treatment of colorectal tumors studied in 3D cell culture model. First, I would like to come up with some brief history why we started this work. We actually started something similar more than 5 years ago in my former group in the United Kingdom, where we were interested to develop nanocarriers that can target biofilms, bacterial biofilms. And the scheme by which these nanocarriers are working is actually very simple. We loaded nanogels with antibiotics. In this case, we used carbopol nanogels. And we coated these nanocarriers with a cationic protease called alkylase, which is actually able to target the extrapolymer substance of the bacterial biofilm. So the bacterial biofilm is very complex origin. It has a lot of glycoproteins, extracellular DNA, lipid membranes, and vice versa. But the protease, actually with the protease functionality of the nanoparticles, enables these particles to digest their way through the biofilm, EPS, and reach the bacteria which are buried inside, delivering antibiotics directly to their surface. So you can see very briefly here assessment of the action of these particles compared to the control, which is on the upper left corner, and the bottom left corner is the bottom right corner is the result of the treatment of the biofilm with this type of particles. You can see also AFM comparison of the biofilm scan and the scan with the treatment with the protease, which completely strips off the biofilm from the substrate. So you can also see the biofilm residue after the drying of the biofilm of the control and what has happened after the treatment, where most of the EPS has been stripped off and the particles have concentrated on the bacteria, in this case the Forius. So compared with the treatment with equivalent amounts of antibiotics and equivalent amount of enzyme, we actually couldn't find any colony-forming units on the bacterial biofilm after treatment with the nanocarrier loaded with the same amount of antibiotic with the alkylase coating. So we asked ourselves recently the question, can we repurpose this technology, which we have developed originally for treating bacterial biofilm, to help us to target solid tumors? So what is the rationale? The rationale is that both the biofilm, the bacterial biofilm, which contains EPS, and the extracellular matrix in solid tumors, which basically impairs the infiltration of the anti-cancer drugs, are made from cross-linked polymers, cross-linked proteins in this particular case, glycoproteins in the case of the bacterial biofilm, and lightly cross-linked fibers of collagen, which are linked with laminin and integrin in between the cancer cells in the solid tumor. So in both cases, we have protein component, which has got cross-linking properties, and if we target the same material with protease functionalized nanocarriers, in this case, when we have solid tumors anti-cancer drugs, we may be able to help to facilitate the penetration of anti-cancer drugs inside the cellular, the extracellular matrix. Our study here was focused on colorectal cancer. So as you know, this is the third most widely spread cancer worldwide, and the second leading cause of death, cancer-related death. So 40% of these cancers have KRAS mutation, which is highly mutagenic and leads to resistance towards anti-cancer drugs. So we have focused our study on two specific cell lines, which are both KRAS mutants for colorectal cancer, HCT116 and SW620. And we have tried to develop a combination of oxidative drugs which acts on this pair of cell lines. And in this case, we focused our attention on the recently developed combination of arsenic trioxide and D form of vitamin C, which was developed here by Professor Sarbasov in his previous studies, which are highly oxidative drugs. Basically, arsenic trioxide is highly toxic, but in micromolar amounts actually dials up production of reactive oxygen species inside the cancer cells. In high concentration, of course, it has toxicity. We can't use it at very high concentration. But when it is combined with vitamin C, it has a synergistic effect, as the vitamin C in micromolar amounts is antioxidant, but in millimolar amounts has pro-oxidative action, which brings the cancer cells into overdrive and production of hydrogen peroxide in their mitochondria, which leads to the apoptosis and the cell death. So this combination actually we explored, loaded in the case of nanocarriers, which are functionalized with another protease, which I'm going to show you in a minute. But the idea is why we actually use the protease in the nanocarriers is that when you normally test anti-cancer drugs, people do them in 2D models where the cells are on the surface of a plastic and the cells are completely accessible by the drug solution, while in the solid tumors you have cells which are in the interior of the tumor, which have different microenvironment than the cells that are in periphery and are exposed to the drugs. So therefore, we expect that the drug combination will be effective on the peripheral cells in 3D cell models, but the core will remain unaltered. And our nanocarriers with the savinase or protease functionality is going to be able to loosen up the ECM and to allow the drug to penetrate fully and to target the core of the clusteroids. So our enzyme of interest, which we used in this case, is another protease called savinase 16L, which is alkaline enzyme, has very high isoelectric point at normal physiological conditions, have cationic charge, very highly positively charged. So it's optimized to be used at elevated pH, but it works very well at physiological pH. And the model that we used is 3D cell culture, which we have developed in our laboratory based on aggregation and clusterization of cells using aqueous two-phase system, where we emulsify the cells in the emulsion prepared from dextran and polyethylene glycol solutions. The cells concentrate in the dextran droplets, and then by adding more polyethylene glycol to the solution, they are osmotically compressed, which compacts the clusters and forms spheroids in a very rapid fashion. So we break the emulsion after that and isolate the spheroids and culture them in culture media for 24 hours. And you can see here how these spheroids grow over the course of 6 days for both cell lines, how the size of the spheroids consistently increases with time. So after 24 hours, we apply the treatment with the oxidative drug combination or with a free arsenic trioxide or free vitamin C and the nanocarrier-packed oxidative drugs. And we assess the results after that compared with the 2D cell model to see what is the effectiveness of the drug. So this scheme here shows very briefly how we pack the arsenic trioxide and the vitamin C inside the nanocarriers. We mix everything together in one pot with soluble form of shellac together with surface active polymer and drop the pH from 8 to 5. Shellac precipitates, forms nanoparticles. And then in a second step, we add the cationic protease, which forms the coating. So you can see here that the nanocarrier is positively charged once it is coated with the savinase, while it is negatively charged if it is uncoated. And addition of savinase, we optimized a little bit the preparation to make sure that the nanocarrier has positive surface charge, which allows to accumulate on the surface of negatively charged cancer cells. You can see here the release curve of these nanocarriers, which means about 12 hours it loses about 80% of the payload of arsenic trioxide, and approximately on the same time scale for the vitamin C. We experimented with cells in 2D cell culture and in 3D cell culture after incubation with these nanocarriers. And you can see here the comparison with the control, which is untreated on the upper left corner. The morphology of the cells which are treated with these nanocarriers is showing the particles accumulating in large concentrations on the surface of the cells. In other words, they deliver extremely high local concentration of A2 and DVC, which amplifies tremendously the action of the drug. We also tested the degree of penetration of these particles inside the clusteroids, and you can see after 24 hours of incubation, the nanocarriers, which in this case have been marked with a fluorescent tracer in red, accumulate on the surface of the cells which are inside and cause also loosening of the spheroid to improve the action of the drug. So here you can see the action of the free A2 and free DVC on the 2D cell culture on the left-hand side panel. And on the right-hand side panel, you can see the same action of the combination of these drugs on 3D cell culture. You can see that although the 2D cell culture responds, and basically most of the cells die as a result of this treatment, the 3D cell culture is unaffected, simply because most of the action is on the peripheral cells, while the cells inside the core of the clusteroids are unaffected. You can see here, in terms of cell proliferation and in terms of cell viability, the action of the drug combination of A2 and DVC has the most profound effect than compared with the individual drugs. So also the effect of the drug combination on the ROS production in the mitochondria is most pronounced in the case of combination of A2 and DVC on 2D cell culture. But there is no proportional effect in the 3D cell culture. So you can see here the comparison between 2D and 3D cell culture. In the case of a percentage of dysfunctional mitochondria, the effect is most pronounced in 2D cell culture compared with 3D cell culture. In other words, 3D cell culture and solid tumors don't respond in the same way as people test them in 2D cell culture. So there is a protective effect for the cells which are incorporated in the core. Similar is the case with the other cell line. This was first with the HCT116. This is with SW620. We have absolutely the same effect. It's even actually more sensitive towards the A2-DVC combination. So in fact, the two cell lines behave in a very similar fashion. So we decided to see what will happen if we pack the drugs in the savinase-coated nanocarriers. And you can see here on the top panel a consequence of the exposure of the 3D cell culture to the different combinations, the control, the non-coated A2-loaded particles and DVC-loaded particles, the non-loaded particles. And here we have the combination of savinase-coated DVC-loaded nanoparticles, savinase-coated A2-loaded nanoparticles, and here a combination of both. You can see over the course of 24 hours, of course of 48 hours, the clusteroids almost completely disintegrate, and the action of the nanocarriers is dramatic on the 3D cell culture. You can see here, for example, for the combination of the A2 and DVC, the proliferation is the lowest. For 3D cell culture, the cell viability is the lowest, and also the apoptosis is the highest. So that's for the HCT116, and very similar picture is the case with SW620. In other words, it works universally across two different colorectal cancer cell lines. In terms of mitochondrial response, ROS production in the both cell cultures, you can see the effect of the ROS production in the combination of savinase-coated A2 nanoparticles and savinase-coated DVC-loaded nanoparticles for both cell lines is the highest, and the dysfunctional percentage of functional mitochondria is the lowest in this combination. In other words, the combined nanocarrier amplifies the effect of the oxidative drug combination. So that's what I wanted to tell you. A short story to wrap it up. We surface functionalized A2 and DVC-loaded shellac nanoparticles with a cationic protease, savinesins. The protease coating of the nanocarriers promotes active targeting of solid tumors by electrostatic accumulation, attraction to the negatively charged cancer cells. accumulates on the surface and amplifies the action of the drugs, but also acts on the extracellular matrix and helps to loosen up the clusteroid or the solid tumor and enhance the penetration of the drug inside the core. So with this, I would like to thank the main actors here who did this study. This is Anel Myun, who was a master's student when she did this, and Nuria Nurlankizi, who was in her 4th year BSc biology. Also Sauri Kalmagambetova, who was working as a research assistant in my lab. Currently, she is doing a PhD in the biology department. Also, collaboration with Dr. Burska and Professor Dr. Sarbasov is appreciated related to this study. Also, I would like to acknowledge the original contributors to the biofilm story from my group in the UK, Dr. Wang, Dr. Weldrick, and also Saba Alobaidi. With this, I would like to thank you for your attention and to take any questions that you might have.
Thank you very much, Professor Parunov. Unfortunately, we don't have time for questions. I'm very sorry. Thank you very much again. So our last speaker of this session is Professor Gorka Orif. He's a professor at Pharmacy and Pharmaceutical Technology Institution of the University of Basque Country from Spain. And his presentation is entitled A Scientific Journey from Regenerative Medicine to Diagnostics and Health Sustainability. Please, the floor is yours.
Thank you. Good morning, and thank you so much for the invitation, for the kind invitation, and of course also to Gonzalo Hortelano for hosting me here. Well, it's a pleasure being here. I'm going to try to be short because we are a little bit late on time, and I will try to share with you some of my scientific research fields just very shortly. I come from Spain, specifically, oops, I think it's not working. Let me see. Okay, I'm coming from Vitoria. Vitoria is just located in the north of Spain, in the Basque Country. My research group is Basque Sustainable Pharmacy and Biotherapy, so today I will give you some few ideas of the work we are doing in biomedicine, in the development of new drugs and medicines, but also in the concept of health and sustainability. And of course, if you want, you can also contact me through the social media. Well, these are the 3 short topics that I want to share with you in which we are actually working. The first one is related to regenerative medicine and new the discovery and development of new type of medicines. The second one is more related to diagnosis, so I will try to speak very shortly about Alzheimer's. And the last one is a new research field that I have been talking with Gonzalo these 2 days about sustainability and how we have discovered that we have dozens, I will say hundreds of drugs as microcontaminants in the environment. Well, let's talk about regenerative medicine. The first topic in which I started working was the design and development of these cell-laden hydrogels, what we say usually as microencapsulated cells. These type of medicines are suitable for a very long drug delivery, so they could act as living drug delivery systems, so you could use them to release any kind of protein or growth factor, or even you could use it as a model, for example, as an spheroid to test different kind of drugs in cancer cells. My research, especially during 10, 15 years, was more related to the first topic in which, we have tried many different models. You can see here, for example, the study we make with David Mooney in Harvard, in which just by only one shot, we were able to release a biologically active product from these encapsulated cells for almost 1 year. We have made also very small capsules, even for the eye, for just drug delivery, even in loading these capsules with different kind of nanoparticles. We are able to track them in vivo. And we have designed also different kind of systems to control externally the behavior of these cell-loaded systems. Even there were some attempts also for treating some of these basic Alzheimer and Parkinson diseases by releasing BGF continuously and placing the capsules close to the brain. In the last few years, we have moved from these tiny microcapsules to bigger macro devices. This is a work done in collaboration with Professor Guertin and also Bob Langer from MIT. And as you can see, we decided to create this kind of macrocapsules. They are full of cells. We can encapsulate here cells for example, releasing insulin and treating patients without the use of immunosuppressant drugs. This is the main idea of these devices. The cells are protected by this kind of membranes, and they are alive and releasing the products that you designed for. Also, in the last few years, we have made several attempts in tissue engineering and regenerative medicine by developing different kind of biomaterials and hydrogel combinations, many of which have been used for orthopedics. I'm working in bone, in tendon regeneration. This is the paper that we made with a group of Portugal, of Rui Reis. And we were designing this kind of biphasic hydrogel that you can see here, which is a combination of gelatin and hydroxyapatite and cellulose. And we are tailoring, as you can see, this kind of dual system, just mimicking the bone and also the tendon connections. And of course, with the collaboration of DTU scientists in Denmark, we are developing different kind of systems, usually hydrogel combinations, creating self-healing hydrogels, adding, for example, nanoclays, this kind of nanoparticles that give you strength. These kind of systems are very well suited, for example, for bone regeneration, right? As my title of the lecture said, some of these discoveries that were later patented have also made possible to found different kind of companies. This one is Cybosense. It's a startup that is working with smart materials, usually electronic materials, and we are using it for different purposes, of course, especially for health issues. And I was happy that previous speaker was talking also about PRPs, because we have been working with human autologous growth factors. Let me just give you one short definition. This is the process by which a patient, we take blood from the patient, we spin it, we just use the yellow fractions. This is plasma enriched in platelets, and then we can transform these yellow fractions into different kind of formulations that we use in the same patients for tissue regeneration. I mean, just from your blood, we are able to create this kind of different formulations. As you can see, we have liquids that are three-dimensional matrices, membranes, hydrops. Of course, this is absolutely human-based, and it's autologous, and then we use it in the same patient. For example, every time we are placing dental implants, we coat them with this kind of autologous growth factors in order to improve implant installation on patient's bone, right? For example, we have made several clinical trials in patients just for treating vascular ulcers. As you can see, this is a lady that had this ulcer in her right knee, and we use this kind of yellow stuff, right, especially the cloth, and we injected growth factors around, and just 7 treatments later, you can see the last photograph, the ulcer was absolutely closed. The same, for example, in orthopedics for treating this kind of injuries. You have here tendon tears, and as you can see in blue, that was the usual treatment for this kind of disorder. and in gray, and in blue is when we use the autologous growth factors. As you can see, we were able to reduce in time, in weeks, the weeks needed to start training, raining, and so on, right? Running and so on. So this kind of approaches really became very famous. Now I don't think there is in the world any kind of football, basketball, sport club that they don't use this kind of autologous growth factors, right? And of course, here in Spain, we have been able to treat different sport leads. Probably you may know some of them, not maybe the younger people, but the ones of my age. And you have here Rafa Nadal. Of course, this is me some years before. Interestingly, we are also developing the same technology, for example, for treating eye problems. We are fabricating this kind of autologous fibrin membrane and even an autologous eye drop. Okay, in the case of Alzheimer, just a couple of minutes to talk about one of the startups that we founded 10 years ago is Geroa Diagnostics. And the aim of this company, the reason to found it, was that we discovered in humans' saliva one protein that seems to be helpful for diagnosing patients with Alzheimer's. As you know, the diagnosis of this kind of patients is not easy. Usually you need around 20 months to have a proper diagnosis of the patient. There are like different biomarkers, usually taken from a lumbar puncture or just using the PET, but of course they are invasive, they are expensive. So we discovered that if you use lactoferrin from the saliva, you can have like a good correlation between the risk of having or not the disease. I'm just going to give you several numbers. You have here the controls, and you have the 80 patients. And just take into account, this is the concentration, micrograms per milliliter of lactoferrin in your own saliva. And you can see that when the levels are lower, significantly lower, there is a higher risk of having this disorder. This is in 80 patients, and this is also in MCA1s, the mild cognitive impairment ones. So just when the disease is still earlier. And as you can see, we also observe the same difference. So we have developed one kit by which you take saliva from the patient, right? And you can just measure this protein to see which is really the risk. Because as you know, sometimes the difficulty is just not only diagnosing the Alzheimer's, it's also to distinguish sometimes the Alzheimer's versus all the other dementias. Here we have a beautiful paper we made with one of our best scientists in Spain, Valentin Fuster. And you can see in green, this is the the protein saliva from healthy patients. And in blue and red, you can see what's going on with the lactoferrin when you have the disease. As you can see, levels are significantly lower. And if you allow me 2 additional minutes, I will talk about another different thing. I think this is quite fancy. It was for me. You know, I'm full professor in pharmacy. And I've been like 20 years teaching my students, you know, and I used to talk them about the life cycle of a medicine, right? Okay, you take it, it goes to your blood, then goes to the tissues, and then you release it, right? And my story ended there. But suddenly I discovered that that story was not ended, really, because when that drug is released from your body and goes to the wastewater, unfortunately, the wastewater treatment plants are not designed to clean those drugs, so those drugs in different proportions and concentrations goes to the rivers. From the rivers goes to the seas. And of course, it's been taken by the fishes, by the biota, and it's related to some very, I would say, problematic effects in the environment, and as I will show you, right? So this is a paper in Science we published some years ago showing for the first time which was the whole cycle, really, of the drugs, right? By the way, there are around 1,000 drugs, human drugs, that have been already measured in the environment, right? So we published several papers related to this topic, and we started doing something that it was really different from me. I always been in the lab, or just with patients, and suddenly I was in the rivers taking some samples and measuring microcontaminants. I know for sure you know many of the microcontaminants that are currently under research. I will say, for example, micro and nanoplastics. Do you, right? Have you ever heard about PFAS also, of course? But maybe not so much about drugs, about medicines, right? So we were taking, and we are doing that in Spain and in different places, taking samples. We can analyze by wastewater epidemiology viruses. We can also analyze drugs, and we can analyze also, of course, pharmaceuticals. By the way, our research group was the first one in the world to detect and observe that even in dolphins, there were human drugs. There are human drugs in their tissues, right? Indeed, in this paper, we detected 3 different human drugs. So as pharmacists, we are working hard not only to design the drugs of the future that have to be safe, have to be efficient, of course, but we are also trying that they have to be also sustainable, right? And that means that we need to make an effort in maybe designing greener drugs, and of course, an effort in cleaning, better our rivers, just because this human health cannot be only understood as a human problem, but this is a one health problem, and everything is just related. So I'm finishing. Just let me only show you people from my group, right? Some in the labs and some others, as you see, outside the lab, which is quite fancy, especially because your day-to-days change a little bit. Also people in the company that we are working with human growth factors, and also in Alzheimer's, where we are introducing this product in different countries so far, and it's been a very exciting experience. Thank you so much.
So, unfortunately, thank you. No time for questions, and so you are invited for lunch now. It's time for lunch. We are late, but still we have some time.
Yes, lunch for speakers is at Fuzzy Cafe, and someone from our organizing committee members is waiting for you over there. Okay, and the next session will start at 14:40, but please arrive earlier so we would start on time. The session on neurodegeneration research will start at 14:40. Thank you.
Нам надо селфи сделать. Подождите.
Раз. Раз.
Okay, all right. Yeah, it does. Okay.
Just a little.
dear colleagues, I think we'll start soon. So I just let you know in advance that several speeches will be in Russian. So those of you who need translation, please take it in advance, like microphones and and headphones. Yes, спасибо, забыла. Сейчас подойдет. Куаныш, оператор, дайте этот.
Would someone in this session present in Russian?
Yes, because we have medical doctors actually.
Well, I need one as
They
well.
don't speak English, and I will let them present in Russian.
Yeah.
Because we want to see real doctors who work with
Okay. Okay.
Yeah. Then there are the channels. There are two languages. So you will kind of, yeah. And then here the volume.
Spanish? This one as well.
This one doesn't work.
Okay. I wasn't prepared for
Yesterday.
No, yesterday I didn't. There was one or two presentations in Russian, and I just skipped that one.
Check it. That's very strange.
All empty.
Actually, shouldn't be. Maybe the system doesn't work. Let me check.
So, dear colleagues, we are starting our session right now. Please proceed to your seats. And just a small announcement before we start. Since we're a little bit late on time, I hope we'll finish by 6:15. And the buses for speakers will be waiting on the main hall entrance at 6:15. Yeah. So, once again, it's my great pleasure to welcome in Astana our guests who came aboard. And I'm very glad to see, to welcome people who just came to listen our bright speakers. And we're starting our last session for today, but actually it's the longest one. Neurogenerative research. And it is my great pleasure to introduce our keynote speaker, our first keynote speaker, Professor Daniel Anthony from the Oxford University. Daniel, please.
Well, good afternoon, everybody. I hope I'm not feeling too tired after lunch. In a way, of course, lunch, the reason I mention the lunch, of course, is because what I'm going to talk about, I'm going to talk about how your blood chemistry affects the way that you think and feel. If you had a sort of a very good sugary lunch, you might be feeling a bit excited by the prospect of listening to all the talks this afternoon. If you, on the other hand, skip lunch or you decide to have something that's a little bit sort of less exciting, it's not switched on, is it? That's right. That's quite helpful. There we go. It doesn't look like I had enough because I'm not obviously functioning on all cylinders. So, what I'm going to do is I'm going to take you through, basically I'm going to present mainly unpublished and new data. So I don't think there's one slide that has some old stuff on it. But otherwise, it's all brand new. And it's about presenting to you a hypothesis that we've developed very recently in the lab that we think we've come to that point through a number of recent observations. And I'll just explain very quickly. When you have a cold or you have any kind of an illness, you feel rubbish. And you go to your doctor and you say, you know, I've got a cold, but I feel bad. I feel slightly depressed, actually. And some of the signs, some of the things you'll be feeling look awfully like some of the manifestations of major depressive disorder, for example. And we call this sickness behavior. And the doctor will say to you, well, you feel bad because you're sick, but that's not very helpful to you. But many of us have been thinking about this for quite a long number of years. People like Robert Dancer. Actually, he was pretty much one of the, and Adrian Dunn coined this idea of sickness behavior. And it's a set of real stereotypical behaviors that accompany your sickness. And from there, you get a peripheral infection, you mount an inflammatory response, you make peripheral cytokines, you make central cytokines. Those central cytokines reprioritize your behaviors. So it's not a question of can't do it, it's won't do. And that's what happens to you. You change your sleep patterns. You don't want to see people. You feel low. You lose, you become anhedonic. Many of the things, oddly, that are exhibited by humans, but also by mice. And a lot of the stuff I'm going to talk to you today is about things we've done in mice. So fundamentally, you can, if for example, you take a dam with her pups and you give her an injection of a bacterial product, LPS, for example, and then you bust up her nest that contains all her pups. She just can't be bothered. She'll go and get the pups back, but she won't rebuild the nest. But if you reduce the temperature in the cage, then what will happen is she will actually still go and rebuild the nest and get her pups. So she can do it, but doesn't want to do because she feels rubbish. So that's sickness behavior. For the last 30 years, people have been obsessing about cytokines basically changing the way that neurons behave. I think there is undoubtedly some element of truth in that, and neurons are bristling with receptors to cytokines and chemokines, but oddly, we think it actually could be a little bit more simple than that, and I'm going to tell you about what we call, what we decided to call, the lactate steal hypothesis. And this is going to be about how an astrocyte in your brain basically becomes activated by cytokines or events in the periphery that it's sensing. It then reprioritizes and says, I want to do my own thing. Instead of worrying about giving neurons lactate, it starts taking the lactate and effectively using it up and generating and using that substrate in its own mitochondria. And I'm going to show you data for how that goes. But let's start off really by having a quick wander through some of these observations, just to sort of highlight the ways in which the periphery can affect the way that you behave, and then I'll show you. So I'm going to start with, because it's a neurodegenerative disease topic, I'm going to start by basically something that you're all thinking about right now while you're listening to me. You're thinking, is my brain shrinking? Is it shrinking right now at this moment in time? Is it getting really, really small quite quickly? And for some of you, your brains will be shrinking. They'll be shrinking slightly faster than other people in this room. Now, obviously, if you're young, your brains aren't shrinking very quickly. But if you are likely to develop dementia, then your brain actually, sadly, is losing quite a lot of volume. We heard earlier about how you can diagnose the presence of Alzheimer's disease or dementia by a lactoferrin test. That's a really interesting thing. First experience that I have that one. But we weren't so interested in knowing whether people have dementia. When you go along to a sort of dementia clinic or a memory clinic, you kind of know that there's something wrong. What you want to know is you want to know if you've got vascular dementia or whether you've got Alzheimer's, and that was actually addressed by the lactoferrin. But you also want to know how fast your brain is shrinking. And one of the things that Teresa Kacharova has done with me is she took a whole load of samples from individuals with, you're not going to be able to see this, are you? She took a whole load of samples from a trial. The VitaCog trial was looking at vitamins, but we took the control elements of that, and we looked at the levels of GFAP, neurofilaments, and p-tau in particular, and we were able to show that we could very poor, our ability to say that somebody actually was going to have their brain shrink in a couple of years' time wasn't, these markers just weren't very good. And so we also took our blood biochemistry. So we're thinking that these molecules, neurofilaments, p-tau, for example, they're GFAP, they're coming from the brain into the periphery. Then we took peripheral metabolites and we did some metabolomics, and we mixed those metabolomics with these neurofilaments, GFAP, p-tau, etc. And we were able to develop models and predictive models to say that your brain is going to shrink over a 2-year period. So right now, if I took your blood, I could tell you whether your brain is shrinking. But what this also highlights is it highlights the fact that there are peripheral changes. The brain is basically informing the rest of the body, changing its metabolism. And one of the things that neuroscientists never used to like to admit is that anything existed below the neck. And I think we've seen today and yesterday that actually there's an awful lot going on in the rest of the body, that the brain is, and that there's this fundamental crosstalk that we're getting more and more sort of excited about that, because obviously if things are happening in the periphery, we might be able to do things about them. Another student, Carolina Costa, who's coming back as a postdoc, she was working on psychopathy, giving people questionnaires, trying to decide whether they had psychopathic tendencies. Having looked at some of the questionnaires and filled them in myself, and also looked at some of the responses, these are people that you would not necessarily want to stay in the same house with, in my view. You would lock your bedroom door. But they are out there in the community, and what we were doing is we were taking blood, we were taking fecal material from them, because Carolina's a microbiologist, and we've heard a lot about the microbiome. And oddly, there was very little in way of a signature of basically psychopathic behavior in the community in this cohort. Whoops. But what she did find was that actually the fecal metabolome had changed in these individuals. And she was also able to sort of stratify them into these sort of individuals who with psychopathy and anxiety, and sort of found that there were very distinct signatures, and those distinct signatures had distinct microbiomes, and those distinct microbiomes, whether or not they chew up, whether they generate histamine or whether they generate branch-chain amino acids, all slightly different in those cohorts. So that was kind of interesting. So again, that's a behavior that seemingly is ultimately tied to what's going on in your gut. So many years ago, and this is principally what I'm going to talk about, I started off with sickness behavior. This is this concept that if you get an infection in the periphery, so that was no infection, that was Alzheimer's, that was psychopathy. Now we're going to move into the realm of sort of disease in the periphery. So you have an infection in the lung, you generate an acute phase response, and peripheral cytokines. These mobilize leukocytes that are able to fight that peripheral infection. If you have CNS pathology, they also affect the way that CNS pathology behaves. And as I mentioned, it switches on, I'm now understanding why everybody was having trouble with this pointer. Where are we going? Because it's not a pointer. Here we go. There we go. Whoops. Okay, now I'll try not to click on this without thinking very carefully about what I'm doing. So this is effectively, we switch on this cytokine production, de novo synthesis of cytokines in the brain as a consequence of having a peripheral infection. And many diseases in the periphery will do that sort of thing. And they will change the way that you behave. And we've already discussed the sickness behaviors. This is an interesting one. This is osteoarthritis. And of course, if you have osteoarthritis, which is basically non-immune, or felt to be non-immune, then what we can discover is that we can build models to distinguish people with osteoarthritis versus controls using metabolomics very simply. And you would expect us to be able to do that. But what's interesting is we can also do it with CSF. So having that peripheral joint disease, having pain for an extended period, basically changes the metabolome in your central nervous system, in your CSF. And that was an interesting phenomenon. One of the, if we look at this sort of correlation plot, we can see that some of the interesting ones that pop up that correlate with, so for example, serum histidine here is an interesting one, just to highlight one particular phenomenon. We find that it correlates with many of the other serum amino acids and with pain scores inversely. As the histamine goes down, we find the histidine goes down, you find that your pain goes up. Let's just have a look at the next slide. This is a set of individuals who we followed for a couple of years after they experienced long COVID. So they came to us with long COVID. Some of them got better, and some of them didn't. Those that improved, basically their metabolome looks now just like the metabolome of a healthy individual, somebody who had just recovered from COVID without actually having experienced long COVID. But in those individuals that didn't recover, oddly, their blood metabolome now looks very much like somebody with fibromyalgia. And you'll notice, interestingly, that we've got the usual suspects pop up again. So we've got basically histidine is down again, and glutamine is down. And these are the molecules generating neurotransmitters, for instance. We've got our branch chain amino acids, which are altered in those people that did not recover from long COVID. And we've got a reduction in acetate. So this looks like, effectively, in these individuals, they're suffering from sort of a paucity of metabolism. Basically, it's energy failure in these individuals. Now, is it energy failure because they've adopted a different lifestyle? And that's really quite possible. So cause and effect is an issue here. But certainly, there is a distinct signature for these individuals. Another illustration of this sort of idea of the crosstalk between the brain and the CNS is, for example, when we take exogenously administered, whoops, I've done it again. Let's go back. We take exogenously administered extracellular vesicles. These are the system, when you injure the brain, you release these vesicles that carry the signal to the liver and switch on the acute phase response. That's how the brain lets another organ know that it's been injured. Interestingly, it's also how a kidney would let the liver know that it had been injured. It's how the gut would know. It's how basically these structures, these extracellular vesicles that bleb from cells, carry information, injury signals to the rest of the body. And we see that if we give a TBI to an animal, and then we inject it with some saline, we get a blood-brain barrier breakdown, we get a whole series, we get recruitment of neutrophils, for example, and we see that if we then just take extracellular vesicles that come from, say, macrophages or activated endothelial cells, or we just take plasma extracellular vesicles from another animal that's been injured, we can then augment the TBI. Again, illustrating how crosstalk, the existence of a CNS disease, in this case a traumatic brain injury, plus further disease in the periphery, and you get this sort of synergism and an increase in the extent of the pathology. So again, highlighting that importance. So let's go back to the concept of sickness behavior and the ideas that I wanted to introduce you to today. So I've mentioned the cytokine theory of depression, and I'm telling you that I think that we haven't got, the reason we don't think it's right is because if we inject a TLR7 agonist, it's basically R848, and we inject it into an animal, the animal exhibits sickness behaviors, but it doesn't actually express interleukin-1 beta in the brain in the way that we expect it to. So it's independent. So if I inject endotoxin, you get lots of IL-1 in the brain. If you inject 848, you don't. So we're kind of interested in this as a model. And we have seen that when we actually inject the 848, or indeed LPS, we see that we see these changes in energy metabolism, and we see that brain aniline, lactate, glutamate, acetate, liver lactate, these are all changed by mimicking an infection. And oddly, if we give a probiotic to these animals, it recovers them. So that was kind of interesting, and we wanted to sort of try and work out how that works. So what we've decided to do is we wanted to, well, we're not fans. I have to say, I'm not a fan of changing my microbiome by basically taking somebody else's feces or whatever Americans like to do these days. But this is a thing, but I think it's a bit crass. And obviously one of the problems with trying to chase individual bugs in your gut is that in actual fact, it's really convergent in terms of what you really want to know is a particular constellation of bugs in your gut give rise to metabolic changes in your body. And the way that you get to that can be via a lot of different combinations of bugs. So actually just thinking about what is the downstream product of all those changes in your microbiome is actually more important. But we did have a look to see what probiotics and indeed other things do when we try and manipulate the gut. So But our idea here was one of the things that we knew happened with the probiotic is that we increased peripheral lactate. And so we set up an experiment where we actually just fed lactate to animals at relatively high concentrations for an extended period of time, for 27 days, and then we did some behavioral testing. We also gave some of them probiotics, and then we chased them with a labeled acetate. And let's just have a quick look at the outcomes of this. First of all, the treatments increased lactate, so, and that happened both in the animals that just were fed lactate. It's quite important that if you give lactate, you actually do change the levels of lactate in the blood, and we can say that we go from the placebo, and then there's the Biocult, the probiotic that we gave, and this gave rise to those peripheral changes. And then interestingly, what we were able to show is that these treatments basically reverse the effect. So we have this 848 challenge, and we found that in the case of giving lactate, we were effectively normalizing all the behaviors that we were seeing in these animals. So effectively, we also saw some rather interesting things. This is what happens when you inject the R848, this TLR7 agonist. We get a massive increase in this, an acute phase protein SAA2, massively upregulated. It's not really changed by the probiotic or lactate, but the behaviors get better. So that's already a sort of an interesting dissociation. If we look at IL-6, for example, we see that we have an increase for the 848, and this is normalized by the two treatments, which again is rather interesting how it has that pro-inflammatory, anti-inflammatory effect. In terms of what was going on in the brain, we discovered that in the 848 and in the lactate-treated animals, we reduced GFAP, we increased BDNF, and these changes also had rather profound effect on these molecules that are responsible for sort of neurogenesis and rather positive things that are happening in the brain. So, effectively, and to sort of sum it up, we found that when we started doing the metabolomics, we discovered that actually giving lactate or the probiotic effectively improved the supply of glutamine to glutamate and improved neuronal function. And this is where the other markers we were looking at sort of indicated to us that in actual fact what was happening was that there was a steal effect. So the lactate, when we gave this R848, when we induced the sickness behavior, when we made the animals feel really bad, we were able to overcome that by giving lactate. So the astrocyte now was shuttling the lactate from blood through the astrocyte into the neuron instead of stealing it and turning it into other things that the astrocyte needs in order to combat or to respond to that inflammatory challenge. So oral lactate enhances astrocytic metabolic function, and it's compromised by 848. When we have a look at these correlation plots, one of the really interesting things that happens is when we look at this acute phase response marker, SAA2 that I mentioned earlier, which is very much correlated to altered behaviors and to many of the changes in, for example, in GFAP, for example, as soon as we give the lactate or the probiotic, those correlations all go away. So we still get the acute phase response, but we break those correlations with these other behaviors and these other other molecules that are expressed as downstream of the 848. So inflammation in the brain, done it again. Inflammation in the brain drives astrocytic structural metabolic change. I give 848, astrocytes become activated. The activation increases mitochondrial demand and diverts lactate away from neurons. You feel miserable. Neurons lose their energy partner. The steal leads to impaired neuronal function and sickness behavior, and then we give lactate, and then we can overcome that. So this is just basically treating depression with a metabolite, which is rather surprising that you can actually do that, and people, I promise you, have tried for many years to achieve similar things with all sorts of interesting immunomodulation that hasn't worked. It's the biochemistry, I think, that's important. So we didn't set out initially to prove this. We were led in this direction. Tolkien said, there's nothing like looking. If you want to find something, you certainly usually find something if you look, but it's not always quite the something you're after. So I finished there, and that's a list of a number of my collaborators that were engaged in this project and others, and that's a reminder to phone home at some time. Thank you very much. I'll end there and take a question if you can.
Thank you very much for that interesting presentation. Dear colleagues, we have time for questions and for discussions. Please answer questions and let's discuss this topic. So, well, I think I would like to ask a question. First of all, recently we kind of coming to the same idea that about peripheral metabolism, brain functions, Alzheimer's, and et cetera, And what I was, I forgot what I was supposed to ask. Yes, about liver function. What we know, we did the some population study, based study, and we found that in people with Alzheimer's, liver function basically affected. So what do you think, what is like egg, what is a chicken? Is it brain damage because of the liver and or vice versa? Thank you.
Yeah, I think provocatively, I would argue that you can certainly trigger, as we see, CNS pathology and changes in the, as a consequence when there wasn't anything happening there before. In sickness behavior, you do get proliferation of microglial cells. You get activation of astrocytes. It's rather strange. I think on the whole what happens is that CNS pathology is, we often think about a continuum, that actually Alzheimer's disease, Parkinson's disease, effectively what's happening is your neurons are slowly degenerating. And everybody says, okay, it's going to, you know, as I mentioned, your brains are shrinking. Some people's brains are shrinking faster than others. But the question is, do they shrink in a continuum or do they shrink in a step function? And in actual fact, I think what I've tried to show you today is that all those interactions that happen, events that are happening in the periphery are going to be changing. Sometimes you're going to have pathology on board, chest infections, UTIs, you're going to be stressed, the sort of things that are actually then going to cause the steps on your neurodegenerative disease. So it's that interaction that I think is going to be interesting and what we need to think about in the future. You might not be able to do anything about that slow rate of decline, but you might be able to do something about the steps. You might be able to make the steps smaller by intervening when you know that there's peripheral disease on board.
Thank you very much. Yeah, please.
Okay, so actually a friend of mine has a question, but I will ask for him. So we have, for example, in intense physical activities, we have lactate in huge amounts from the muscles. But you said about PEROS, correct? So does it need to go through the guts, through microbiome, and then reach the...
Right, so that's the really hard question. So we've given labeled bugs, and we've tried to find out whether metabolites from those labeled bugs make their way into the brain. We know that that acetate that we gave to the animals, we found metabolites very quickly in the brain after giving oral acetate. We found elements of the bugs that we had injected in other organs, but in actual fact, I think the jury is still out. Is it that the metabolites, short-chain fatty acids, for example, are they definitely derived from the bug, or are they within, is it happening within the gut, or is it, I think the problem is that these questions are, oddly, so much work is done looking at the microbiome, but the mechanisms by which and the way in which the metabolites make their way potentially to the brain is much less poorly studied. You know, and it'd be crazy not to mention that, you know, it's possible that the vagus nerve and the nodose ganglia, that this is actually, you know, there are direct connections between the gut and the brain as well that can also be influencing behaviors as you know, and it may not just be about circulating metabolites. But certainly in our hands, we see these profound changes.
Okay, thank you very much.
And you mentioned exercise and stuff. And the other interesting thing about the lactate is you can generate those levels of lactate just by doing some strenuous exercise. Yeah. So that's why people do it, I think.
Hi, thanks for the talk. It was fantastic. We all know that any peripheral disease is going to impact, you know, triggers the inflammation process, and it's going to impact lots of systems. And one of them is a brain-blood barrier. So my question is, did you investigate that? And can you speculate if that impacts on a triple B, how it's going to, you know, impact the role of it?
Yeah, I think that's I've spent many years working on the blood-brain barrier, and I think that there is this, I keep on reading, and I think it's mainly ChatGPT generated, that as you get older, as in the Alzheimer's disease, there's significant blood-brain barrier breakdown. The truth of the matter is there really isn't. These sort of phenomena are occurring in terms of sort of supplying energy demands. extracellular vesicles are interesting because they literally can cross the blood-brain barrier as though it wasn't there. It's really bizarre. So you package up your payload, and we've shown, the guy called Norm Hawkey and Alex Dickens, we were able to show that astrocytes will release extracellular vesicles. Those will then travel across the brain endothelium into the circulation and make their way to the liver. So some things can cross. I don't think there's a requirement for losing the patency of the blood-brain barrier. When that happens, bad things tend to happen to you in terms of neuronal function. But so the blood-brain barrier is an obstacle to solutes, oddly not through extracellular vesicles. In terms of how does, most of the things I've talked to today, there is clearly a very distinct metabolome in the serum that is distinct from the CSF. And you can see changes in the periphery that are not reflected in the CSF because, of course, the way in which the blood-brain barrier excludes them. So it's, the barrier function remains, fascinating, but I think that in the context of the sort of things I've been talking about today, the blood-brain barrier is intact. And that might be my time up, I would have thought.
Thank you very much. So we should stop discussion here. And if you have some questions, then you can go during the coffee break. And now, so let me introduce our next speaker, Professor Tatyana Strikalova from Graduate School of Life Sciences, University of Wolfsburg, Germany.
Okay, hello everybody. It's really great pleasure for me to be here and to meet new old faces more and more. So I'm very grateful for ongoing collaboration to Shalpan and to, well, other people, and we'll show some pieces today of former work we've done together. So I will speak about amyotrophic lateral sclerosis and some new treatments that we were trying to try out in this model, even though this is not the main topic of my lab, but it was fun to try out this model and some new compounds or old compounds we had a chance to use and some other treatments. So amyotrophic lateral sclerosis is a fatal motor disease, and it's most common motor disease in adults. It can be sporadic, it can be familial, and it can be caused by very different mutations, but what common thing about it, that the symptomology is very, very similar, regardless the causing gene mutation, and also there are no standard treatments that would be effective or somehow prolong life of patients sufficiently. So they're all costly, and maybe a part of reason for that actually the real mechanisms of this disease remain to me unknown, even though many multiple pathways, many secondary pathways that underlie the disorder and pathology have been identified. But they all converge to quite uniform kind of pathological mechanisms. For example, one of the main genetic cause of disorder is a mutation of superoxide-1 dismutase gene that is a regulator of oxidative stress. However, there are many genes whose mutation is underlying the disorder have been discovered in the meantime, and there are around up to 100 genes now known to be involved in the disorder. They are mostly DNA, RNA binding proteins, genes encoding those are underlying the disorder. But interestingly, well, as I said, they all, regardless, their functions result in quite uniform pathological changes in terms of symptomology, symptoms, disease manifestations, and the cellular mechanisms that underlie the disorder. Those are aggregates in the motor neurons which have toxic properties. Those are, well, also changes in the oxidative stress mechanism defense, and also, interestingly, what is typical for the disorder is anatomical specificity, because it affects particular parts of the spinal cord, such as upper and middle part of it, and motor neurons of spinal cord and of cortex, of motor sensor cortex. So it has been described like 150 years ago by French Dr. Charcot, but still the let's say mechanisms of the disorder are not well understood. What is known is that oxidative stress and neuroinflammation, as well as glutamate excitotoxicity, are important mechanisms of disorder. They lead to the aggregation of neurofilaments in the cells and the death of neurons. One of the genes whose mutations are causative to the disease is a fusin sarcoma protein encoding gene, or FUS gene. And our colleagues in Cardiff University in their time were quite successful in developing a new mouse line which expresses a transcoded form of the gene, 12359. So actually, the so far existing models, genetic models of ILS were not very successful. So they poorly recapitulated the neuronal deaths and the symptoms of the disorder. Well, with this new mouse, one can observe, well, symptoms, let's say, typical symptoms of the ILS, as well as cellular abnormalities which are typical for this disease and associated with FUS dysfunction. So the mice that they have actually generated are quite unique in terms that they have a very defined time window of manifestation of the pathology. In comparison, for example, to SOD1 deficient mice, which start to develop pathology at the age of 9 months. So these new FUS1359 transgenic mice start developing paralysis in a time window of age 3 months and 3.5 months. Actually, it's time at age 3 and 12 months, they already reached the human endpoint. So it's really handy just to looking at the, let's say, lifespan of mouse, judge whether any treatment is effective or not in the mouse. Well, these mice display muscular atrophy and, of course, neuronal death. Already at this pre-symptomatic phase, they have deficits in behavior and overexpression of pro-inflammatory factors in the brain and in the serum. Well, this work has been done by Alexander Trofimov sitting here. Yeah, they are sensitive to pro-inflammatory stimuli, and they show then deficits in behavior such as helplessness, anxiety, deficits in novelty exploration, and then also high response to pro-inflammatory factors. Symptomatic mice have severe muscle atrophy, severe deficits in motor functions in classical tests for motor functions, Paul test and Wire test, for example, and they have clear pro-inflammatory changes in the brain and periphery. What we were trying to do, we were trying to use standard treatments and new treatments to validate the model. So the standard treatments are very limited in terms of number of drugs, at only 2 drugs which are approved. So this is riluzole, which mostly acts on glutamate release, and edaravone, that is supposed to be antioxidant compound. So we were trying to see whether some new antioxidants, low weight molecules, actually that we found to be effective in other animal models we were using, reducing oxidative stress and inflammation, would also be effective in the ILS model we were using. We also used stem cell therapy and started to use some new nanoparticles that potentially target protein aggregation. While the stem cell therapy was explained here today nicely, so thanks to collaboration with Neuroplast company, we had a chance to investigate a mixture of human mesenchymal and hematopoietic stem cells, which supposedly exert paracrine effect, secreting neurotrophins as well as anti-inflammatory cytokines. So we were injecting the cells, but in fact they were already shown to be effective in the other model of regeneration, such as APP-PS1 mice, a model of Alzheimer's disorder. This work has been done in collaboration with a group of Shalpandaskarova, and we showed that injection of these cells could reduce the expression of pro-inflammatory molecules, pro-inflammatory cytokines, and reduce the formation of amyloid plaques. That was already quite nice. So in the very same experiment is cells, stem cell therapy, ameliorated expression of neurotrophin and insulin receptor mediated factors. It was also quite impressive. So in the study, we were using the cells as well as riluzole and a classic anti-inflammatory drug, celecoxib, selective COX-2 inhibitor. And what we have found that as a symptomatic phase, when we could already observe a start of paralysis in these mice, for example, neurocell-treated animals had normalized emotionality in terms of helplessness, anxiety, and novel object memory. So we also found the rescue effect on the muscle atrophy that was only observed in the neurocell-treated group, but not, for example, in mice receiving riluzole. We found also the reduced expression of cellular stress markers and inflammatory markers in the spinal cord and the serum. Interestingly, celecoxib, for example, had peripheral effects, like we had reduced markers of inflammation than in the serum, but it was quite ineffective in terms of central markers and the physiological effects in these mice. So not surprisingly, motor functions, well, all improved in the stem cell therapy treated animals. So it was quite impressive result. Well, as I said, we were quite keen to study compounds that were very effective in other models in reducing oxidative stress, for example, in testing them in this new ILS model. So for that reason, we used compounds which are derivatives of vitamin B1, that is thiamine. So we found again clear effect on paralysis in these mice, muscle atrophy, and reduction of oxidative stress markers and inflammatory markers in the spinal cord. That was quite clear effect. And thanks to collaboration with University of Oxford, we also had a chance to do the metabolome. It has been done there. Again, so it was a clear effect on these parameters. So we had some pilot experiments done with some H2S donor, sulfide hydrate donor that is called POM16. So this mechanism is a kind of new, a relatively new mechanism of epigenetic regulation, which seems to be involved in the ILS pathology, and well, still remain to be studied, of course. But then we could also found increased lifespan of the animals treated with this low molecular weight molecule. Well, but interestingly, we found that many antioxidants that would be quite effective in other animal models in which we see oxidative stress and inflammation would be completely inefficient in the ILS mice. So we've tried, well, as I mentioned, silicoxib, for example, is ineffective, but we've tried other analogs of thiamine or even herbal antioxidants that would have zero of effect. So some antioxidants are effective and some are not. All right, so this, all right. Yes, well, we've tried new nanoparticles, and for example, fullerene, which is supposed to have not only antioxidant effect, but also have other mechanisms of action, acting via microbiome, for example, and zirconium oxide. But this work has to be continued, so it's only pilot, let's say, data so far. But clearly, the model is has high validity, and some of the treatments, maybe like stem cells or dibenzine can be probably used in clinical studies rather soon. And I would like to express my gratitude to my collaborators. Well, most of the work was done when I was in Würzburg University. Some of the collaborators are here. You could recognize them from the photos. Thank you for your attention.
Thank you, Tatiana. That was an awful lot of data. It's a truly horrible disease that kills very quickly. So buying any kind of time is good. I was just interested, before I open it to the floor, I was just interested in I don't really understand, why are there peripheral cytokines floating around in this model? You see the animal's waist. You showed that although they should be able to do lots of pull-ups, that animal was obviously having trouble with his pull-ups. But why is there peripheral? 400 nanograms per mil is a lot of IL-1. Why is it there?
Well, I think you have, well, the tissue is wasted, so it's just a general neurodegenerative kind of process going on, and also muscle degeneration itself
sort of
probably results in this. And then maybe it's kind of over compensatory effect as reparation.
I mean, it's not that these animals have just got infections on board, is it? Because of course they don't move around so much, and maybe they get sort of UTIs or something.
This is probably not the case.
Yeah, it's odd though. It's right, a lot of IL-1. Okay.
Okay, but thank you for your question.
Do we have some questions from the floor? Evgeny.
I guess it's a general question. Actually, I've always been surprised. In ALS, there are proteins involved in just general cellular things, like reactive oxygen species or fuses involved in many different cell types. Why alpha motor neurons are suffering the most?
Well, there are some hypotheses, well, trying to explain this. For example, that motor neurons are very kind of sensitive to energy exchange and mitochondrial function. So, and many genes, mutations of which causing disease actually are regulating mitochondrial function somehow. So, but actually there is no clear answer to this question. It's also not clear why only specific parts of the spinal cord are affected. For example, neurons that from the cortex innervating optical nerve. So optical nerve, for example, is not affected at all. It is also not clear. Yes, so, well, as has been said, still the understanding of this pathology is quite far from, from ideal, therefore.
But muscle cells also are sensitive to energy. Maybe it's
yes,
coming back to Daniel's question.
At least experimentally, there are no evidences for explaining why, motor neurons suffer in the first place.
Excellent. Any more questions? We've got time for another question. Yes.
Maybe I can ask a question, a simple one, because
we have a microphone.
And Daniel mentioned biointelligence before. Do you have any evidence that your neural cells are having any additive effect, or is it simply reflecting what the effects that we see with currently approved drugs, which have a limited effect of a couple of weeks in human patients?
Additive effect. Well, we've tried to find a combination of stem cell therapy and lecanemab in a time. That's in a different model, in the model of Alzheimer's disease. So I think it's hard to claim. It was rather maybe a negative effect of this interaction, to be honest. Well, we think that this acts as secretome, so to say. Well, but so far, well, maybe it's a good question to study experimentally using, for example, a combination with riluzole. That's something to do in the future. Thank you.
Yeah, that's great. So we'll close there and we'll move on to our next speaker. I think just a comment there, finding orthogonal drugs is definitely necessary. As you say, if you're additive but you don't actually have that added effect, but you need to obviously to find something that's orthogonal to what you've already got, which is, and that's the way to go. Right, we're going to hear about periostin, and looking forward to hearing about what I'm going to be smearing on my legs in the future. No.
Now? Yes, okay. Okay, so first of all, I'd like to thank the organizers for giving me the opportunity to present in this meeting. Okay, so I'll start off with the title, and it's a very simple question. Is the multicellular protein periostin involved in peripheral nerve regeneration? Okay, so the first thing is to start off the story. So where did it all start? And actually, it's a long time ago that we knocked out a gene and to our surprise found that there were severe neuropathies in these mice. They die at birth, some of them even embryonic. And the most striking phenotype that we could see was that during the embryogenesis, there's a situation when Schwann cells have to migrate out in the peripheral nerves, and this is completely stopped, as you can see here in these mouse mutants. You also see here staining with SOX10, and you can appreciate that there is no SOX10 signal in the periphery, while within the dorsal root ganglia where these to be migrating Schwann cells are located, there is a lot of signals, so they are there, they're present, but they're not migrating. So this lack of Schwann cell migration, so the absence of them further away from the ganglia, prompted us to ask the question, what is happening there, and maybe to establish an in vitro system and find some genes that are involved in this process. At the same time, we were lucky to see that there were some protocols published to cultivate embryonic dorsal root ganglia, so we established that in our lab. And as you can see here, we had the mouse mutant ERP3, and here the wild-type animal, and you can appreciate that after a couple of days you have tons of Schwann cells that migrated out along the axons that are very well developed, mature and healthy, while here you have very thinned out axons and hardly any Schwann cells. It is, of course, an artificial system, but we tried to make use of it, and you see here in a little bit of a time-lapse photograph that this is a process that is going on for several days. So we actually looked at it at a very early stage when a few cells had migrated out and tried to find genes that are upregulated in the system. And ultimately we came up with a list of different molecules confirmed by several different techniques, and the most important ones we analyzed further, and one of them was periostine. So this is an extracellular matrix protein, and it's not just a matrix protein. In the meantime, some of you may know it's a family called matricellular proteins. So it is a secreted protein, it's present in the matrix, but it's not just a structural protein, it's doing more than that. And this is reflected in the structure, and you see here a very unique structure for periostine is these Fas domains, 4 of them. You also have some other domains, but the important thing here for the Fas domain is, yes, it interacts with other members in the ECM like tenascin or fibronectin or collagen. That's mainly done by the EMI domain. But the other important thing here is that it also interacts with integrin. So you can see different integrins that are strongly bound by periostine and activated by periostine. You can also interact with bone morphogenic proteins. So it is doing more than just structural things in the extracellular matrix. So when we identify this molecule, the obvious thing, as I'm a mouse geneticist, we did the inactivation, and you see here in the upper thing line the genomic situation, 23 exons in the mouse. We chose the safe approach to knock it out by introducing Laxet into the ATG of the endogenous periostine gene, and by that not only inactivating the gene but also have a nice marker that we can trace. I just want to show you briefly the classical thing. At that time when I was doing that, we still had the classical approach of going via ES cells, generating a targeting vector, then proving that we did the right thing, and as you can see and appreciate here, we managed to do exactly what we wanted to do. We inactivated the gene, we have reduced protein in the heterozygous, no protein in the mutant. But then, when it came to publishing, unfortunately, other groups were quicker than us, and actually all these phenotypes that are described in these publications, we actually had been working on and had, I would say, even stronger and better data than the ones published, but we were always scooped. So the first paper actually out, there was a paper claiming that these mice are dwarfs, and then they added something after discussing with me and also getting some of our data. They added incisor animal defects. Then the next paper here is actually focusing more on the incisor eruption defect. And there were more papers published. I add just 2 that focus on the situation periostine absence causes for the heart. So in one case it was a problem with atrioventricular valve formation or maturation, and here an effect after myocardial infarction. So all these things could be true or are true. Unfortunately, we did not publish any of this, although we worked on all of these things. But what we actually described and did not publish is to be seen here, and this is actually a rodent incisor as it should look like, lower and upper, and this is how it looks like in the periostine deficient animals. So it is clearly obvious that there is a severe problem, and in order to establish the constantly happening eruption of incisors in mice, we found a method to mark them and actually measure it, and we could actually show that the incisor eruption in periostine deficient mice is severely impaired, and this is also the reason why the incisors look like they look in these poor animals. And you may also realize in these graphs there that there is a differentiation between hard food and soft food. So the very simple explanation for this dwarfism is that the standard food in mouse holding is pelleted food, very hard, and the animals simply decide to rather not eat and die than to consume this pellet food. If you provide them with soft food, they are actually normal, I would say. Normal means they have a phenotype, but subtle, and you have to look for them. What we also found during this process is, and you can appreciate it here, that hard food chewing actually induces the expression of periostine in the periodontal ligament cells. Okay, so we found the abnormality of incisor appearance is due to the eruption. Periostine is essential for this process, and actually these animals can lead a fairly normal life. Okay, so we were still interested because we pulled it out in our essay of Schwann cell migration, embryonic Schwann cell migration. So we tested this in these animals. And as you can see, and hopefully appreciate here, the dorsal root ganglia cultures that we had established clearly showed that there is a migration defect in these periostine deficient animals also in vitro. So there is an effect in vitro. However, the animals somehow managed to overcome this. So if you look at the nerves in these animals around birth, during the embryonic development, Schwann cells can be found, and I would say there is no significant change. There may be a subtle reduction, but not a significant change of the migrating Schwann cells. In this assay, as you can appreciate here, you clearly see a reduction of the migrating Schwann cells. Again, in this artificial system, we found no evidence of any additional proliferation or apoptosis. We also tried to do it on embryonic nerves. And I would say these data are consistent. There may be subtle changes, but not really strong changes in proliferation or apoptosis. So this means now we have an animal that is more or less healthy. We know that there is a secretion into the extracellular matrix, so we were interested in looking at interactions. We found that there is a strong dependence on calcium, can interact with collagen, laminin, fibronectin, everything fine. If we look for laminin, for instance, you can see that we can co-IP it with a laminin antibody and then find the periostine. And if we look for cellular localization, we find a large overlap, so they actually localize together in the cell. However, when we looked at the mouse, it was healthy. So we asked the question, what happens if you actually challenge these animals? So we induced a nerve injury, and as you can appreciate here, starting with the 12 weeks, which we see, let's say, called the normal situation in an adult nerve, you can hardly see any periostine expression. However, 1 week, 4 weeks, and not shown here, 8 weeks after injury, there's a massive upregulation of periostine distal to the injury also slightly proximal, but mainly distal to injury. Interestingly, in the homozygous, we see that this upregulation persists much longer than it does than in the heterozygous animal. So we, together with a colleague, had a method how to analyze nerve injury. Actually, it's a colleague from Hamburg, Andrei Irinchev, who had established this model and taught it to us. So you can actually injure this nerve, femoral nerve, and then when you cut the nerve, you can bridge the cut and actually use a silicon tube to connect both ends. And in the silicon tube, you may also add goodies like growth factors, modified Schwann cells, matrix, whatever. But simple story here, when you do this, you can follow what happens after this operation. And you see here a wild-type mouse that is just simply walking home to its home cage, while here, after injury, the mouse is still doing it, but of course it is impaired. And when you look in more detail, you can appreciate that there is a difference between the walking styles. So there is a so-called foot base angle that's smaller than 90 degrees here, this angle, and the heels tails angle that's larger than 180. And in the mutant, in the injured animals, these angles change significantly, so bigger than 90 degrees or smaller than 180. And this can be followed longitudinally, and thereby you can monitor the regeneration capacity of these animals. You can also add other parameters like some treatment options, whatever. So it is an interesting tool that we have in hand. We have also the mutant, and preliminary data, as you can appreciate here, clearly show that there is a delayed regeneration capacity in these animals. So coming back to this question, is the matricellular protein periostin involved in peripheral nerve regeneration? The clear answer is yes. What we also did, and I have no time to go through this, is we're working with, on bladder cancer with periostin deficient animals. There's some evidence that periostin plays a role there. We have RNA-seq data, and you can see here that there's a massive difference between wild-type bladder cancer cells, mouse cell line, and one that we remove periostin with CRISPR-Cas technology. And if we reintroduce periostin, different splice variants, the pattern changes again. So definitely periostin is not just doing something in the extracellular matrix, it's doing something inside the cell. Actually, the cells are different, completely different, whether they express periostin or not. So I just want to briefly mention the team. I don't go through all of these. These are the members of the team here in Kazakhstan. For this project, I want to thank again Andrei Irinchev, who taught us this method. Of course, Eva, who did a lot of the experiments. Alim and Anton are the ones that work on the bladder cancer project and provided this RNA-seq data. And in general, I also want to say that we have other collaborations going on with Dr. Eugene Tulchinsky and Marina Krajewskaya on the bladder cancer project, and also very briefly on a ALS project with Dr. Larissa Kuanova and Tursun Jantokai. I had no time to talk about this today. Thank you.
Thank you, Dieter. That was really, really beautifully presented and right on time as well, which gives us plenty of time for questions. I wondered, I'll kick off, but I wondered if there's an advantage. So obviously you saw more regeneration. I'm just thinking in terms of sort of neuroma formation, or whether actually inhibiting periostin would actually be useful in certain circumstances after sort of, we don't know who's going to develop sort of neuromas, but is there something, could there be a benefit in suppressing? And then a follow-up question, obviously it's all about peripheral nerve, but what happens in the CNS? And is there any evidence that this is at all useful for encouraging any kind of regrowth, or at least the sort of the appropriate rewiring of the CNS?
Okay. very good questions. Let me start from the end. In the central nervous system, we were of course also interested in that, and having the knock-in mouse, so we were able to, with Laxet, easily look for periostin expression, and it is hardly visible in the central nervous system. Interestingly, over the years, many people were looking in more detail, and upon injury, periostin is upregulated in these areas. So there is a potential implication in several aspects. My current interpretation of the data would be that it may be more involved in neuronal inflammation rather than in trying to facilitate regeneration. As you know, there's a significant difference between the capacity to regenerate peripheral nerves and the central nervous system. So I have a little bit of doubts that adding a few, let's say, pro molecules that enable neuronal extension or migration would actually work in the central nervous system, which is basically inhibitory.
I mean, and obviously we're familiar with the idea that actually trying to do chondrogenase treatments, for example, in spinal cord injury, I mean, that may go somewhere, but oddly getting rid of matrix seems to be often the issue. And for the neuromas, and whether it might be a sort of an advantage in sort of not having these sort of, if you have peripheral nerve damage, that actually it could prevent some neuroma formation?
We have no evidence that it would be at all beneficial. I think to have periostin in peripheral nerve is definitely a big advantage, and I think it's also most likely involved in the improved clearance of debris after injury. So yeah. It may have a dual role there.
Fascinating. Questions?
So let's look like periostin involved in two functions. In one, migration of Schwann cells, and second is regeneration of peripheral nervous system. What's the mechanism? How?
Very good question. Having shown you the structure of periostin and the interaction with integrins, I think very likely the interaction with integrins is the key to the answer here. You may know, as you asked this question, that integrins are expressed in neurons. They are strongly expressed in developing neurons, but they are pretty much downregulated in the adult. However, upon injury, they're re-expressed, and one of the candidates that is upregulated in neurons in that period is alpha V beta 3. So this is actually the one that is interacting with periostin. So I think there's good evidence that periostin may do good things in peripheral nerve regeneration, not only by giving a favorable surface, but also stimulating neurons via integrins.
That's great. And we've got a question over here.
Thank you for the presentation. My question is, so periostin increases the migration of the Schwann cells during the traumatic nerve injury. Yeah? What about the non-traumatic causes of peripheral nerve injuries? And can it be a target for the future treatment, in the future, for the treatment of the peripheral neuropathies?
Well,
thank you.
Given our preliminary data, I would say that there is a potential that it could be a potential target for future therapies, but we need to have a lot of more data also about the mechanisms. The reason why we don't have these data, because we have the tools in hand, is simply that we tried twice, also here in Kazakhstan, to get money for it, and we weren't successful. So right now we don't have the money to do the experiments. But if we can, we will hopefully be able to prove the principle, and then it may further develop into some treatment in the future. Let's hope.
Okay, sorry colleagues, we need to stop here. We can continue discussion during the coffee break. Dear editor, thank you very much for such an interesting presentation. And it is my pleasure to introduce our next speaker, Professor Leojan Sugurova, head of the Center for Paleogenetics for Institute of General Genetics, Kazakhstan. First of all, I would like to thank organizers for inviting me as a speaker and for such interesting, bright event dedicated to the 15 years anniversary of National Laboratory Astana. And I would like to say that I remember time when all beginning, when team collected here under the, in the Nazarbayev University. And now, 15 years later, we see that each bright, talented young scientist now is a leading, has a leading position in this university and help us to support broad international collaboration and help to construct here really friendly scientific environment in our country. Thank you. Let's go to topic of my speech. If you look at human history, human evolution, the most interesting and most surprising thing is evolution of brain function. For example, if you compare cognitive functions in children, nowadays children, and cognitive function children with 50 years before, you can recognize big differences between it. But this evolutionary process has obviously side effects, and the increasing rates of autism spectrum disorders, increased rates of neurodegenerative diseases like Alzheimer's disease, Parkinson's disease, oh, sorry, I forgot. Parkinson's disease is a side effect and depend on both increased lifespan from one side and another side improved diagnostic tools. So if we look at statistics of Parkinson's disease and Alzheimer's disease in Kazakhstan, we see the big difference between diagnosed cases. For example, in 2023, Parkinson's disease cases officially registered in 11,000 individuals older than 18 years. And third part of this amount was individuals younger than 60 years. But if we look at statistics in this year regarding Alzheimer's disease, we see only officially registered 155 cases. And of course, this disease is very this disease is the main reason for dementia, and this is the first diagnostic sign of this disease. So, we, a little bit early, we provided some studies of epilepsy, non-traumatic forms of epilepsy, and autism spectrum disorders. It was case control and family studies. And what we discovered that in families with strong family history, the parental mutations not inherit from one of parents very often, in 30% of family. But de novo pathogenic mutations we can detect in these families. So, another study which was promoted by Shulpan Askarova, case control study of Alzheimer's disease patients, and we helped to collect material from Almaty region and also did the genotyping of case and corresponding control cohort. using InfinoImmunoArray kit and iScan Illumina platform. So the primary treatment of these case control cohorts, not so representative by ethnicity, age, and other populational characteristics, but we recognize SNPs that can be associated with Alzheimer's disease, but probability was quite low. When we removed didn't annotated SNPs and intergenic SNPs, for example, only 13 SNPs can be considered as associated. If we look at distribution SNPs between the patients suffering from Alzheimer's disease, we noticed that sometimes appeared haplotypes. This plot present the number of SNPs and the number of patients where this combination of SNPs appeared. And for example, this haplotype analysis revealed combinations, most significant combinations of 5 SNPs, 4 SNPs, and 3 SNPs together. And if we look at related genes, there are 3 genes involved to these combinations. It's TOM40 genes that is outer mitochondrial membrane translocase per subunit. APOE, famous apolipoprotein E encoding gene, we know about association with Alzheimer's disease. And another MIT12L gene that is the famous mediator of all kind of transcription, because it mediate the RNA polymerase II. And association of this gene with intelligence disabilities, with autism spectrum, also famous. MIT12 genes located in 3rd chromosome, but TOM40 and APOE encoded by they are in the close proximity in 19 chromosome. And analyzing the possibility interactions, we discovered that we can use SNPs despite of different function of these genes. We can use genes of TOM40 genes as a marker for fast revealing APOE gene status. It may be useful for clinic for faster diagnostic some defects. But after this, we gave our results to teams from Nazarbayev University, and today I hope you will got acquainted with new results where Alzheimer's disease represented only by Kazakh population, and maybe it's something new will be eroded from this. But because in our institute we have Center for Paleogenetics and Ethnogenomics, and our former and current projects related to analysis of modern population and ancient populations, and we have really big database, gene bank representing 3,000 modern Kazakh individuals, Kazakh ethnicity, who knows his family history, tribal affiliation, geographical location, etc. And also our current projects related to the search of signals of natural selection in ancient individuals. And I will try to compare these results. Central Eurasian region was started to populate only in the late Stone Ages, in Neolithic time. And more population came to this vast territory in Bronze Age, distributed in Bronze Age and early Iron Age. And then other migrations, massive migrations from west to east, from east to west, Hunnic migrations and medieval migrations led to creation of ethnic background for this territory. So we combined our data and all published data of ancient individuals from this territory and made groups. You see on the plot the map where necropolis is distributed and time periods, Iron Age, Bronze Age, Neolithic, in early Middle Ages also. So this plot demonstrates the principal component analysis for these populations, and you see the ancient individual colored by different colors pointing the time period and cultural traditions, archaeological traditions. And the gray, it's individuals of modern population. You see below the list of used populations for this analysis. And if we looked on this distribution, genes components, we see that population of Neolithic time, it's how it say, on the north of this cut. Had a few samples of different ethnic background, but Bronze Age populations in the West, and you see in the center of this blue-colored individuals, it's a main Sakapul middle late Bronze Age steppe component. And our nomad tribes, which we call Saka, mostly distributed here. And then, for example, Hunnic influence or influence of Silk Road, it was gene flew from east and south mostly. So, and here the admixture analysis yesterday was in your presentation. I think you remind that you found out Hunnic Chinese percentage in your DNA analysis. And first of all, we need to know reference populations to compare, to find the components of admixture, our gene admixture. And we put on this slide all typical representatives of archaeological culture, and we see that how differences gene flow can reflect and inherit in some populations. And this plot represents principal component analysis from obtained from genotype at also using global scan kit. SNPs, more than 650,000 SNPs of whole genome, modern Kazakh. And then we excluded all relatives, first, third degree relatives. We imputed data, and the analyzing number of SNPs exceed close to 900 SNPs. So, and the same analysis yesterday showed Ulukbek, Kairov. We also used data from 1,000 genomes and 5 populations, European populations, Southeastern populations, red, and East Asian populations. And you can see that, for example, yellow, market but yellow, oh sorry, Han Chinese, yes, Han Chinese populations yellow, it's close by genetic mixture to Eastern Asian populations, and also Japanese go to this cluster. And Kazakh, modern Kazakhs have a central position and quite homogenized to the moment. So then we started to do natural selection analysis using PC-adapt method. And we see that according to false corrections, if we use all general 470 individuals included to the analysis, with high significance, 413 SNPs can be considered as candidates for selection process. But if we divide our sample size to time periods for millennia, we can see that in early times, the Stone Age, for example, different, no really selected candidates for natural selection because admixture analysis showed different origin of genes in different individuals with different genetics. And then it is obvious that the older the samples, the greater the number of SNP candidates for natural selection. And you see it's quite big numbers of signals. But we use other filtering, RF mix method, which can count genetic drive and genetic gift and admixture. In taking into account this, we excluded all not really changing in allele frequency and do the annotation of results of these SNPs, and you see morphological traits, you see diseases here, and representative by chromosomes. Here, yes, I close to finish, thank you. Here, we see clustering, and you see that if you look at gene ontology, many factors, many biochemical processes can be reflected on development of nerve system and appearing of some pathologies. Very interesting results of haplotype analysis, because you see first plots reflects ancient populations and a signal of selections on 6 chromosome, where mostly genes of immunity localized, and chromosome 8 here, where genes association with different processes, also nerve signaling and premature aging localized. And very interesting results that our Kazakh population shows strong signals on 6 and 8 chromosome also. So, it's the last slides. This slide present data on neuro category, included Alzheimer's disease, Parkinson's disease, autism, cognitive functions, etc. And number of reliable SNPs, K genes, selections type, positive, negative, and time period where we noticed it. So what we can conclude? We can conclude that the main genetically determined causes of nervous system function and dysfunctions may be associated with mitochondrial dysfunction, signaling system disorders, immune response, oxidative stress, DNA compactization patterns, regulation of gene transcriptions, protein activity, protein utility, and other general mechanisms. An increase in genetic load has been noted in the modern population of the Central Eurasian region. In historical view, the selection of negative polymorphism predisposing to the development of neurodegenerative diseases possibly began 3,000-4,000 years ago, which is a period of massive inhabiting of this Central Eurasian region. In current millennium, the tempo of negative selection is quite strong. And I'm finishing, only thank our, yes. Could you please acknowledgements put, yes, yes. I would like to thank the teams from National Laboratory Astana, Chopin Askarova, Lukbek Kairov, our lab staff who was treat the modern DNAs, Azada Hamdieva, Kanagat Yergali, Jesulan Niyazov, Naza Maltinova. Our collaborators who help us to look ancient DNA, Johannes Krauss, Max Planck University of Evolutionary Anthropology, Chung-Won John, Seoul University, Guida Alberto Riscona, Tübingen University, Aynash Cheldebayeva, who represented our institute, and now also Texas University in Austin. And our lab staff, Saida Takmurzina, Maxim Shtilkind, Aykurke Mustafa, Tamerlan Kireva, Lazat Musrailina, who is sitting here. Thank you very much. Thank you very much for that interesting presentation. I'm sorry that we don't have time for questions, but the good thing, there's coffee break, and if you have questions, you can reach her directly and ask questions. And please, please, please be on time in 30 minutes. We'll be here at 4:45, because we are already running late, so please don't be late on the coffee break, okay? Please.
еще речь. Специально поставила, что нет? Нет, так жестко. Значит, 5 лет будемся. Все наши. Ну, не моя вся, кто захотел пойти. Все наши спикеры будут, твои будут, Гитлер и Ленсин. Нет, ты что, все уже считано? Нет, изначально списки есть. Ты же списки посчитаны. Но если ты не придешь, то вместо пропадет. 35 тысяч говорят. Конечно. То, что мы же сами спрашиваем подтверждение. Ну, подтверждение. Ты у нас, вот как называют, выбрал тоже истоков. Все наши. Дымом пригласили на это. Не важно, почему. Мы же знаем. Мы-то знаем. Мы же приглашены. Вот Биом даже приехал. Ну, приехала сюда. Спонсор это ДНС, который вот зал мой, который нам много остальных контактов. Ну так это было. У нас спонсоры, у нас вот зал мой. Сейчас не 7 миллионов долларов. Это мы им дали здесь. Вот это наш 7 миллионов. Это наш день роста. Фарус. Ну, я бы Олимпус не брала. Мы же купились, вот так на Олимпус купили, лучше царь купили. Мы же тоже показали класс, там интерфейс очень удобный, там много всего.
Я говорю, Денис молодец. Умный, прекрасный, лучше нас.
Почему лучше? Только сами коллаборируют активно. Его PhD-студенты, это наши PhD-студенты. У нас совместных статей много.
Наука это вот так, то финансы есть, то финансы нет.
Ну ладно, они каждый год всякие ресурсы продать, не продать, не продать. То же самое. Потом у нас это, знаете, что? У нас флексибилити больше предлагается раньше. Мы не привязаны к урокам, к семестрам. То есть, я вот хочу, сплю, хочу. Это же для женщин тяжело было. Это же не просто лекцию же пришел, читал, там готовить, все приходилось. Я не сказала, не их жрать, приходите, говорю, я сам жру. Нет, мы за министра. У нас министр пришел министр, мы думали, он там 5 минут скажет, что-то приветственное слово, а он на 20 минут только. Здравствуйте. Ничего, ваши дела. Ничего, нормально. Послушать, да. А я знаю, вы специально пригласили. Ну да. Вас пригласили специально, что ли? Да.
Хотели мы завтра
Конечно, Мы вообще, мы с вами хотим приехать. Приезжайте, приезжайте. Я могу уже. Она говорит с Китаем что-то. Ну, она вроде хотела. Каких числах? Я вам информационно. Хорошо, хорошо? Спасибо. Да, да. Ладно, все, тогда. Тогда еще связи будет. А этот, Ленар Сагадыковна, на банкет? У нас Сибирь, Гарден. Младшая, да, там сейчас все надо контролировать. Это тот ребенок, когда вы были в декрете, когда мы искали с кем работать? Или другой ребенок? Это уже следующий. Это, да, мы-то с 15-го года, мы вчера вспоминали с вами. Это еще была маленькая, которая средняя, она уже в 6-м. Мы давно. Вот я и хотела ужаснуться, думаю, неужели тот ребенок, который... Нет, это не тот, это другой. Хорошо. Поэтому, понятно. Все, ладно, на связи. So, we're supposed to do wait for people finishing.
Shall we go and shout? What time is we meant to? I think I'm assuming they're going to come back. We can't start with this number of people in there. Do you want me to go and shout at people and tell them to come back?
No, we have keynote speaker, and I want people coming in.
Yeah. I was going to say, do you want me to go and I can go and tell people?
I think it's useless. Do you
know anybody who's there that you can telephone and ask them to tell them to come?
I think you will start the session.
Yeah, sure.
No, all my people are here. I can't go to anybody.
Tatiana is stood over there. I'll tell her. We are waiting for a slightly bigger crowd. It doesn't. We're having some bloody thing. Go and play with it. Well, all I would say is don't press the buttons. So it doesn't actually have a pointer on it. Whoa! That's silly. This screen. So yes, I kept on trying to make it work, but it. Is it this or this? Well, so I was trying to do it with that, and every time I hit that, I managed to hit them two together, and that's how I managed to keep changing the slide.
So the top one is the one you should go with.
It just doesn't, not in a way that you can see it. That's all you've got. So my suggestion is you walk over to the screen, point at it, and then backwards and forwards with that.
So arrow is the moving the slides.
Yeah, and as I say, you saw, I seem to be incapable of not squeezing it at the wrong time.
Yeah, I saw you. Slide over.
Well, we're getting there.
Let's wait for 2 more minutes.
Yeah.
Ask people not to spend more time for coffee break.
Let me just phone Tatiana and ask her where she is. And whether she's got anybody else there. Hello. Are you still at coffee? No, you just walked in. No, don't worry. I was just. How many people are left at coffee? No, how many people are there?
Людей на кофе-брейк отпускаешь, потом не вернешь. Хоть вообще вообще их не корми. You don't need to go for offspring. Stay here, listen.
Yeah, I know.
And suffer from whatever, but if you cannot come on time.
Раз.
Right, everybody, I think we'll probably make a start, and our colleagues will slowly filter back in from the coffee. We made a mistake of allowing them to go to coffee in the first place. I think we should have just barred the doors. And they don't know what they're going to be missing if they don't come back soon. We're I'm very pleased to be able to welcome and introduce Professor Joseph Lee from Columbia University, from the departments of epidemiology and genetic epidemiology and neurology, which interestingly is the other way around on his slide, neurology to genetic epidemiology. But we shall now listen to hear what wonderful things he has to tell us about extreme sampling. Thank you very much. Yes, and I'll stand up when you get sort of closest to the end.
Okay. Hi. It's very nice to be here. I think I was here about 15 years ago, and I thank the organizers for inviting me here and to share what we've been working on Alzheimer's with you guys, and some of that may be insightful to you, and other things may be a waste of time, but it's for you to decide. Okay, let's see. Okay, so just in case some of you don't work on Alzheimer's disease, so let me just briefly go over. Alzheimer's disease is, as you all know, it's a progressive decline in memory, language, orientation, and motor function. And as they get older, there is some mood behavior problem, and activities of daily living becomes more and more difficult. And I'm going to show you what an artist view himself as in a self-portrait by this artist. So it happened over 8 years. This is how he saw himself. So this is what's happening on the surface, okay? And then the next slide is going to talk about underneath what's surface. This is what's happening in the brain. You see amyloid plaques building up, and then tangles are also building up, and it's killing off the neurons, and as a result, brain volume is shrinking, and then there's a problem with processing and metabolism and so forth. So this is very basic review, so that you and I are about on the same page on this matter. So just to remind you that Alzheimer's disease is a complex trait, meaning that there are many genes, as you can see. I was told not to use a pointer, so I will use my hand. You can see there are many genes, and family-level genes or population-level genes, and the environment factor all affecting the trait. And in Alzheimer's disease, studies have already shown that in order for amyloid to happen, there are many different pathways that are being involved, including cholesterol pathways, and whoa, it's very difficult to read. Well, I can't read any of these. I'm too close. And inflammation, and various other things that you already heard from previous speakers. And as it says, there are likely to be many pathways that will be involved, and this is a very simplified version of the true biological model. Whoops, what the heck did I do? Okay. Okay, so for many years since 1980s, people have been doing GWASs. As you know, GWAS is looking at the genotype-phenotype relationship, and it's very mechanical. You don't think about the biology, you write a code, and then the machine does the whole thing, and you find a bunch of genes that are associated with phenotype. Alzheimer's, or memory performance, or something else. And you can see that when the gene is making various proteins, amyloid and tau and some other protein or some other X factor that are involved, and these things lead to cell death and organ change and then eventual clinical change. And this is a different way of presenting Cliff-Jack's model. And then for each of these things, people have been using genomics, transcriptomics, proteomics, metabolomics, and imaging, PET imaging, and MRIs, and so forth. And the question is that I'm interested in is this. As people get old, these protein level goes up, and the brain shrink, and then some people somehow manage to escape dementia. And how the heck does that happen? And if we can understand what that factor is, maybe we can translate that into therapeutic means, and that may be useful for all of us, right? And then if this simple model is true, and then there has been anti-amyloid therapies recently, and then when I talk to my neurologist colleague, they tell me that anti-amyloid therapies are quite effective in removing amyloids, and yet when you measure their cognitive performance, they don't really come back to normal level, even though their amyloid level is not at the risk level that most people think is not good.
Excuse me, but if there are these drugs, if you treat at early stage, helps.
It may slow down, but it still will go up. But even at the early stage, if it goes down, your cognitive performance is not what it should be. Sorry?
Why FDA approves this drug?
I have no idea. If I knew that, I wouldn't be here. I would be in Costa Rica enjoying my son, right? So then I ask, well, what other proteins and other processes are involved? And then if that's the question, then you can say, hey, there may be some genes involved, right? And then it has been shown that in about 1/3 of cases, who are not eligible for these anti-amyloid therapies because they have severe adverse reactions. So then, taking all these into account, how can we understand the process better and prevent people from suffering from this disease? Whoops. Okay, so this is what we know so far is that this is a GWAS. That has been done in 2023, and you can see that they published 75 different loci, of which about 40 some odd were brand new candidate loci. And this was done by Belingues and colleagues. It used about 111,000 cases and 677,000 controls. That's a massive number, okay? So if you think about $100 per GWAS chips, that's a massive $80 million spent on this. So what did they find? They found that immunity, lipid metabolism, and APP metabolism are likely to be involved. And this is the kicker. When you take the significant SNPs that were genome-wide multiple testing corrected, average odds ratio was about 1.1. You're never gonna get published your paper if you find a gene that has an odds ratio of 1.1. But this was published in Nature Genetics. That's a pretty impressive journal, right? And the range is 0.7 to 2.4. Some are protective, some increase odds by 2. So that's not bad. But when you put it all together, it is average pretty trivial, right? And then when you put all the SNPs together and so-called PRS and various other measure of doing meta-analysis, you can assess that collectively they increase about twofold max. So that's not too impressive. But if you think about the amount of money that went in to get that results, is pretty impressive that we spend that kind of money, because it explained very small proportion of variance explained by the thing. So I've done those GWASs myself, so I'm not accusing anybody of being stupid, because I probably am stupid myself. So I do something slightly different, that I try to use extreme samples. So for example, I go to Puerto Rico. In Puerto Rico, there's only one PS1 mutation, G2068, glycine 206 to alanine. And so if you have a PS1 mutation in Puerto Rico, you always have that mutation. And then, so that if you can see how some people with PS1 mutation does not get affected, all I need to do is look for second modifier, right? PS1 modifier. And then I can sort of understand how two genes are interacting with one another. However, if you just study any autosomal dominant form, that's going to be a little problematic, and I'll tell you a little later. And then I also study Down syndrome, because people who have Down syndrome, third chromosome 121 has APP gene, and the APP is the maker of amyloid, so that they have, on average, 1.4 to 1.5 times higher level of amyloid in their brain, which is not good for you, right? And they have all kinds of other things as well, because there are about 200 genes on chromosome 21 that has third copy. And then these two high-risk group gives me an opportunity to find protective modifier. And then if I say, hey, these variants give me a protective effect, then I look at these families, longitudinal long life family study, are families that have 2 or more people who live to 100 years old, roughly speaking. And then my reasoning is that if these people have protective variant, I would say these people who are getting to 100 years old without dementia is because they may have a higher frequency, or the effect size of those modifiers may be stronger. So that's my reasoning. So by looking at these two as a discovery set, then looking at that and look to see what the heck happens in long life study in it, okay? So it's sort of like a stepwise program. So I just explained that gene environmental factors are working and they may interact, and the modifiers are a great way to understand how a network of genes are working with or against one another that may lead to AD or otherwise. However, when you have so many factors, you want to minimize heterogeneity, because when you have lots of heterogeneity, it's very difficult to figure out what the effect of a single SNP mutation is doing for a phenotype. And then after doing so many GWASs on my resume, I realized that all these things, multiple testing corrected p-value doesn't give me any satisfaction. So what? I found a SNP that has a 10 to minus 12. You know, if you have a rare variant, you can very well do it. What I want to see is that what the heck does it do functionally? So that's why I start doing iPSC on our samples. And then that PS1 started that concept, and then I can apply to Down syndrome. In Down syndrome, people, once they reach 45 years of age, they have essentially pathologically, neuropathologically, they have Alzheimer's, meet the Alzheimer's criteria. And then by 65, about 80% of those people get dementia. And what the heck happens to 20% of the people? How do they escape that? So that's, and then the lastly, as I said before, are those things more frequent and stronger in the long life, healthy aging cohort? So it's a logical way of progressing to see what I'm saying is not just a chance fluke. And so in 2001, we discovered this PS1 G2068 mutation, and I'd like to say that in this issue, Francis Collins wrote an editorial that says, in so many years, with a drop of blood, doctors will be able to predict who's going to have what. And I was quite impressed that I was part of this, publication, because it kept me busy for, since 2002 to 24 years, doing various other things. And just to show that this particular PS1 mutation, compared to the wild type, it has about 2 times higher level of A beta 42 to 40 ratio. And compared to Swedish APP, which has more toxic, it's about half of in between Swedish APP and the wild type. So it's halfway between. So that's sort of modest weakness, pathogenicity that this PS1 G2068 Puerto Rican type is experiencing. And let me tell you one thing, though. In addition, I had a summer student who had nothing better to do, so I gave her this assignment. Pull every PS1 mutation publication and tell me what their mean age at onset is. And then she complained all summer long, and then she showed me that some PS1 mutation mean age range from 35 to 60 something. So single autosomal dominant mutation is highly variable. That's an onset difference of 30 years, okay? Then the second thing I ask is that after they get diagnosed, how long do they live, right? Because if they die next day, the family doesn't suffer so much. If they live 10 years, then the family has to live with that for 10 years. And then this is what you see. On this side, the average years they live is very few. On the left side and the right side, that's over 10 years, right? So the pathogenicity varies significantly, even among people who have one SNP that changes amino acid from wild type to something else. And so after so many years, I decided to collaborate with Colombians who are known to have E288A mutation, and the Mexicans, they have A431E mutation, and Argentinians have M46L. So we got together and we wanted to do a study to see what the heck is clinical heterogeneity would be looking like. And then there were some NIH policy changes so that we were unable to submit the grant. However, and then they are in different exons, and you can see from here shows different pathogenicity of each mutation. And if the mutation is above 20, they're quite pathogenic. So Columbia mutation is 27.4. Their mean age at onset is about 44. My mutation is about 26.9. Their mean age at onset is about 55.7. It's not linear, but there is, as you can see, there's a significant variability. And last column, I left it blank because I have no idea what it is. And I understand Professor I have a hard time, Zoldo Silva is working on that. So we'll see how it comes out. So if you look at the PSO mutation, and at that time I had about 40 families, and then within one family, there are multiple people with PSO mutation carriers, and in some family as a whole, it has early onset. This is 55.7. And then as you go see, some families, primarily, most of the members are later onset, even though they have PSO mutation. You have to pay attention to this group of people because within the same family, same mutation, some people in the family have age of onset at around 40. Some people live to 80 without dementia. What the heck, right? So you can see that variability is really obvious, and we've been following them. When I have funding, then I go there 2-3 years and then reevaluate those people to see and collect blood samples and store until next funding comes along. And similarly, we can do the same thing with Down syndrome people. Okay, so this is one study we did on PSO mutation. We did linkage analysis, and we did association analysis under the linkage peak. And from these two loci, we found about 8 genes and the variants that have significant effect. As you can see that this column tells you if it says 9.3, that means having one variant is going to delay 9.3. If it's negative 11.69, it accelerates about 12 years. And this one, STOX2, will delay about 13 years. And you have to remember that age at onset is a very rough measure. But when it says you have a positive or negative number, some things delay, some things accelerate, okay? So this is early onset study. So when you tell clinician that, hey, we study 500 early onset people, and then neurologists will say, who cares? It's less than 1% of the Alzheimer's patients. So what, right? So these things may mean nothing to general population. So I looked at general population of same Caribbean Hispanic population, and because they don't have a mutation, I couldn't condition a mutation study, so I did gene-wise analysis. As you can see, some of them were very significant. For example, I said Shire-3 was in the later onset, still had a significant p-values. And you can see that some of these things are based on much bigger sample size, and they had a significant association. However, what is important to remember is that here you see the acceleration and deceleration anywhere between 5 to 10. When I looked at it in the late onset in general population, the change was around 2 to 3 years. The magnitude for modifier was much smaller, but because sample size was larger, significance was stronger. Yeah, so when I gave this list of genes to my cell biology colleague and say, hey, like I said before, I don't give a damn about p-values. Show me how it works. And then he chose SH3RF3, fine. And then we got a larger sample and we did association study again. And this is what happens among PS1 carriers. There is a significant, strong, consistent haplotype that are appearing in PS1 carriers, but not significant in non-carriers, right? And this is what's happening, carriers and non-carriers. So then it took them about 3 years to figure out that, went through all the cell biology experiment and show me that when you knock down SH3RF3, you see essentially no effect on APP transcription or any of the processing of APP. So, and then a year later, and then they knocked down SH3RF3, and they show that it lowers pro-inflammatory cytokines, so that he was arguing that microglia have reduced inflammatory responses to A-beta-42, and then further reduction in microglia inflammatory response in PS1 is carried by protecting, by reducing the effect of amyloid and neuroinflammatory process. So I buy that because I don't know any better.
Running out of even question time now. You've only got 18 seconds left of the slot.
Okay. All right. In addition to gene, we did exercise, looked at exercise, and then we found that people with PS1 mutation were, exercise was protective in those who were carriers. And then you could see that their memory was much better improved in those who are PS1 mutation carriers. Okay, so I'll have to, I don't have much time. Okay, so I'm gonna have to skip. So essentially what I did was, in a healthy aging cohort, we looked at lipids to understand how AD process works. And then going through what we found in that centenarian study and looked at it in Down syndrome and in the general population, we see a same signal in all three populations. And then we did transcriptomics and GWAS jointly, and it suggests that it was ALDH1A2 is the cause. And then lastly, my postdoc is working on iPSC cell lines, and we created a bunch of comparison group because by comparing DSAD and disomy, we're testing the genes associated with neurodegeneration versus trisomy. DSAD versus no DSAD gives you neurodegenerative process, and then by doing no AD and disomy, we're looking at the effect of trisomy. And she's done that, and the iPSC cell lines does what it's supposed to do. So we did 35 days and 90 days. And then this is principal component. And about 35 days, you still see cluster. And about 95 days, you see separation from those DS went on to develop AD versus those who do not, and disomy cluster together. So, summary, it says, DSAD includes expression gene associated with neurocrest differentiation starting from undifferentiated state to the, and then difference in all DS includes GABAergic primarily, and then neurodevelopment may impact neurodegeneration from these genes that we have studied. And these are my collaboration, PS1, and then Down syndrome and centenarian study. And these are the NIH funding that I got to support these efforts. And that's it, folks.
Okay, thanks very much for that. I'm sorry we're well over time, but that was a fabulous study, and looking, I think the allostatic load, or not allostatic load, the allostatic response you saw, where it's again not about the APP, it's about what else is going on. Although
that was the accent arrow that I drew.
It's absolutely fascinating, and hopefully, can I just recommend? I really can't. I'm afraid. It's go on then.
Sorry?
Why chimpanzee? Chimpanzee do not have Alzheimer.
Well, they only live for 10 years.
I've never studied chimpanzees, so I cannot tell you.
All right, well, look, thank you very much, and I'm sure Joe will be very happy to take questions later. Okay, can we move on to the next speaker, please? That's fantastic. Are you going to do this one, Sheva?
Our next speaker is Dr. Zhibek Zholgasova. She's the founder of Brain Center in Almaty. It is a center that people suffering from dementia can seek for help.
Thank you. Dear colleague, I'm pleased to welcome everyone in this hall and to congratulate organizers with this anniversary event. It is an honor for me to speak on this platform. First, a lot of people want to live long and healthy, but what we should to do for this? I'm just doctor psychiatrist, psychotherapist. 2 years ago, large family came to me. They brought their 46-year-old brother, and they were suffering from severe dementia. The family asked me only one question: why does everyone in our family die so young? None of doctors before me could answer these questions. We were able to, but I will return to this at the very end. I always say that aging is a physiological process, not a disease. For example, verbal memory should improve with age as vocabulary expands and life experience grows. The period of 2020 to 2030 has been declared the decade of healthy aging by the United Nations and World Health Organizations. Healthy aging is developing and maintaining the functional ability that enables well-being in older age. Over the past 100 years, life expectancy has increased significantly, but longevity also has another side. Aging is often associated with chronic disease, and many people live with several at once. In the first table, you see the prevalence of major disease in people over 65. Arterial hypertension in 60–80%, diabetes in 20–30%, ischemic heart disease in 25–40%, dementia 7–10%, increasing up to 30% in those over 80. The second table shows how many people over 65 live with 2 or more chronic conditions simultaneously. 2 chronic conditions are found in 65–80% of people, and 3 conditions in 30–50% of people over 75. The most common combinations are hypertension with diabetes, hypertension and ischemic heart disease, osteoarthritis, and obesity. We all want to live long and healthy lives. So what is necessary for this? According to the American Heart associations, we should follow the life essential 8. It is two group factors. First group is health behaviors, eating healthier foods, being more active, quitting tobacco, getting healthy sleep. Second group is health factors, managing weight, controlling cholesterol, managing blood glucose, managing blood pressure. To reduce the risk of dementia by 45%, 14 factors must be addressed. Physical inactivity, smoking, exercise, alcohol consumption, air pollution, traumatic brain injury, social isolation, low education, obesity, hypertension, diabetes, depression, hearing loss, vision impairment, and high cholesterol. So, dementia is a broad concept characterized by progressive cognitive decline up to complete loss of self-care skills in severe stages. There are about 100 different conditions that can lead to dementia. Alzheimer's disease accounts for 60–80% of all cases, followed by vascular dementia and dementia with Lewy bodies. In Kazakhstan, more than 13% of the population is already elderly, more than 2 million people. By 2050, this number is expected to reach more than 16%. Every 6 person in our country, every 6 person will be over 60. Today, more than 400 centenarians live in our country. If we use data from other countries as a reference, Kazakhstan should have around 200,000 people with dementia. Every year, I request data on dementia from the Ministry of Health, but the statistics are discouraging. In 2021, only about 10,000 people had an official dementia diagnosis. The number has grown slightly in recent years. If we include neurological diagnoses such as encephalopathy and post-stroke consequences, the figure rises to 46,000. In the drawing of the famous artist William Utermohlen, we can clearly see how a person gradually loses all his life experience under the influence of Alzheimer's disease. From one drawing to the next, we observe the decline of his skills and cognitive functions. Today, early detection of Alzheimer's is a priority, and many new diagnostic approaches are being developed. The most reliable results come from PET scans and cerebrospinal fluid analysis for beta amyloid and tau proteins, but these are expensive and invasive. Increasingly accessible blood biomarkers are now available, such as tau 217 and p-tau 181, neurofilament, light chain, and others. This is crucial given the development of new therapies. 2 immunotherapy drugs, lecanemab and donanemab, are already approved, with more on the way. These drugs are intended only for patients with mild dementia or prodromal stages. What is the situation in Kazakhstan? Let me start with only positive. Our patients can receive anti-dementia drugs free of charge, according to Ministry of Health Order number 75. Donepezil and memantine are included. However, there are many problems. Doctors often ignore or replace dementia and Alzheimer's diagnosis with others, which means patients cannot access these drugs. Misdiagnosis also leads to inappropriate prescriptions. Elderly patients are frequently denied disability stature or care allowances, or forced to go through psychiatry, leading to stigma and discriminations. There is no support for caregivers facing burnout. Many must quit their jobs to provide round-the-clock care. Rehabilitation is absent. Still, there are new projects and solutions. Sales of memantine and donepezil in Kazakhstan have increased more than 5-fold since 2018, though they remain insufficient. Calculations show that only about 2,600 people receive treatment, while 10,000 are officially registered. I also lead a working group that created a new national protocol for the diagnosis and treatment of dementia, incorporating all recent advances. It is now under review and approval. In Inbekshi Kazakh district of Almaty region, together with the local hospital, we launched the project Save the Memory. We trained rural nurses to use neuropsychological tests and detect cognitive decline, and trained doctors in diagnosis and treatment. In more than 2 years, we identified 743 patients out of 173,000 residents. It is more than 6% among the at-risk group aged over 50. Importantly, 83% had mild dementia, near 16% moderate, and 6% severe. I also developed a novel test for detecting cognitive decline. Cognitive decline through fine motor skills. This is a set of hand gestures that patients must reproduce. The idea is that the hand cortical representation is large and sensitive to brain changes. We applied machine learning, which recognized gestures with over 99 accuracy. My son wrote the code and trained the model. In the same program, we added hourly assistants who privately provide cognitive training, health monitoring, walking assistance, adaptive exercise, and more. And how let me return to family who asked me why they die so young at around 50 years. We collected blood samples from 20 relatives across 4 generations. The 0 patient was their grandfather who died around 50. He had 2 marriages and 8 children. 5 daughters died of dementia. Currently, 1 daughter from his second marriage is alive at 63, along with 2 sons, aged 53 and 50. We conducted 2 whole exome sequencing in Korea, which revealed a rare variant in the gene presenilin-1. Sanger sequencing in London confirmed presenilin-1 carriage across 3 generations, a rare variant with a mutation in intron 4. Family members typically developed Alzheimer's disease between 40 years and died around 50. Across 3 generations, we managed to grow fibroblasts from the skin of 4 family members, collected additional blood samples, and performed RT-PCR, which confirmed gene expression. The most important finding: 2 members in the family, 53-year-old man and his 63-year-old sister, carry the pathogenic gene but have no dementia symptoms. Their gene expression was 4 times lower than in patients with classic early onset Alzheimer's, both in fibroblasts and in blood. This means they have natural this is our fibroblast. This means they have natural protective mechanisms against the disease. We now plan to identify these mechanisms. It is our RT-PCR, blood, and fibroblast. To do this, we are conducting transcriptome studies and seeking support for proteome analysis. Just 3 days ago, I received results of whole genome sequencing of 20 family members. Our goal is not only to reveal natural protective mechanisms against Alzheimer's, but also to discover cellular and molecular targets for new drugs. With today's technology, this is possible. 2 years ago, such a project seemed like science fiction. Today, much has already been achieved. We answered the family's questions, but we did not stop there. We continue our work and seek collaborations for further research. I hope you found these presentations interesting. I'm glad to share our results with you.
Thank you very much for your presentation. We have time for 2 questions, maybe. I would like to ask my first question. Could you please tell the ethnic origin of that people with PS1 mutation which you studied? Ethnicity, are the Kazakhs, Russians, or what?
I don't know, because we need to research this.
No, I mean, Казахи, русские, кто?
Kazakh nation, yes.
Kazakh nation, okay. And we can have time for
Shymkent.
2 more questions, at least 1 question for sure. Anybody wants to ask questions? Okay, no. Then thank you very much. And our next speaker is Professor Nazira Jarkinbekova. She is the head of the Department of Neurology from South Kazakhstan Medical Academy, Shymkent, Kazakhstan. And the title of her presentation, because he is in Russian, let me translate, is Clinical and Demographic Characteristics of the Parkinson's Disease in the South region of Kazakhstan.
Здравствуйте, уважаемые коллеги, дорогие друзья. Сердечно хотела бы поздравить с 15-летним юбилеем Назарбаев университет как ведущего научного образовательного центра нашей страны. И за эти годы университет показал себя как символом передовых знаний, инноваций и международного сотрудничества. Желаю вам дальнейших успехов, новых открытий и, конечно же, вдохновения на благо науки, образования и процветания нашей страны. И я хотела бы сообщить сообщение по нашим работам, результаты. Международный проект по нейродегенеративным заболеваниям. Отари, можно там? Возьми. Можно? Международный проект по нейродегенеративным заболеваниям, и один из разделов у нас была клинико-демографическая характеристика болезни Паркинсона в южном регионе Казахстана. Что это за проект? Я сейчас немножко скажу. Так, это университетский колледж Лондона, Центральная Азия, Закавказье, проект по исследованию генетической разнообразности болезней. И это проект был в рамках PhD проекта Кайыржанова Рауана Богдановича, который защитился в UCL университете. И сейчас он является нашим доцентом кафедры неврологии при Южно-Казахстанской академии. В настоящее время он является куратором этого проекта. Этот проект входит в международный большой международный проект, называется Synapse, и посмотрите, сколько стран в него входят, и финансируется некоммерческими организациями, как Wellcome Trust и Medical Research Council. Центры, которые участвуют в проекте «Генетика Центральной Азии, Трансзакавказья», посмотрите, какие страны. Это Казахстан, Узбекистан, Таджикистан, Азербайджан, Армения и Грузия. И в Казахстане мы первыми инициировали проект «Войти сюда». Южно-Казахстанская медицинская академия в последующем присоединились другие центры нашей страны. Предмет исследований – это болезнь Паркинсона, семейные мозжечковые атаксии, наследственные хореи, дети с задержкой развития, детские церебральные параличи, эпилептические энцефалопатии и другие генетические расстройства и синдромы. Изучение генетики нейродегенеративных заболеваний в Казахстане, это вот с 2017 года мы начали. Коллаборацию Южно-Казахстанская академия начала с Институтом неврологии университет колледжем Лондона. Ну вот, видите, спонсоры проекта Wellcome Trust, UCL и Medical Research Council. И в октябре 2018 года он стал официальным хабом проекта UCL Центральная Азия, Закавказье, именно по проекту моторных двигательных расстройств. Получили предварительные данные исследования, научные публикации, и в дальнейшем мы вошли в другой проект международный Майкл Курс, Майкл Фокс, вернее, Foundation на изучение генетики болезни Паркинсона в Центральной Азии. До 2029 года мы этот проект будем проводить. Руководители проекта профессор Джон Харди, профессор Генри Холден, и наш проект именно Центральную Азию, Закавказье руководит куратор Роман Каиржанов. Ну вот в данном проекте мы что делаем? Создаем биобанки нейрогенеративных заболеваний в Центральной Азии, открытый обмен данными, обучение специалистов, совместные научные публикации, совместные международные грантовые заявки и ко-супервайзинг докторантов. Двое из моих учеников были именно в нейрогенетической лаборатории под руководством профессора Холдена. Вот в исследование также было включено изучение редких неврологических заболеваний, и более 20 неврологов из Центральной Азии прошли обучение, специализацию в UCL. Более 1000 пациентов с болезнью Паркинсона мы провели анализы, 1000 здоровых пациентов, более 80 семей с спиноцеребральной атаксией и более 50 семей с болезнью Гентингтона, 150 пациентов с болезнью Альцгеймера. Были поданы 7 научных заявок на международное грантовое финансирование. Всем пациентам с редкими заболеваниями было сделано полноэкзомное секвенирование. И вот были получены предварительные данные, которые мы докладывали на МДС в Конгрессе. Предварительные данные генетических исследований. Сейчас мы продолжаем эту работу, и докторант в этом году у меня поступил, и вот эта работа по спиноцеребральной атаксии мы будем продолжать, и болезни Гентингтона. Ну вот, что мы нашли по предварительным данным генетических, сейчас более 50 семей у нас есть материалы. А в то время мы 23 семьи, в основном это южный регион Казахстана, и нашли 9, значит, больных спиноцеребральной атаксией первого типа, 4 больных спиноцеребральной второго типа и по одному спиноцеребральной 17 и 7 тип. И сейчас эту работу мы будем продолжать, и дальше корреляцию проводить между фенотипами и генотипическими результатами генетических анализов. Далее были предварительные данные генетические исследования болезни Гентингтона. Также мы доложились на МДС Конгрессе. 60 семей с гиперкинетическим синдромом, 90 пациентов. И было выявлено, что все-таки это была в основном в большинстве случаев болезнь Гентингтона. Средняя длина CAG-тракта была 44,96, и средняя длина CAG-тракта 50 здоровых аллей, это были казахи, 17,94. Посмотрите азиатская средняя длина CG-тракта была 16,4, 16,6. А вот европейские страны, это 18,4 длина. Да? И наша, значит, средняя длина CG-тракта, она имеет промежуточное место, 17,94. Чем длиннее CH-тракт, тем высокая экспансия и риск возникновения данного заболевания. Поэтому вот в азиатских странах как бы распространенность данной патологии меньше, чем в европейских странах. Далее неврологические детские расстройства, также были приведены результаты 444 семей. В Казахстане были положительны 39%, это также у нас есть публикация по этим данным. В синем цвете показаны гены, которые впервые были выявлены, и сейчас ведется работа по этим генам. И вот тот первый у нас, наша статья, это была опубликована в американском журнале человеческой генетики, высокоцитируемом. И вот второй ряд, это наша семья, туркестанская семья. И был туркестанская семья, эти 3 детей из одной семьи, они под диагнозом состоят на учете детский церебральный паралич. Грубая задержка развития, как можно включить? Включить? Грубая задержка развития, врожденная микроцефалия, лицевой дисморфизм, эпилепсия, спастичность. И в первую очередь у таких детишек это присутствует на первом месте отсутствие зрения и слуха. Но, к сожалению, этот ребенок умер. Значит, эта семья состоит из 3 детишек, маленьких детей. И вот пробанд, где отмечена стрелочкой. И эта семья, ну вот посмотрите, 3 детей вошли в 20% риск заболевания, наследования в данной мутации. Мама не инбридинг браки с папой, да, то есть не кровно-родственный брак. Мама из Казахстана, папа из Ирана. И вот мы нашли новый ген SPATA5L1, который известен был миру, но не был связан с моногенным заболеванием. Вот такой мозг у детей, атрофия мозга и гидроцефалия. Это другой тоже пример, кейс. Это наша девочка тоже из Чимкента. Допочувствительная дистония GCH1, мутация в этом гене. И, конечно, дав маленькие дозы препараты L-допа, мы, конечно, привели ее в порядок. Сейчас она уже развивается, девочка, подросток. Мама болеет Паркинсоном. В этой семье где-то, сейчас скажу, еще 2 сестры болеют и тоже около 3 детишек. Леводопочувствительной дистонии. Ну вот молекулярно-генетическое определение болезни Паркинсона – это моногенное заболевание, где-то 5-10% – это следующее исследование у нас. Спорадические случаи от 90 до 95 случаев, да, и на 2018 год выявлено 23 генетических локусов, 19 причинных генов. И вот у нас статья была в 2019 году опубликована, где мы смотрели генотипирование именно гена ЛРК2, варианты, 8 вариантов, да, и здесь мы выявили азиатские гены и выявили европейские гены в основном. Вот первый ген, вот этот 2019, он характерен для Ашкеназии, еврейской национальности. Этого гена у нас не было. Ну вот что мы выявили РК2, две семьи абсолютно независимые, аргинин 1441, и это вот наша семья тоже в южном регионе, вот в Чимкенте, из Чимкента, смотрите, это мутация европейская, но это казах, никакой не европейской национальности, и у него вот конкретно этот ген мы обнаружили. Его семье еще младшая сестра болеет. Эта статья была опубликована в 2020 году, Parkinson Disease in Kazakhstan, клиника демографические дескрипторы, и мы выявили, что течение заболевания популяции казахской где-то в течение 7 лет, дискинезия 17%, дискинезия плюс флюктуация 14,5%, и терапия монотерапия на леводопе терапии где-то 34%. Но тоже сделали из этого выводы, и дальше мы работали. В рамках этого проекта мы решили проверить, исследовать клинико-демографическую характеристику болезни Паркинсона в нашем южном регионе. Это город Туркестан, Туркестанская область, и город Шымкент, как мегаполис, 3-й республиканский город. Вы знаете прекрасно, что этот регион, он в основном заселен коренным населением, это казахская национальность, плотность населения очень высокая и рождаемость высокая. Выявили, значит, распространенность болезни Паркинсона, оценили выраженность клинических, значит, проявлений в зависимости вот именно с определением влияния внешних факторов на развитие болезни. И именно в нашем регионе мы создали, валидизировали и утвердили казахскоязычную версию шкалы МДС-ЮПДРС и МДС-ЮДСРС. Разработать и внедрить опросник для выявления болезни Паркинсона на разных стадиях и провели цифровизацию диагностического поиска. Были взяты когорта Туркестанской области, города Шымкента, и были выбраны больные в соответствии, значит, критерии МДС 2015 года. Что мы выявили? что в Туркестанской области 67 случаев и в городе Шымкенте 65 случаев на 100 тысяч населения. То есть мы отсеяли тех больных, которые обладали еще паркинсонизмом плюс или синдромом сосудистой деменции. И получается чистая болезнь Паркинсона, именно идиопатическая болезнь, это 65 случаев или 67, это Туркестанская область. В основном было высокие цифры, это в городе Житсай и Махтаральском районе. Мы, конечно, удивились и начали дальше исследовать. Почему? В 64 случаях 450 пациентов превалировали пациенты именно из села. Женский пол больше преобладал и составил 57,4 и 55,2 соответственно. Далее, общая характеристика, значит, возраст дебюта заболевания в городе составил 58, в селе раньше 56 лет, самый ранний дебют наблюдался 27 лет, поздний 80 лет, и достоверно выявлено, что у мужчин дебют заболевания приходится на 60-69 лет и женщин на 50-59 лет. Коморбидность была очень высокая с артериальным гипертонием, и мы с вами прекрасно знаем, что сосудистые факторы, они приводят к усилению нейродегенеративного прогрессирования, заболевания именно нейродегенеративного. Далее травмы головного мозга и, конечно, сахарный диабет и анемия. Ну вот клинические формы болезни Паркинсона в основном в городе и в селе превалировала акинетика ригидно-дрожательная форма. По стадии Хины-Яру в городе превалировала первая стадия и третья стадия, на селе первая и вторая стадия. Далее по осложнениям течения заболевания леводопоиндуцированные дискинезии развивались через 6 лет. От начала заболевания у городских 5-8-3, а у сельских 6-11. И отмечалось, у женщин дискинезии отмечались позже, чем у мужчин. При корреляционном анализе определены были достоверно значимые взаимосвязи между стадиями по Хины-Яру и 4 разделами шкалы УПДРС. Это мы ввели эту методику определения шкалы в своем кабинете двигательных расстройств. И среднее значение УПДРС, именно вот достоверно значимую связь имела третья часть шкалы, моторная шкала. И чем больше было баллов в этой шкале, тем было, конечно, выше стадия по Хины-Яру, и это были очень тяжелые больные. Далее, анализ используемых лекарственных препаратов на терапии леводопа 70%, без терапии 25%, на терапии другими противопаркинсоническими препаратами 4,9%, и анализ, мы проводили корреляции между леводопотерапией и наличием дискинезии, показал достоверно значимую связь между, значит, где-то у пациентов городских жителей 41%, у сельских 45,5%. И на терапии другими препаратами дискинезия возникала очень, посмотрите, редко, 1,6%. Анализ факторов риска болезни Паркинсона показал, что проживание в сельской местности и употребление колодезной воды по сравнению с водопроводной связаны с ранним дебютом заболевания по сравнению с городским. И это результаты множественной линейной регрессии показали, что проживание в селе достоверно увеличит риск раннего дебюта болезни Паркинсона 2,47 раз, и как употребление колодезной воды увеличит риск раннего начала в 3,85 раз. Ну вот это потом, когда мы проверяли, мы спросили, конечно, это Махтаральский, Житсайский район, там у нас хлопковый промысел, и землю они удобряют вот этими пестицидами, точно вот этот список пестицидов показали, и оказалось, да, этими пестицидами удобряют эту землю, вот хлопкоробы. И колодцы, очень часто колодцы на этой территории находятся, и колодезная вода как раз-таки она оказала, мы так думаем, наша версия, что на ранний риск дебюта заболевания именно в этих регионах. Одобренные обществом двигательных расстройств МДС являются официальными версиями шкалы МДС ЮПДРС, МДС ЮДИС и РС, и доступны на сайте общества двигательных расстройств. Вот это та группа группа из Казахстана, которая создала, валидизировала эту шкалу. И вот публикация в высокоцитируемом журнале «Валидизация казахской версии», вот казахской версии шкалы, вот как раз-таки сейчас она помещена на сайте. Именно эта версия шкалы казахской языка. Разработка опросника для выявления болезни Паркинсона. Это наша разработка уже. И всего 128 пациентов мы раздали по поликлиникам, так как я являюсь главным неврологом Туркестанской области, являюсь куратором города Шымкента, как академический наставник. И мы вот, конечно, помогаем и по поликлиникам раздали этот опросник, чтобы выявляли болезнь Паркинсона.
Excuse me, sir, but you are already only 1 minute left, and for questions, will no be time for questions, so you go to hurry up, please. Sorry.
Все. Вот наш биобанк. Мы хотели показать коллаборацию. Вот наша коллаборация до 2023 года. Мы работаем, и вот эти наши планы. Спасибо за внимание.
Спасибо большое за презентацию. И да, мы все-таки имеем время на 1-2 вопроса. Пожалуйста, коллеги, у кого-то есть вопросы, если, задавайте. Вопросов нету, да? Okay, then...
Yes, it's my pleasure to welcome Professor Nurgozhin, who's going to tell us what we should be eating tonight to stop us dementing. So I'm very excited by that. I hope it's going to include horse meat.
Yeah. I will recommend it.
Thank you very much.
Это работает? Тогда я включу. Да. Добрый день, дорогие коллеги. Разрешите дать поработать транслитером, чтобы переводили. Поэтому у меня будет сегодня презентация. Вопрос связан с питанием и вмешательством, профилактическим вмешательством терапии деменции. Так. У меня не поменялось здесь. Да. Ну, вы прекрасно знаете, я не буду комментировать много данный слайд. Примерно по данным Всемирной организации здравоохранения 50%, 50 миллионов, около 50 миллионов населения страдает деменцией. К 2050 году примерно планируется удвоиться эта цифра. Есть возможности профилактики, и мы это хорошо знаем, около 40% деменции можно предотвратить. Это путем психоэмоционального состояния, образа жизни и питания. Так, у меня вот это. Наш проект связан именно вопросами, связанными питания, психоэмоционального состояния и здорового образа жизни. Да, нами был выигран грант Министерства науки и высшего образования, распространенность факторов риска и основных триггеров возникновения деменции в Казахстане, включая разработку нутритивных подходов к профилактике. В рамках данного проекта мы обследовали тревожность, депрессивные симптоматики, особенности питания и потенциальный риск когнитивных людей у пожилых людей. В исследование вошло более 1000 респондентов в возрасте от 45 до 90 лет, средний возраст 67, и оценка психоэмоциональных состояний мы прошли по шкале МОКа, HADS, и анализ пищевого поведения опросника составлен потребности FFQ. Сама методология здесь тоже хорошо описана. Это клинико-психологические методы, эпидемиологические методы, генетические методы, клинико-психологические методы, экспериментальные методы и статистическая обработка. Оценка самого распространенности, неправильно сделал, да? Хорошо видно, вообще, оказывается, красочно нельзя делать. Оценка распространенности модифицированных факторов риска, основных триггеров. Вот здесь в ходе работы выявлено, что примерно легкой степени страдает около 20%, средней степени страдает чуть больше 60%, и тяжелой степени деменции страдает около 20%. Таким образом, основная часть пациентов с деменцией примерно составляет 80%, и они под наблюдением специалистов уже в стадии средней и тяжелой, выраженной когнитивными нарушениями. Психоэмоциональное состояние, вот результаты показывают, что примерно старше 80 лет выраженная симптоматика у каждой трети респондентов. Также был подтвержден это как мировыми эпидемиологическими данными, и подчеркивает важность здесь – это тревожность, депрессия в пожилом возрасте. Это часто сопровождает именно психоэмоциональное расстройство и еще факторами, основной фактор риска деменции. Здесь мы показываем когнитивное психоэмоциональное состояние исследованных наших групп. Состояние респондентов мы разделили на 3. 45-54 – это те группы, которые не страдают, а 55-64 легкой степени, выше 65 лет уже средней и тяжелой степени. Здесь показываем оценка немодифицированных факторов риска деменции, каково питание этих людей, и мы связали, что питание и психоэмоциональное состояние близко взаимосвязаны. Люди, которые страдают психоэмоциональным состоянием и депрессией, плохо питается или употребляет больше углеводов, нежели овощи, а также не ведут здоровый образ жизни. Вот здесь мы показали питание пожилых людей, примерно 45% ежедневно употребляют овощи и фрукты, 30% из них 1-2 раза в неделю реже, 30% также регулярно едят рыбу, морепродуктов, 15% примерно практически полностью исключают рыбу. Женщины старше 70 лет, живущие однообразное питание, основным это хлеб, каша, чай и минимум белков и овощей. Мужчины чаще используют мясо, редко рыба, и 25% респондентов в основном употребляют сладости. Здесь также показан риск когнитивных нарушений, высокий уровень, умеренный, низкий, сколько ключевые показатели среднего потребления, один из главных факторов риска, сахара, тревожное расстройство 36%, депрессия 33%, и когнитивный статус, норма 28%, легкие нарушения 29%, деменция 42%, и 61%, чуть больше 61% страдает деменцией и диагностирован сопутствующие состояния депрессии и дефицит питания. Особенности питания, здесь мы еще раз хотим показать, что ежедневно употребляют они овощи. Здесь я еще раз хочу сказать, что основным это мясо, сахар, колбасные изделия, морепродукты крайне редко, бобовые субпродукты тоже недостаточно в рационе, овощи и фрукты потребляются недостаточно, и таким образом рацион пожилых людей насыщен продуктами высоким содержанием насыщенных жиров, соли и сахара, нет других питательных веществ. Здесь мы показываем связь психоэмоционального состояния и питания. Наиболее важный вывод, фактически формируется порочный круг Депрессия приводит к ухудшению питания, а плохое питание усугубляет депрессивное состояние и проявляется когнитивными нарушениями. Нами был предложен механизм защиты мозга. Да, мы подобрали 5 групп продуктов питания, которые содержатся омега-жиры, кислот, антиоксиданты, витамин С, Е, полифенолы, витамин группы В, и также пробиотики и магний, цинк и селен. Для чего они были расписаны? Вот функциональное питание на основе полученных данных, такие меры уже доказали свою эффективность, сочетание диеты и когнитивными тренингами контроля сосудистых факторов, что позволило когнитивные нарушения, ухудшение пожилых людей из группы риска. 28-дневные профилактические питания. Это практическая модель функционального питания. В рамках 28 дней лица, которые будут управлять, это простота и доступность меню. Блюдо готовится из недорогих первичных продуктов. Функциональность, профилактическую направленность питания, баланс питания, а также поддержка когнитивного здоровья. Это включая психоэмоциональное состояние больного. Сама структура меню вот, нами была предложена такая. Вот на завтрак каша, молоко или вода. На обед это суп овощной или с крупами. Рыбы, бобовые продукты. Полдник кисломолочные продукты. Ужин запеканка, тушеные овощи или рыба. И перед сном стакан кефира или травяной настой. Меры профилактических акцентов. Вот 2 раза в неделю обязательно включение рыбы, каждый день овощи, фрукты, не реже 2 раза в неделю бобовые продукты, регулярно цельнозерновой хлеб. И также, конечно, это включая грецкие орехи, фундук, а также семена тыквы и другие подсолнечные и льняное масло. Ограничение сахара и жиров и солености. Значение самого меню – это нормализация психоэмоционального состояния, снижение сосудистого риска, поддержка микробиоты кишечника, прямое нейропротективные действия, следствие применения тех групп пищевых продуктов, которые я назвал, это омега-3 жирные кислоты, антиоксиданты и другие. Таким образом, 28-дневное меню представляет собой практическая реализация концепции функционального питания, которая позволяет снизить у пожилых людей устойчивые пищевые привычки, снижает риск когнитивных нарушений, обеспечивает доступ к правильному питанию и широких социальных группах. Практические значения данного исследования, результата нашего исследования, это психическое здоровье, и питание нельзя рассматривать как изолированно, поэтому функциональное питание может стать основным инструментом профилактики деменции. Мы уже показали, примерно 40%, кто страдает деменцией, можно методом профилактики или отсрочить, или можно таким образом привести к определенному уменьшению риска. Сама оценка роли нарушения питания, формирования, это нами вот было тоже разработано, коррекция рационов питания. В связи с этим мы разработали специальные продукты, биологически активные добавки питания, вот аминокислоты, которые содержат L-тирозин, полифенолы, антиоксиданты, ресвератрол и другие нутриенты с доказанным нейропротективной эффективностью. И таким образом мы подали на патент на этот продукт, специализированный пищевой продукт для профилактики деменции. Наши выводы – это 3 основных ключевых мысли. Это психоэмоциональное здоровье, функциональное питание и 28-дневная программа. Это социальный рабочий инструмент для клиники социальной медицины. Это реальная возможность сохранить качество жизни у больных. В заключение я хочу сказать, что разработка и внедрение программ функционального питания – это не просто дополнительное направление в стратегическом задаче здравоохранения 21 века, но это основной ключ качества и образа жизни людей. Спасибо.
Thank you very much. So it's fish on the menu tonight, is that right? So I'm happy to, it's perfectly on time, so there's room for questions now.
I don't know the special menu for the dinner, but I know you will eat horse meat.
Well, so I'll start the questioning, because one of the interesting things about sickness is that, is that your diet often, you change your diet when you're sick. So the interesting thing is that somebody who's dementing, and let's say that they're losing weight, potentially, when you become sick, there is a shift towards high protein, high fat diets, and the reason that is to usually, if you're sick and fighting infection, this is to sort of provide the necessary fuel that your body has decided you need. So I wonder, when people make these life choices about what they want to eat, is this actually being driven by the pathology demanding these foods? And so, I won't deny that it seems like a good idea to sort of take, oily fish as something that's going to be anti-inflammatory, etc. And we already heard how there's that angle of inflammation in Alzheimer's. But how much do you think the pathology is driving that desire for that food?
For the dementia, who have a dementia, they never control it, what they will eat.
Themselves?
Themselves.
Okay.
They never control it, because sometimes we know who have a dementia high risk, and they have only 2 functions. Is the eating, is safe, and second, and sex. This is 2 function is the only who have a high risk of dementia.
Right. So really, it's not. So it's probably that's because they have little control over that. That gives you the opportunity to. And then the only other question I had for you is, in the general population, the difficulty is the diet you were describing can often be seen as an expensive diet. I mean, meat is expensive, but it's interesting. But in England, for example, fish is expensive.
Absolutely, you're right. Fish is expensive, but you have a control on not only fish, but you eat meat. Another one is option for the change the fish, and also we recommended. Not the highest price. It's products. We recommended cheapest one.
Yeah.
And second one, most important, the safety, psycho-emotional status of the patient. And second, control of your life. And third one is the control your diet.
Absolutely. That's very useful. Right, and so I don't want to monopolize. Do we have any other questions from the floor? Do you know, in that case, I think what we'll do, bearing in mind that we've got, everybody wants to go for dinner, I think, but we do have one last speaker. So
Exactly.
Thank you once again for your talk. That was lovely. And I should also say, bloody good simultaneous translation. I can't quite believe how good that is. But anyway, what I'd like to do now is welcome Chopin to her own institution. It's a pleasure to have you here. I'm glad you can make the journey. And we're going to, there it is, it's up there. And we're going to hear about more Alzheimer's disease. Yes, so thank you very much for that. And let's get on.
Finally, final speech for today. Yeah, we're happy. Everybody's happy. We're happy too. So anyway, let me start. It's kind of hard to be the last presenter during the session. Yeah, this is for me. That was already discussed. What was already discussed? What should I do right now? And I decided, instead of showing slides, to tell you the story, basically the story of my life, and I explain why. I do it. So basically, I'm involved in Alzheimer's research disease since 2006. Then I started my PhD study in United States, and back to that time, I did that in a cool cell membrane biophysics things, cool cell downstream, upstream cell signaling, and blah, 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 et cetera, which is related to Alzheimer's disease, yes. After I came back to Kazakhstan, of course, I expectedly continued that direction in my research, and that in one day I started noticing that my mom is, acting strange. She changed her behavior, and I for a long time I ignored it. My thought was, well, Mama is just getting old, and it's normal. She's getting childish because she's getting old, and it took me honestly 2 years to realize it's something wrong. It's not it's aged, it's something pathological. And after that, I started seeking for the help. I brought her to the physician, to our family doctors, and what they said, she's old, probably it's because she had high pressure, blood pressure. They said it's a cerebral vascular encephalopathy, like Dr. Zhubov-Zhudasova just mentioned here, and say you can do nothing. They just prescribed her vitamins, and they didn't send her to neurologist, and just said, this is, you can do nothing. That was the answer to me. And I started looking for their help, and I found neurologists who could really diagnose her, and then, what I realized that something is really wrong. One of the tests is the clock drawing test. And my mom is still, she can talk to you, she still remembers. It's not like crazy, like, not remember, like, but she could not draw that clock. She could not do it. And then she started getting nervous, and, she put away the pencil, and at that moment I realized, yes, this is true. My mom has dementia. And that was one of the moments when I decided, put my face to what the people was, instead of doing that, interesting, but I believe at the moment useless stuff in the cells, I turned to the real people. I decided to help them. But how can I help them? I'm not a medical practitioner, right? I'm not a doctor. I'm just a scientist. So I could help only by doing science and by trying to, attract the attention by doing science. And basically we started that, and we started recruiting people with Alzheimer's due to dementia. And we faced problems, like I already said, and it's extremely difficult to perform that study in Kazakhstan. First of all, low national priority, people already mentioned that. Again, cultural factors, even me, a person who is involved in disease at that time I was involved in that study already for more than 5 years, maybe 7 years, and I didn't want to believe, I couldn't recognize. I thought, wow, it's normal. She's getting old. And the doctors doing the same way. Doctors not only in Kazakhstan, this is, I think, worldwide situation. Doctors usually treat that pathology like not pathology, but a norm. But it's not norm, actually, we know it, right? But and again, in Kazakhstan, we have what? We have resource constriction. We have very limited number of neurology specialists who could basically do diagnosis and do some recommendations. And I think half of them we have here this audience for sure. And besides, we have limited capacities for diagnosis, etc., etc. It's all what's, of course, bring a lot of difficulties to do study here. And finally, this leading to global underrepresentation of our country and the entire region in the global studies, which basically Alzheimer's Disease Association constantly states that there is lack of research from Central Asia region, including Kazakhstan. And now that are all the reasons. And I faced, I can tell you from my own life, I know what I'm talking about, absolutely. And about future, when you face, without any support from government, from anybody, and I went through all that stuff, so I know it from first hand. But okay, and after that we started our study. It was case control study. Basically, it was not easy, I already explained it, but we did something. At least we did something. And what we did, basically. We analyzed some clinical characteristics, we did some genetics, we did gut and oral microbiome assessments. Basically, we collectively recruited around 400 plus patients and healthy people with same age and same gender, but without signs of dementia. And we found some variables which are statistically different from in our AD group from control group. That were smoking, depression, stroke, incidence in the health history. Then we found liver dysfunction. It was not like really dysfunction, but we found that certain parameters were below or up compared to healthy people. We also found some, HDL-C increase, which is many factors already were explained in other groups. We also found that ALT drop and triglycerides drop. So which was not explained, but we think that it's because of malnutrition and vitamin deficiency, and basically our study of dietary patterns of our patients confirmed that data. What we found that in our group of AD people, basically there is a significant and sufficient consumption of almost all the major macronutrients and micronutrients. And besides, there's, but it's kind of, it's logic, because people with dementia, they really start eating very small amount of food at certain point. It's expectable. But what is interesting, that group of old people who are not demented, basically the diet is far away from the diet which is considered to be healthy for brain. And this is basically a normal pattern for our traditional, let's say, food, at least as a food for older people. So what we notice is no difference basically, like in what they eat. The difference only how much they eat, that's it. Then what we did also, we compared Almaty. So basically what I need to say that we recruited our patients only in 2 big cities of Kazakhstan, Almaty and Astana. And why we did it? Because that cities, at least they have, some facilities, and they have trained specialists who could help us with recruiting, with diagnosis, and basically the 2 cities, Almaty and Astana, and we found regional differences. They are not that huge, but at least we found that in Astana region, stroke was more prevalent among patients, while in Almaty we observed like some glucose metabolism disbalance, yes? And what we also found why I mentioned that. Further, we investigated ApoE4 polymorphism. As you all know, that ApoE4 polymorphism is found around in 40% of the people with Alzheimer's disease, with late onset Alzheimer's disease. And as expected, in our cohort, it was like similar with global reports, 25 people, 25% people were with ApoE4, at least one allele of ApoE4, and it was twice higher than control. But when we went back to compare Almaty and Astana, we found that in Astana, this percentage is higher than in Almaty, significantly higher. And we found the papers that it's not only our finding, there is data indicating that this polymorphism increases from south to north for some reasons. This is like we observed too. What next? Next, we studied gut microbiome. We compared gut microbiome composition in our patients compared to healthy controls. And of course, we found huge difference. Again, as already was reported, we saw the increased number of pro-inflammatory taxa and decreased number of, like, let's say, healthy taxa like bifidobacteria, etc. And we also found that the level of dysbiosis was correlated with disease severity. In certain bacteria taxa we found in like middle or late stages. So we found that already reported basically. Here I just, let me skip that slide. I wanted to explain the connections between brain and gut microbiome, but I think Daniel already started this journey. And basically what's the main notion right now, that gut microbiome can influence brain functions, the modulation of systemic inflammation. Different pathways, but it's systemic inflammation. And we investigated cytokines profile in the blood of our patients, and we found, of course, as expected, difference in pro-inflammatory cytokines that was high in Alzheimer's patients. And but we also found the strong correlation between certain cytokines and certain bacterial taxa, specific bacterial taxa that we found too. Next, we also assessed oral microbiome composition because there is data indicating that oral health also can affect brain health for some reasons. For some the same reasons like gut microbiome, I guess. And we found, yes, again, we found a difference in healthy people and people with Alzheimer's. And we also found region-based difference. In people with Alzheimer's, so Almaty, oral microbiome was different from people from Astana. And next, this is our pilot study. We performed case control GWAS study. Professor Jansugourova already reported data that is from our cohort, but the difference is that in her cohort she reported all the ethnic groups, like Kazakhs, Russians, and all. And here in that cohort, we selected only Kazakh people with Kazakh ethnicity, and of course it significantly reduced our sample size, but results were slightly different. We identified 28 SNPs, and most of them were non-coding regions, but several were from coding part, and none of them were previously reported in the GWAS studies before, but they were found in the genes which already been reported to be associated with Alzheimer's disease in our studies. Those are genes. That data not published yet, and I don't know if you publish it is that number, but we'll try. So that's conclusion. Let's go to conclusion. Conclusion is this first cohort comprehensive report in Kazakhstan. Yeah, but what I want to say, also I made my title like so attractive, like Alzheimer's disease is Kazakhstan. It's basically not in Kazakhstan. As you mentioned, it's Almaty and Astana, small cohorts, and we found regional difference as well, but again, that two cohorts. Certainly, there is limitation. Case control design, small cohorts from single urban centers, and limitations and difficulties already explained to you. And so why am I telling that? Of course, we published all the data, but I say it is not something like really conclusive. I would say this is something pilot, that they're doing something, we're showing something. And what is the next? And the question, what's next? Next, we need to, it was intention to invite here all the people who almost all people who I know who involved in dementia study in Kazakhstan. And also we have here brilliant speakers, brilliant scientists from abroad, like Professor Daniel Anthony. He's a great expert in brain health and metabolism. Professor Lee, he will explain, he's a renowned geneticist. He has experience working with countries with limited resources like our country. And we have people, doctors who could have access to the patients. And I think it's time to us, instead of doing small researches, to unite, maybe create one national program, try to write that program, and it's just proposal, for your consideration. And after that, go to and start knocking to the government officials and say, hey, we need that program. We need to find out the epidemiology of the dementia and other neurodegenerative disorders in Kazakhstan. We need to know the genetics. We need to know all the factors, right? And after that, like you remember, idea just came to my mind. Our minister today mentioned AI is cool, AI is live, blah, blah, blah. I say, we can collect the data and let AI analyze it. And give us, national dementia plan. I'm not joking. I really think that we can do it. But AI will never collect that data, right? We collect that data. But we can ask the ministry to help analyze the data and tell us how to help, what government should do. And basically, that's it. Because I didn't stop on my results, they're almost all of all published, you can find them. So, in his acknowledgement, I would like to thank you, my colleagues from Laboratory of Microbiome, Laboratory of Bioinformatics, Professor Jan Sugura from Almaty, and my colleagues from Astana Medical University, and Ministry of Education and Nazarbayev University for supporting my research. Yeah. It was combined 5 grants maybe, it's not a single grant. We tried to collect here. We did what we did. At least we did something. Thank you for your attention.
Shaban, thanks so much for that. That was spot on time and very interesting. I was always told that Almaty was a nice place, but I now realize that the depression rates were 60% amongst the AD sufferers in Almaty versus 20%, I think, here. So Astana is obviously a nicer place to live, or is it some other reason like diagnosis?
You know what? Again, you should look at our results with caution. I already mentioned this small proportion people, which is selectively collected from. That was not like it was not cross-sectional study. We didn't go and study everybody. It was just medical doctors who patients come to visit and they collect information, questionnaires. So that's why I said we will have a national plan to do that in population-based, to do some conclusions, really.
Yeah, I think.
But for me, Almaty is a nice city. It's not depressive at all. Answering your question.
That's right. But I think it raises the difficulties associated with these sort of studies and reporting and how and different centers adopting different approaches to how they do that. But I think looking at your study, though, I think that it raised all sorts of fascinating questions, really, about sort of how these. I'll ask the question in a different way. I remember when I was young, I went to see a neuropathologist called Margaret Azeri, who's really she's still going, but retired several times and keeps coming back. And Margaret and I were sat at the microscope, and she said, Daniel, there's no such thing as pure Alzheimer's disease. We ran this interesting program called the Optima program, and where people longitudinally were studied, and then at the end they donated their brains. And she said, I haven't seen, its diagnosis of Alzheimer's disease post-mortem, look at the brain, what's this person got? And she said, out of this cohort of literally hundreds of people, there was nobody who had, well, that's not exactly true, but there were virtually no people who had pure Alzheimer's disease. And I think your study raises the question that the thing that we have to be aware of is how to deal with comorbidities, and whether that, when we're looking at intractable diseases, the comorbidities we have to be quite clear about, and then maybe those comorbidities we need to think about how to study those. So I just wondered if you'd comment on sort of the existence of comorbidities in this population.
Yes, you're absolutely right. For example, when I'm going back to a story with my mom, I still don't know was it Alzheimer's or was it vascular dementia, because she suffered from high blood pressure. I don't know, honestly. And going back to our patients, what we see most of the time in our emergency, first of all, it's late stage, yeah? And I think most of the time it's mixed. It's mixed, really. And regarding to how the definition of Alzheimer's disease right now, A-beta plaques or no A-beta plaques. But in a lot of cases, some people with plaques, they don't develop dementia. You know that, yeah? Probably. So it's not if you have plaques in the brain, you have dementia. No. People live with plaques without dementia, but problem is if you're healthy, you die, nobody will open your school and check, do you have plaques? No, right? So honestly, I think we don't know the problem with Alzheimer's disease and dementia, why we still don't know what to do. We don't know what is it. What is the reason? Only in a few cases when there's a clear genetic predisposition, for most of the cases, nobody knows what's really happened, right? And comorbidities, of course, they play, I think, huge role in that situation. Did I answer your question?
Yeah, well, I think it does, and you also highlight, when you refer back to your own mother, I think that these are real problems. In the UK, there is a target to try and give 2/3 of individuals a real dementia diagnosis. But right now, the UK we record dementia without saying what it is. And this is very problematic from the point of view of developing new therapies. It's very problematic from actually even, without being able to establish. When somebody says, I can tell you that this person has Alzheimer's disease, I'm sorry, until their brain is on a slab, you don't actually know precisely what burden of pathology they have in their brain. And so we are it's a rock and a hard place, but I think it raises some interesting questions, and we have to better understand, we have to have better biomarkers. Now, sorry, I've waffled on too long. We all want to go to dinner, but please, we do have time for a couple more questions.
One question.
One question.
Speak up then, just one question, please.
Hans was fastest, so you can go.
If I listen well this afternoon, then genetics is not helping us very well. Lifestyle is not helping us very well to early diagnose Alzheimer. But the necessity to have an early intervention is there because aging population, that was told yesterday. And I think, is it not time to go back to basics? Start in a way on specific symptoms like smelling, disturbances in taste, as a beginning and asymptomatic early signal of dementia or Alzheimer, and then follow that cohort until more complaints comes. Because our difficulty in Alzheimer treatment and Alzheimer intervention is that we are always too late. We are too late. People are over the hill, and then we have discussion on the ethical side. What do you think? Is it possible to do such a thing?
You mean early detection?
A simple cohort follow-up by identifying on simple parameters and follow that cohort in the healthy stage, because they have still normal functioning.
I think such studies are already in place. No, Hans, such studies are already in place. Right now, Irene can answer this question better than me because she's an epidemiologist. But there is studies in several countries, they start observing people from the early 40s. And right now, that cohort, she's reaching, I think, the 60s, yeah, Irene? Do you know that cohort of the people who observed 30? They're reaching their 30s, and we'll see that study is going on. But I don't know when they finish the study, we'll be able to access it. We'll be already in that stage. Hopefully not. I'm just joking, hopefully not. I hope we will be healthy and with healthy brains. And I wish all you guys stay healthy and physically and mentally and live long life, happy life. Thank you.
Thank you.
On that point, we're closing today sessions, and please our guests and our special guests and our speakers, please proceed to the bus. It's already waiting, and we need to go to to the hotel right now.
Can I just say thank you to all the speakers and to the audience for staying around. So can we have just another round of applause for all the speakers this afternoon and for their lovely talks. Thank you very much, everybody.
Thank you very much.